Good morning, everyone. We're delighted to have you all here. May I request all of you to please turn off your cellular devices, and for those joining online, to please put your devices on mute, as we'll be starting momentarily.
Good morning, everyone. I, Kamli Priya,
And I, Iba,
extend a warm welcome to all the dignitaries, guests, faculty, and colleagues who have joined us to celebrate the 15th Foundation Day of THSTI, a day to celebrate our successes and work towards our challenges. This annual event makes us to cherish, acknowledge, and appreciate our teamwork and efforts at THSTI. In addition, today marks the momentous quindecennial year in our mission, which is integrating the fields of medicine, science, engineering, and technology into translational knowledge and making the resulting biomedical innovations accessible to public health to improve the health of the most disadvantaged people in India and throughout the world. Let's do our level best in achieving our mission with the spirit of oneness and togetherness.
I would like to extend a warm welcome to the scientific colloquium, where the air is electric with intellectual curiosity and the hall echoes with the exchange of groundbreaking ideas. It's a convergence of brilliant minds from diverse disciplines, where each participant is a beacon of knowledge and innovation. Presenters armed with meticulously researched findings unravel the mysteries of nature in eloquent presentations that weave together data and theory with the finesse of a master storyteller. We had a senior PhD student scientific colloquium on the 11th and 12th. It was evaluated by an esteemed panel of external experts, and the top 5 presenters will showcase their work today. Now, we'll start with the first presentation. I would now like to call upon the stage Samriddhi Mehta from Dr. Milan Surjit's lab to give a presentation.
Good morning, one and all. I am Samriddhi Mehta, as introduced by our comparer. My today's talk is going to be about development of a potent antiviral against SARS-CoV-2 by targeting the interaction between nucleocapsid protein and its viral genome. Now, as we know, SARS-CoV-2 has had a good heavy load upon our population. We have had a lot of deaths. We have had a lot of hospitalizations. And because of its kind of effect on our population, it was declared as a pandemic in 2019 with a good death toll. I should not be calling it good, with a heavy death toll and a great deal of hospitalization. Now, as far as available treatments are concerned, there are tons of medicines that are already there in the market that are being marketed. Some of them are FDA approved, some of them are repurposed, and others are in the clinical trials. I'm going to delineate a couple of them over here. Remdesivir, it's an analog. It's a nucleotide analog. Then we have a concomitant Paxlovid treatment followed by molnupiravir and a couple of other repurposed FDA drugs, fluxosamine, ivermectin, metformin. And then there are a couple of 6 to 7 drugs that are already there in the clinical trials. Then since we already have so many drugs that are already treating SARS-CoV-2, why develop another drug? Well, most of them, they've got a lot of side effects. Apart from that, there is a huge deal of recurrence in case of SARS-CoV-2. Thus, we need a drug which can solve both our purposes. It can not only treat with least amount of side effects, but also treats without any case of recurrence. I would like to briefly introduce you with the genome of SARS-CoV-2. So here we have around 16 non-structural proteins, 4 structural proteins, and around a 30 kb long positive RNA genome. my talk is going to revolve around nucleocapsid protein. So, nucleocapsid protein is a multivalent protein, and my- it it is around 419 amino acid long. That should be around 48.5 kilodaltons in weight. Now, nucleocapsid protein is known to interact and perform a lot of biomolecular functions, which include replication and transcription. It helps in chaperoning the RNA to different subcellular compartments. Uh, Nucleocapsid protein has already been reported to restrict the host immune response, shows cellular apoptosis in cell cycle, modulates it, and it also modulates the overall metabolism of an infected cell. Now, because nucleocapsid protein is a very important protein, and there were already reports that nucleocapsid protein tends to interact with the viral genome. So we started with looking for segments that would be critically interacting with the nucleocapsid protein. We characterized the protein, and we did a interaction assay. It's a East3 hybrid, where we have cloned 5' and 3' UTR in respective plasmids, and checked if at all they're interacting with the nucleocapsid protein or not, followed by a pull down assay. It was a rapid assay and a EMSA. We observed that 5' UTR is effectively interacting with nucleocapsid protein. This is in vitro assay, just in order to validate what we observed in vivo studies. Now, the next part of our evaluation was to look for the critical segment, which is actually interacting with the protein. So we made overlapping mutants ranging from 50 to 100 nucleotides, and we observed that 51 to 150 base pairs of 5' UTR is essentially interacting with the nucleocapsid protein. So now we have the critical binding region. The next part was to analyze what exact residues, to pinpoint the exact residues, the nucleotide residues as well as the amino acid residues that are present in the 5' UTR and nucleocapsid protein respectively, that are actually participating in this interaction. So we did a computational study. We did a docking, a rigid docking, and a simulation assay. And we observed that R89, 92, 107, Y109, 149 are the essential residues in the nucleocapsid protein that interact. And simultaneously, nucleotides at position 78, 79, 90, 91, 121, 123, and 124 participate in this binding. They are directly interacting in the pocket. So this was a computational analysis. We validated it using an in vivo study. This is again a Y3H data where we have made the respective mutants and then checked if at all there is any effect on the interaction with the wild type protein. What we observed was RM10, which has got all 7 mutations together in its sequence, does not interact with the nucleocapsid protein at all. It's not diminished, it's completely absent. Next, we did in vitro binding assay. We wanted to know with what kind of affinity are these mutants binding to the nucleocapsid protein with respect to the interaction with the wild type 5' UTR. And we observed that the dissociation constant was highest in case of RM8 and RM10. So RM8 has got 4 mutations. It's at 78, 17, 78, 79, 81, 80 and 81 position, whereas RM10 has got all 7 of them. It also has got mutation at 121, 123 and 124 position. They do not interact with the nucleocapsid protein at all. Now, the next part of our study was to analyze what will happen if we mutate the residues that were identified through computational analysis for the nucleocapsid protein. What happens if we mutate them? So here again, what we observed was that even if you mutate a single residue, the interaction almost gets to nil. Although the mutation, the mutant which had got 6 mutations together at 57, 59, 105, 107, 171, and 172, this shows the best result. When you mutate all these 6 residues together, you do not see any interaction at all. Now, this was about the theoretical background, the backdrop of the interaction that we were studying for 5' UTR and nucleocapsid protein. it always comes down to what is the functional basis, what is the functional significance of anything that you're looking at in a scientific perspective. So we wanted to know what was the functional significance of this particular interaction. Now, nucleocapsid protein is known to phase separate. Most of the molecules, when they want to perform some function inside a cell, of course, they are not, they're simple molecules, right? If they want to perform a function, they'll have to come together. Now, these are membrane-less or subcellular structures which are present in the cytoplasm or nucleoplasm along with the adapter molecules, and they're doing some or other kind of biomolecular function. The nucleocapsid protein is known to phase separate. Basically, it becomes relatively more dense in comparison to its media, the cytoplasm or the nucleoplasm. When nucleocapsid protein interacts with 5' UTR, it phase separates more effectively. The smaller particles become more larger in size. Now, this could imply two things. This phase separation could be aiding in the packaging of the virion, as we know that M protein, the membrane protein with other structural proteins of SARS-CoV-2, we know that a nucleocapsid protein interacts with M protein. So either it is helping the virus in packaging, or basically it is helping the entire transcription complex to associate around the viral genome, so that genomic and subgenomic RNA particles can be produced. So looking forward in this direction, we performed a phase separation assay. This is a liquid-liquid phase separation assay followed by a confocal imaging. What we wanted to check was what happens if we mutate the residues in nucleocapsid protein and the 5' UTR. Will there be any change as far as this interaction is concerned in perspective of liquid-liquid phase separation? What we observed was that in absence of 5' UTR, or in presence of a mutated 5' UTR, or in presence of a mutated nucleocapsid protein, liquid-liquid phase separation gets down-regulated. Basically, the particle size reduces to quite an extent. Now, next was to analyze the packaging. For this, we're still working. We already have all the constructs in our hand, but the work is still in progress. So we have designed a very beautiful assay in order to check if at all these mutations will have any effect on packaging or not. What we're going to do is we'll exogenously express a defective RNA particle. Again, we have prepared this in our lab, which has got the essential parts of the RNA genome without which the RNA cannot survive. So it's a defective interfering virus particle along with 4 structural proteins. We're going to exogenously express it. We'll allow it to propagate inside normal cells, harvest the media, and check the infectivity of this particular harvested media. How well can it infect new cells? We call it reconstitution assay, and we've got a GFP molecule integrated inside our DIV. So we'll know whatever cells are infected by this packaged particle will basically shine green. So we'll know ki, okay, this particular particle has got high infectivity. If my particles are not able to infect, then we'll know that they're not packaged. Now, the next was to analyze the effect on replication and transcription. We prepared a replicon, but unfortunately, our replicon did not work the way we wanted it to. So we came down to computational approach again. What we have done is we have basically designed molecules that will be effectively blocking this particular interaction. Now, we designed a couple of molecules through high throughput screening, and keeping a 50 kilocal per mole energy as a threshold, we have decided on 14 different small molecules that are effectively hindering this particular interaction. Now, if I hinder this interaction and I see certain morphological or let's say physiological changes in the overall virus or the viral titer, I will know that this particular molecule perhaps has got something to do with the, perhaps this interaction is very important for replication and transcription. So we characterized the top 14 hits. Basically, this is a workflow. So we did an in silico screening, selected the molecules with a threshold, evaluated their cytotoxicity, studied their effect on the infected cells. We studied at 3 different levels. We did we checked the viral RNA titer. Then we checked the titer through a viral protein. We did a plaque assay. We have got a fancy name for this plaque assay. We call it cytopathic effect inhibition assay. And then the next In the next segment, we're going to check the binding affinity of these molecules in vitro and then study them in animal model. So this graph basically represents the cytotoxicity of these particular molecules with respect to mock, and we can see that most of them have got almost nil cytotoxicity. All of them are above 90%. All of them show above 90% cell viability. The first one basically represents the RNA levels of different, after treatment with all different 14 drug molecules, what happens to them. And we can see that evidently the small molecule 3, the small molecule 8, 11, and 13, they have down-regulated the overall nucleocapsid protein RNA inside the cell, the infected cells. The next graph is representation of the cytopathic effect inhibition assay or the plaque assay. We basically checked the number of plaques after infection and treatment of these drugs. Again here, sorry for the interruption. So I'll continue. Again, here we can see that in case of drug number 3 or the small molecule 3, inhibitor 3, 6, and 13, you get to see a good amount of downregulation of the viral titer. We repeated this assay just in order to be sure, checked their protein levels. And then this is in detail characterization of the small molecule 3 at different concentrations. First one is the cytotoxic assay, followed by checking the RNA levels and the protein levels. And we can see that at concentration 10 micromole, the viral titer has almost gone from the cells. Same has been done for the 13th molecule. As we saw, good effect in all RNA, at the level of RNA, at the level of protein, and in the plaque assay. Now here again, we can see that although the viral titer goes below 50% at 5 micromolar concentration itself, it's almost gone at 10 micromolar concentration. With this, I've come to the end of my talk. I'll quickly summarize all the points that have been discussed in this particular presentation. The first one, the nucleocapsid protein interacts with 5' UTR of the viral genome. We identified the critical region that interacts with the nucleocapsid protein, which is 51 to 150 base pair of the 5' UTR. And then we identified or pinpointed the exact residues, the amino acids and the nucleotides that are involved in this particular interaction. Followed by, we analyze the effect of these mutations on the liquid-liquid phase separation. So we have not only proven that 5' UTR and nucleocapsid protein, their interaction is important for effective phase separation. We have also shown that if they do not interact using these residues, no phase separation or less phase separation occurs. Now, using these small molecules, we have shown that this interaction is very important for the recruitment of replication transcription complex at the site of where the viral genome is present. Future work that is to be done for this particular project is we need to see the effect of these small molecules for after infecting small molecules, small animals, and then check their effect and check the effect of these two small molecules after infection in those animals. And we also need to evaluate the overall binding affinity of these molecules with the nascent or the wild type RNA, the viral genome. And we also need to perform the reconstitution assay that will tell us if at all these mutations has got any effect on the packaging or not. In the end, I would like to acknowledge all the people who have helped me in order to come to this point. My supervisor, Dr. Milan Surjeet, Rohit, Amit, Oinam, and other lab members, the members of my doctoral committee, Dr. Amit Kumar Pandey and Dr. Saikrit Bhattacharya. I would also like to acknowledge Dr. Asthana, Dr. Shrivastava, and Dr. Shailendra Mani for their help in this project. Thank you.
Now, I would like to call upon the stage Saloni Sengar from Dr. Santosh Mathapati's lab to deliver her presentation.
Good morning, everyone. My name is Saloni Sangar, and I'm a fourth year PhD student under the supervision of Dr. Santosh Matpati. And the topic of my talk is development of an in vitro model for non-alcoholic fatty liver disease using human pluripotent stem cells. So let's have a brief look on the NFLD disease. NFLD is non-alcoholic fatty liver disease in which the secondary fat accumulate in the liver that can leads to the steatosis, which is the inflammation of the liver, and that can leads to the cirrhosis and the hepatocellular carcinoma. The global burden of this disease is around 47 cases per thousand population. In India, around 30% of population is affected by this disease. Recently, this NFLD term has been changed into the MEFLD, which is the metabolic dysfunction associated steatotic liver disease, and the NASH term has been changed into the MASH term. Let's have a brief look on the pluripotent stem cells. The pluripotent stem cells are the cells which can renew themselves and have the potency to differentiate into any human cells. As we know that human pluripotent stem cells are of two types, embryonic stem cells and the induced pluripotent stem cells. These, as I mentioned, that these cells can be differentiated into any human cells through ectoderm, endoderm, and mesoderm. The in vitro models which are used in the metabolic disorders are HepG2 cells, HUH cells, and HEPA-RG cells, which are the hepatic cell lines, which are cancerous in nature. But the disadvantages of using these cells are that they have altered metabolic functions and mutation in genes. Like HUH7 has the P53 mutation in the cells. Another model which is used by the research groups for the metabolic disorders is primary human hepatocytes, which has advantages as they are directly isolated from the liver, they display closest phenotype to human liver. But due to low availability of these cells and lack of reproducibility, it is difficult to culture them and use these cells for the in vitro modeling. So these disadvantages necessitates the development of relevant in vitro models that can recapitulate muscle progression. For that purpose, we are hypothesizing that the pluripotent stem cells can be differentiated into the hepatocyte-like cells, and those cells can be used for the in vitro modeling of metabolic disorders, as they can mimic hepatocyte morphology and functions, and they are available in large number. So the objective of my study is to develop a protocol for the pluripotent stem cells derivation into the hepatocyte-like cells and use those cells for the in vitro modeling of MSLD. This is the protocol which we have established in our lab, in which the pluripotent stem cells will be differentiated into the hepatocyte-like cells by differentiating into the definitive endoderm and the hepatoblast. So, starting with the result, the pluripotent stem cells will be differentiated into the definitive endoderm through the small molecules, and the characterization of the definitive endoderm will be done. Here are the results in which I found that the small molecule can result in the 90% of the endoderm differentiation. Now these endoderm will be differentiated into the hepatoblast. The characterization of hepatoblast was done with the fluorescence microscopy and the real-time PCR, and we found that the hepatoblast, which is differentiated from the endoderm, has the state-specific marker, which is AFP and HNF4 alpha. So this protocol is inducing the differentiation of hepatoblast from the definitive endoderm. Next, those hepatoblasts will be differentiated into the hepatocyte-like cells, and characterization of the hepatocyte-like cell will be done through the functional assays of the hepatocytes, which is albumin secretion, CYP450 activity, glycogen synthesis, and urea secretion, which is present in the primary hepatocyte, and it is not observed in the hepatic cell lines. So through fluorescence microscopy, we found the state-specific marker of the hepatocytes, which is albumin and HNF4 alpha, and we found expression of HNF4 and albumin in the cells. Next, we check the functionality of the hepatocyte-like cells also through the CYP3/4 activity, albumin secretion, urea secretion, and we found that more than 80% of cells are positive for the hepatocyte-like cells marker. And when we compared these cells with the primary human hepatocyte as positive control, we found the state-specific markers and the hepatocyte marker were present in the cells. Next, we checked whether the cells has the glycogen storage capacity. Through past staining, we found a positive staining in the hepatocyte-like cells. So all the combinations and the protocol has the function of the hepatocyte-like cells by Tatum. Now we are using this is the pathway for the MAFL progression in which the free fatty acid is treated to the cells and the triglyceride level is checked. So this is already established model of HUS7 which are used by many research groups in which the HUS7 or HepG2 cells are treated with the free fatty acid and neutral lipids and triglycerides were estimated. So we are mimicking this experiment into the hepatocytes-like cells for the in vitro modeling, in which the hepatocyte-like cell will be treated with the free fatty acids and neutral lipids, triglyceride, gene expression, and ROS, reactive oxidative species, will be estimated. So here we can see that with increase in the free fatty acid concentration in our HLC model, the body P staining which binds to the lipids is increasing. Cell viability, there is no cytotoxicity between the groups. We measured neutral lipids and ROS production through flow cytometry, and we found that ROS and neutral lipids is increasing. And through oil red staining, we found that the free fatty acid, with increase in the free fatty acid concentration, the Nile red staining and the oil red staining is increasing. We also measured the intracellular triglyceride level in the model, and we found that the triglyceride level is also enhanced. Through gene expression analysis, we found that lipogenesis markers were non-significant. So this free fatty acid induced model of HLCs can be used for the mast progression disease. Next, we want to check whether our model can reverse the phenotype of the mast disease. So for that purpose, we are using resmetirom drug, which is recently approved by the FDA in March 2024. So first cytotoxicity of this drug is checked on the hepatocyte-like cell model, and we found no cytotoxicity. And with treatment of this drug, we found the triglyceride levels were decreased as the drug was treated with the free fatty acid induced model to the hepatocyte-like cells. So this lipid accumulation in mast-induced model using hepatocyte-like cells can be effectively inhibit resmetirom, suggested that the model can be used for the drug screening purpose also. So the summary of this work is that protocol has been established in our lab, and this hepatocyte-like cells exhibit the hepatocyte functions like CYP450 activity, albumin secretion, which is not found in the hepatic cell lines. And it can recapitulate the mast cell disease progression more relevant to the hepatic cell lines. This can be used for the therapeutics and high throughput screening also. So future prospect of this study is to further validation of the in vitro model and the modeling of infectious disease on the hepatocyte-like cells like HEV. So recently we have patent filed against this protocol also in May 2024. I would like to acknowledge my supervisor, Dr. Santosh Matpati, and lab members, friends, academic support, collaborators, and financial support. Thank you.
Now, I would like to call upon the stage Vaibhav Nain from Dr. Amit Kumar Pandey's lab to deliver his presentation.
Good morning, everyone. Welcome to the Foundation Day colloquium. I will be presenting part of my PhD work today, and title of my presentation is Sulfur Metabolism and its Role in Mycobacterial Physiology and Pathogenesis. So tuberculosis is an age-old disease which has shown to be I think this tuberculosis is caused by the bacteria called Mycobacterium tuberculosis, and it gains entry into the host pathogen host through the airway. After settling down in the lungs, it infects alveolar macrophages and starts to grow inside them. And in an interaction between host and pathogen, a complex granuloma formation happens which try to contain the bacteria. But due to macrophage, the bacteria taking over the interaction, the macrophages tend to die and the bacterium is released into the body. Now, further, even after many years of its discovery, tuberculosis still remains a challenge to treat in short duration because of long therapeutic regimen, which is because of poor compliance. Further, this condition is aggravated by emergence of AMR strains, and recently, persister bacteria is being looked into for this problem of incurable disease. Now, this persister population, these are the bacteria which have low metabolic as well as replication rate, and they do not respond to the antibiotic treatment, and they show a biphasic kill kinetics. The first of all, they are most of the population bacterial cells in the population are killed rapidly. However, there is a small population of bacteria which still lies there, which is not responding to the treatment. So these are the persister bacteria, or called drug tolerant bacteria. So uh, we in our lab, we try to identify the genes and pathways which are responsible in persister formation in MTB. We validate these genes through genetics and in silico approaches, and we see whether these targeting these genes will help reduce the MIC of anti-TB drugs or not. So instead of replacing the presently used anti-TB drugs, we are trying to improve the efficacy of already existing drugs. Lastly, we validate these leads in vivo in combination with the anti-TB drugs. So from that perspective, we have started sulfur metabolism, studying sulfur metabolism, because people have already been shown role of lipid metabolism, carbon metabolism, amino acid metabolism in as prospective novel therapeutic targets. So we came to sulfur metabolism. We know that sulfur is a very important key metabolite for synthesizing very important key factors of the cell, which maintains the bacterial physiology. For that, we uh, grew the bacteria, MTB, in sulfur-free and sulfur-rich medium, and saw that bacteria is unable to grow in sulfur-free media, while supplementation of sulfur helps it growing. So total intracellular sulfur was also reduced when we did using ICP-MS. We checked intercellular sulfur was also reduced, which also led to total reduction in total biomass, indicated by total less protein in sulfur-free bacteria. Finally, next we wanted to see what are the transcriptomic changes in the bacterium after we grow them in sulfur-free and sulfur-rich medium. We found that most of the genes were downregulated in the RNA-seq analysis. Further analysis led us to findings that most of the genes in the operon, which encodes for ribosome large subunit assembly, are downregulated. All the components of electron transport chain, NADH, Q reductase, ATP synthase, cytochrome C complex, and cytochrome BD oxidase, they are all downregulated. Secretory pathway of MTB is downregulated, as well as the cell membrane associated antigens are also downregulated, which also help us speculate that in vivo and even in macrophage based model, this bacteria growing in or lacking sulfur will be attenuated. However, out of few genes which were upregulated were cysteine and sulfur uptake, and methionine metabolism. So closely analyzing the sulfur metabolism pathway, sulfur is taken up by bacteria through a transporter, which is encoded by these genes, indicated in the red arrows. So using CRISPR-I mediated gene knockdown strategy, we knocked down these transporters in tandem, and we found that in vitro the bacteria was severely attenuated for its growth and had significantly reduced thiol content. We also saw that this using this CRISPR-I mediated sulfur depletion model of MTB, that this bacteria, whenever we induce sulfur depletion using CRISPR-I, the bacteria was severely attenuated at whatever time point we did this. So this is the gross pathology of the two groups, wild type and the sulfur depleted bacteria. This is the total granuloma score of the lung lesions. Further, this growth defect is dose dependent. So as you keep increasing ATC, which is the inducer of CRISPR-I, the total sulfur level in the cell decrease. So as low as 10 nanogram of the ATC concentration was good enough to bring down the bacterial viability to 50%. Further, we tested this along with ATC in combination with anti-TB drugs. We found that in most of the cases, the MIC was reduced, indicated by the right to left shift, in except for INH. So we wanted to see whether INH can in vivo can we use this strategy in combination with INH. So we used again this in vivo model of CRISPR-I sulfur depletion model of MTB and did INH bacterial clearance study, and we found that this purple line, which is shown by a double mutant treated with INH, the bacterial burden was reduced below LOD. So it is very effectively working. So this is the percentage survival of the week 4 and week 6 time point. We see that this survival index of the bacteria treated with both doxycycline and INH was bring down to below OD, limit of detection, which clearly shows that sulfur depletion in combination with INH achieves near stability in vivo. So summarizing this work, we have shown that sulfur-rich bacteria, if can be sulfur depleted, leads to a non-replicative MTB, which is de-energized because of low energy, and it leads to reduction in thiol content of the cell, which makes the bacteria susceptible to reactive oxygen and nitrosative species, which are faced by bacteria inside the macrophages, thereby making it more susceptible. Further, in vivo attenuation is very drastic and clearly distinguishable. We also show that increased sulfur depletion leads to increased sensitivity to anti-TB drugs. And lastly, in vivo sterility is achieved along with INH. So more far and large, this system is working. So further, we want to see this screen molecules again, this transporter, and see whether the same happens in presence of a molecule. So with this, I would like to acknowledge all the people associated with this work. Thank you.
Now, I would like to call upon the stage Meenal Chawla from Dr. Babatosh Das' lab to deliver her presentation.
Is this okay? So good morning, everyone. My name is Meenal Chawla, and I'm working under supervision of Dr. Babatosh Das in Functional Genomics Laboratory. And title of my presentation is Discovery and Development of Aminoglycoside Antibiotic Potentiators to Resensitize the Drug-Resistant Escape Pathogens. So to begin with, I give a brief introduction about antimicrobial resistance. So AMR happens when bacteria no longer responds to the antibiotics, resulting into the treatment failure. Now, the question arises why we should be concerned about the AMR research. So AMR basically impacts the different domains of life. It impacts the public health threat. Like according to WHO, it contributed to 4.95 million deaths in 2019 and could result in up to 10 million deaths annually by 2050. It has a huge economic burden also due to the longer hospital stays and lesser productivity. Apart from this, it poses risk to the food system as well as the global health security and impacts the medical treatment, as well as this emergence of the drug resistance is extremely worrisome. So talking about the molecular mechanism of antimicrobial resistance, so these are diverse. The mechanisms such as active efflux or decreased influx reduces the active concentration of the antibiotic inside the bacterial cell. Apart from this, mechanisms such as target protection, target site modification, and target bypass doesn't allow the antibiotic to bind to its target, and there is antibiotic modifying enzyme which inactivates the antibiotic. So to overcome the problem of antimicrobial resistance, several approaches are being used, and one of the common approach is antibiotic potentiators. So these are the active molecules with minimal or no antibacterial activity but enhance the effectiveness of the antibiotics when used in combination. So these basically resist the resistance mechanism so that antibiotic could work again. So there are different mechanisms by which antibiotic potentiators work. They can either inhibit the antibiotic efflux pump, they can inhibit the resistance plasmid replication or segregation, which is majorly responsible for transferring the ARGs, or it can inhibit the antibiotic degrading enzyme or antibiotic modification enzyme. The example of all those antibiotic potentiators are given in these bubble diagrams. And the biggest example of success of antibiotic potentiators is for beta-lactam, which is commercially available in the market. But aminoglycoside, which is the second most commonly used antibiotic, no potentiator is even in the clinical trials. So to fill this gap, we aim to develop the aminoglycoside antibiotic potentiators to resensitize the MDR pathogens. So this is basically our approach. This is the aminoglycoside modifying enzyme, which modifies the aminoglycoside antibiotic, due to which it is not able to bind to its target, which is 30S ribosome subunit, due to which there is no effect on the bacterial growth. So if we are able to find a potentiators which can bind to this aminoglycoside modifying enzyme, due to which the antibiotic is free and it can bind to its target, leading to the bacterial cell death. So when we talk about aminoglycoside modifying enzymes, so these are of three different classes: phosphotransferase, acetyltransferase, and aminoglycoside nucleotide So phosphotransferase transfers the phosphate group, and acetyltransferase transfers the acetyl group, and nucleotide transfers the nucleotide group to the aminoglycoside antibiotic. So to begin with, we have first determined the global prevalence of the aminoglycoside modifying enzyme. And here I'm showing the prevalence in the 4 pathogens from last 5 years, which is from 2019 to 2023. And the pathogens list includes the Acinetobacter baumannii, E. coli, Klebsiella pneumoniae, and Pseudomonas aeruginosa. So these are the critical and high priority pathogens, which are listed by WHO and by DBT as well. So these are the percentage prevalence, and these are the different ARGs from 3 different classes. Like from APH, the most prevalent one was APH3 and STR-A and STR-B. And from here, we have picked the prevalent aminoglycoside modifying genes and moved further to engineer the bacterial genome to develop the reporter strain for screening of the compounds. So for this, we have amplified the resistance gene from the different clinical isolates and cloned it into the integrative conjugative vector, and then inserted into the diff site and chromosomally into the 2 bacterial isolates that we have selected, which is N16961, which is a Vibrio cholerae strain, and E. coli ETCC, which is 25922 strain. So these are highly susceptible bacteria, and they do not have their own resistance mechanism. So they'll show resistance to the aminoglycoside antibiotic only because of the presence of the resistance gene, which is inserted into their chromosome. Apart from this, we have also checked that it is highly permeable, also making it an ideal strain for the screening. So here is the results of all those reporter strains that have been constructed. So the first circle represents the different classes, and the second circle represents the different genes that have been picked. Third circle represents the clinical isolates from where they have been isolated, and fourth circle represents the antibiotic resistance gene. Like for example, APH3 has been amplified from E. coli and it is showing resistance against the kanamycin. Then to begin with, we have done these standardized the screening assay for where we have used our reporter strain, and we have used 2 libraries, which is the natural Selechem library, and the second was FDA approved Medchem library. So screening was done in the 2 phases. The first phase was primary screening. So this was done in the presence of the antibiotic. So all those compounds that were showing the partial growth inhibition are picked further for the secondary screening. And then the secondary screening was done in the presence as well as in the absence of the antibiotic. So if the compound itself is showing the antibacterial activity in the absence, that means it's an antibacterial compound. If the compound is showing better effect with the antibiotic, that it's a synergistic compound. If the compound alone itself is not showing any activity but enhancing the activity of the antibiotic, that means it's a potentiating compound. Then all those compounds has been picked further and checked in the E. coli reporter strain, and later on it was also assessed in the MDR gram-negative escape pathogens. So these are the screening which was done against the NAPH3 with the kanamycin antibiotic. So in short, we have got more than 300 compounds showing greater than 30% inhibition in our primary screening. And these are the compounds which are showing in red circle are synergistic or potentiating compounds. So these are the results. So in primary screening, there are 201 compounds in the library which are antibacterial and are find in our screening assay also as antibacterial. And there are 91 compounds which are found in the literature and reported as antibacterial, and there are 34 compounds which were novel. So as through our screening assay, we are able to pick so many antibacterial as well as the reported antibacterial compounds shows the sensitivity of our assay. Then from the 34 compounds, there were 13 compounds which are showing 30 to 50% inhibition, 9 were showing 50 to 80%, and 12 were showing 80 to 100% inhibition. And so here I'm showing the results for the synergistic and potentiating compounds. These are the 8 compounds in an APH3 reporter strain. So in the presence of the antibiotic, due to the resistance, there was no inhibition. In the presence of the compound, there was minimal inhibition in few compounds, when with the antibiotic, they were showing the significant inhibition. And then they were also assessed in the EAPH3 reporter strain. So here, from out of 8 compounds, only 3 compounds were showing the activity. Then from these 3 compounds, I'm showing here the data for our one of the most lead molecule, which is BMM3. So till now, we have done the activity, checked the activity at 10 micromolar to determine the best concentration. We have checked with done the checkerboard assay. So this we have not done only with the kanamycin antibiotic, which was used in the primary screening. We have also done with the spectinomycin. For that, we have used EAD reporter strain, and also done with the gentamicin. For that, we have used EAC3 reporter strain. So here the MIC was like of the compound was close to 40 micromolar, but with the antibiotic, when used, it shows the potentiating effect at 2.5 micromolar. So from this, then we have also evaluated this compound in the MDR clinical isolate. So the clinical isolate selected here were showing resistance to more than 10 antibiotics. They are not only carrying the aminoglycoside resistance genes, they are also carrying the beta-lactam resistance genes, macrolides, and chloramphenicol as well. So this is the E. coli isolates. As we can show here, we can see here that in the presence of the compound or the antibiotic, there is no inhibition. But with the presence of in the presence of antibiotic and the compound, 100% inhibition was observed. The similar was also done through viability assay and through the resozorin assay. Similarly, we have also determined the activity against the A. baumannii clinical isolates, K. pneumoniae clinical isolates, as well as with Pseudomonas aeruginosa. So we have checked the activity with the 3 different isolates from each of these pathogens. And for E. coli and Klebsiella pneumoniae, the significant results in all 3 isolates were observed, whereas for A. baumannii and Pseudomonas aeruginosa, in 2 clinical isolates out of 3 were able to get the activity. Then to understand the mechanistic insights for these, since these compounds are showing activity only with the antibiotic, and there is only one resistance mechanism which is present in our reporter strain, so it is more likely that it will bind to our resistance enzyme. So for that, we have done the molecular docking analysis. There we have tried to bind this molecule with the ANT enzyme. So this ANT enzyme is responsible for providing resistance to spectinomycin, and it was checked in our reporter strain also. So ANT with the spectinomycin is binding using the 9 interacting residues, whereas our molecule with the ANT is interacting using the 19 residues, and 5 of these are 18 residues, and 5 of these are like common. And through free binding energies also, we have observed that the molecule is binding with the enzyme in much better efficacy as compared to the drug. Similarly, we have seen for the APH also for APH, and with APH we have taken the kanamycin antibiotic because kanamycin is responsible for providing resistance for this APH, and here also we have observed the same effect. However, with the APH and for the AAC, the effect was not as prominent as observed with the ANT. So in the end, I like to summarize like these are the highly sensitive chromogenic reporter strains, and high throughput screening assay established rapid and efficient screening of the compounds demonstrating antibacterial, potentiating, and synergistic effect. So from the screening of natural and synthetic compound libraries, BMM3, the most promising compound was observed, which was showing 100% potentiating effect with spectinomycin, kanamycin, and gentamicin against the drug-resistant gram-negative escape pathogens at a low concentration of 2.5 micromolar, and through our in silico analysis indicates that the compound is binding with strong affinity with APH, ANT, and AAC enzyme and competes with antibiotic to form the complex. So these are our future perspective where we want to validate the in silico findings through site-directed mutagenesis, and then we want to assess the efficacy in UTI and GI infection model and to conduct the toxicity test. So lastly, I'd like to acknowledge my supervisor, Dr. Bhavatosh Das, my co-supervisor, Dr. Dinesh Mahajan, and my RAC members, Dr. Shailendra Sahana for the in silico help, and Dr. Dipti Jain, THSTI, CSI, my funding agency, and my lovely lab members. Thank you.
Now, I would like to call upon the stage Taruna from Dr. Nitya Vadwa's lab to deliver her presentation.
So, good morning, everyone. I'm Tarna, fourth year PhD student. Today, I'll be talking about the role of microbiome in perpetuating inflammation in the initial phase of acute pancreatitis. So, let me first walk you through what acute pancreatitis is. So, pancreas is basically a small organ, vital organ, which is situated in the retroperitoneum. It basically secretes certain digestive enzymes and also some hormones, which are important in maintaining glucose homeostasis. Acute pancreatitis is nothing but the acute inflammation of the pancreatic organ, which is clinically presented as the acute onset of upper abdominal pain. So the diagnostic criteria that we use for acute pancreatitis is the upper abdominal pain, which is radiating towards the back. Serum amylase is more than 3 times the upper limit, and if the CT is showing some enhanced pancreatic homogeneity, that is also one of the diagnostic criteria for acute pancreatitis. So the common etiologies of acute inflammation are mainly stone, ethanol, idiopathic, and there are certain cases where the higher concentration of TG might lead to acute pancreatitis. So the conceptual model of acute pancreatitis is whenever there is acute inflammation of the pancreas, it leads to the local injury. Since acute pancreatitis is a disease of varying severity, the mild cases are usually don't get this kind of local injury. I mean, they only experience the pain. So what clinician does, they simply give them the anti-emetic or the painkillers. They get discharged easily, but the severe cases usually develop systemic inflammation, which leads to organ failure. So the question that we are asking in this study is the pancreatic inflammation, which is mainly the tissue injury of the pancreas, leads to the release of certain DAMPs or damage-associated molecular patterns or unsaturated fatty acids, which leads to systemic inflammation, leading to the early primary organ failure, which mainly occurs in the first week of illness. Furthermore, the infection of the pancreatic inflammation, which occurs later on the illness, leads to infected necrosis and which finally release certain pathogen-associated molecular patterns since there is infection, leading to late secondary organ failure. So the question that we are asking is, if the inflammation is only due to pancreatitis per se, then it should not lead to the organ failure with the mortality as much as high 40%. So the question that we are asking here is, are there any microbial infection which is playing the role in exaggerating the systemic inflammation in acute pancreatitis? So in order to question this answer, we have conducted a prospective observational study. We consecutively recruit the patients with acute pancreatitis in the first week of illness itself. So these are the groups that we had recruited. The mild acute pancreatitis, a number of cases 15, severe acute pancreatitis. So the severity criteria of acute pancreatitis, as I discussed, is the persistent organ failure, that is organ failure which is last more than 48 hours, which could be single organ failure or multiple. So mainly the three organs are majorly affected in severe acute pancreatitis, which are the heart, kidney. The third group is the infection confirmed patients, where we had recruited a total of 15 healthy controls, number 15, and the reagent controls 8. So we had collected samples within the first week of illness for both mild and severe cases, which are blood, buccal swab, rectal swab, and skin swab. Further, we had longitudinally collected the samples for the severe cases in the second week of illness as well as in the third week of illness. So what we have done is, since we wanted to study the role of microbes in these low biomass samples, we need to standardize some strategy or some method that is able to isolate the bacterial DNA, or I would say the microbial DNA, from the low biomass sample. So what we have done is, rather than going to the conventional approach, we have used a benzonase digest approach, which effectively lies all the DNA, I mean the unprotected DNA, and leaving only the DNA preparation leads only from the living microbiome. So using this approach, we had isolated the DNA, we had proceeded for the sequencing using NextSeq 2000 platform. So these are the one of the infection confirmed sequence patients, which shows the the Escherichia shigella positive patient, which is positive by the culturomics. So we have done the sequencing of the sample. We could see that Escherichia shigella is there. So we confirmed that, yeah, our method is working. So finally, we proceeded with the blood to look into the blood microbiome of acute pancreatitis cases, where we could see there are certain pathogens such as Streptococcus bifido, Escherichia shigella, Bloschia, Enterococcus prevotella, and Klebsiella floating in the systemic circulation of both mild as well as in severe cases. Next, we wanted to see, like, is there any differential, or I would say varying microbes in the, or the microbial load in the mild or severe cases. So as we could see, the Vibrio is more in the mild cases as compared to the severe. So now we would like to study like what kind of functions they are performing. Also, since we got a lot of gram-negative pathogens in the systemic circulation, we wanted to know whether they are actually there or not. So for that, we have checked the LPB, which is LPS binding protein in severe versus mild cases, and we could see that the LPB is more in severe cases as compared to the mild. Next, we wanted to culture those organisms that we have found through the metagenomic samples. So we used a method where we had inoculated the sterile culture bottle and we have done the blindly subculturing. We had isolated the single colonies, performed the Sanger sequencing, and yes, we are able to isolate those microbes from the blood. So then we have performed a concordance analysis where we could see that culturomics and metagenomics study, the metagenomics and culturomics results were showing the positivity, like the 6 organisms we could able to isolate, which are out of the 20. Next, we wanted to study like from where these organisms are coming into the systemic circulation. So for that, we have collected the buccal swab. So we could see that there are certain organisms such as Pseudomonas, Klebsiella, Acinetobacter, and Enhydrobacter, which are highly abundant in buccal cavity, might be coming into the systemic circulation from oral. Next, we wanted to, since oral and gut are the most diverse or the abundant connection of the location. So we wanted to study whether the gut is responsible for the transmigration of microbes to systemic circulation or not. So here also we have found certain organisms such as Pseudomonas, Enterococcus, Bacteroides, Prevotella, Corynebacterium, and Faecalibacterium, which could migrate from rectal to acute pancreatitis to the systemic circulation. So next we had mapped top 20 bacterial signatures among different body sites, and we could see that most of the organisms that we found in the blood are coming either from rectal, skin, or buccal. So the outcome measures that we had find from the study is the presence of microbes and the microbial load in the blood of both mild as well as severe cases, and their correlation with the CIRS and organ failure. Now we wanted, since we had isolated these organisms, now we'll be doing the genomic analysis. We could see whether those, what kind of functions they are performing, whether they do possess certain virulence genes or not, and also the infection patterns between the gallstone-induced rape and the alcohol-induced rape, since alcohol is mainly making gut more leaky. So we are supposing that we could found more or exaggerated response of those organisms in the systemic circulation. With that, I would like to conclude, acknowledge my PhD supervisor, Professor Nitya Vadva, my co-guides, Professor Pramod Garg, Professor Bhavtosh Das, Professor Amit Avasthi, Dr. Soumya Jagannath Mahatpatra, my doctoral committee members, Professor Shinshni Bhatnagar, Dr. Deepak Gunjan, Dr. Shalumida, Dr. Deepak Rathod, and Dr. Kajal Jain, my funding agency, lab members, THSTI and AIMS, friends and families. Thank you.
With this, we've concluded the scientific colloquium. Thank you to all the presenters.
It is always intriguing for the young and thriving minds to read and hear about the success stories and the hardships endured through the journey of success. Today, we have one such personality with us, Dr. Ramesh Hariharan. Dr. Hariharan is a co-founder and CEO of Strand Life Sciences and an adjunct professor at Indian Institute of Science, Bangalore. He did his BTech from Indian Institute of Technology, New Delhi, and his PhD at the Courant Institute, New York University in Computer Science. He was on the computer sciences faculty at Indian Institute of Science before starting Strand Life Sciences. His efforts over the last 2 decades are aimed at bringing together cross-disciplinary teams to solve the problems in genomics. Dr. Hariharan is a fellow of the Indian Academy of Sciences, the Indian National Academy of Engineering, and many more. We invite Dr. Ramesh Hariharan to enlighten us with the enriching experience of his entrepreneurial journey. The title of his talk is Translating Science into Business, the Strand Story and Lessons Learned.
Either I need it in Google Drive with a PowerPoint, or can you push it to? Can you just open the link in on Chrome if possible? Yeah. Otherwise, actually, it's okay. That's yeah. That's, okay. Thank you all very much for inviting me here. As you will notice, I changed the title a little bit to Translating Science into Impact and not to Business. Business is just a means to an end. Eventually, one wants to take all the interesting work that folks like you are doing here and convert that into some eventual impact that affects, patients or health in general, or whatever be the eventual end goal of your research. There are many ways to do this translation, and I'm going to just, I can't say that I know all of those ways. What I can say is that over the last 2 decades, we've tried one way, and I want to share with you the experiences and the lessons learned along that path. But there may be many, many different ways to do this, and those ways may evolve over time. Okay. But let me go through our experiences to see if there's anything that you, as you go forward, can learn from there. Our story began, 2 decades ago, which seems like ancient history now, but at IISc, we spun Strand out of IISc in the year 2000. This was just at the when the Human Genome Project was about to culminate. Prior to that, there was a lot of excitement in the area of both, genomics and computer science, because suddenly we were seeing data at a scale that was not seen in the life sciences prior to this. And it was clear that there were so many questions of the computational and data variety that were being asked that a multidisciplinary approach to this was very necessary. And that's around the time that 4 of us got together. We were all 4 faculty at the computer science department of the Indian Institute of Science. And we came with different computer science skills. Some of us were, I worked on sort of, you can say, sequence analysis problems, but from a very theoretical computer science perspective, where you try to engineer algorithms to show speed and scale. Some of my colleagues worked on optimization systems, others worked on graphics and visualization engines, and yet others works on systems and system engineering capabilities. And this was a good mix to come together. And we were driven by, again, inspired by the Human Genome Project, to say, how do we bring all of our diverse computational skills to apply to data in the life sciences to make an eventual impact on human health. That was the goal. as we went about, we worked on a variety of different problems. But our first problem was very interesting. I thought this would be interesting to you. Crystallography is something that is still done to identify protein structure. X-ray crystallography is still one of the primary means. And other than that, that's modeling, which has made big leaps and bounds in the recent years with AlphaFold. But 20 years ago, modeling was still very weak, and crystallography was the primary means by which one could get protein structure. But there are so many proteins, and so there were companies coming up saying we're going to industrialize the process of protein crystallography. They would run hundreds of thousands of experiments where you try different conditions to make proteins crystallize and do that for a variety of proteins. And at that scale, there was just so many images, all these wells were being monitored through cameras. And the question was, which of those wells has a crystal in it and which doesn't, and how do you pick out those crystals? And there was no easy way to do this manually. There was too much effort. So this is the type of image recognition problems that we do routinely nowadays because AI has evolved so much today. But 20 years ago, AI was practically at its infancy. And as you can see, there's a variety of different sorts of images that show up. Some of these are of interest. So these are the ones that you can harvest and do crystallography on. Maybe these, certainly not these. And these are some examples of things that you need to do. You need to get into the region of interest and identify the crystals, etc. So you can imagine interesting sort of image recognition problems. And it took us about 6 months to solve this. We created the system. It had beautiful performance. We licensed it out to this company that was running this high throughput crystallography that gave us revenue flow very quickly and so on. So very interesting first problems. And we thought that here is something that is very promising. We can sort of use this to scale to many different, either on this problem on a wider scale or on many different problems. And there we ran into our very first realization, how picking your problem is very important, because we realized that while this is a powerful problem and clearly it can have an impact if we can enable more crystallography and more protein structures to come out more efficiently. But this is not something that a lot of different people do. This is something that a couple of companies might try to industrialize, and it is down to a very small sort of market size, right? So you cannot make an engine from this. You cannot make a larger platform using this. So this is just one problem that you can solve. So then the natural question arose, how do we broaden from here into a broader variety of problems? And at that point of time, 20 years ago exactly now, the most common source of data, if you look back then, was these microarrays that were being run to identify gene expression, identify SNP genotyping. And these microarrays were our best bet to generate high throughput data. Sequencing was not there then, remember. So you had to guess what you want to measure. You then put it on a microarray, and then you measure those and you get those readouts, and you usually get 20,000 genes in an experiment. Lots of data that results. And the world was just starting to apply these to a wide variety of problems. Every single system you can ask, if I treat it with a drug, what is the gene expression change that happens? What is the underlying genomics in terms of SNPs that might have an impact on these changes? And so there was an explosion of data that was happening at that point of time. But the tools to analyze these were not very mature. Today they are mature. Most people had to rely on a few people who knew how to do this analysis to enable that analysis. And what we decided to do then was to say, we can really broaden the pool of people who can analyze data by taking all of these statistical algorithms, all the visualizations, all the pre-processing, everything together into a single platform that can do all of this in one shot. And that's what we created in 2004. And this took a lot of engineering effort, a lot of bringing together statisticians and biologists all together under one roof to be able to create all the tools that are needed to essentially do these sorts of analyses. And as exhilarating as that effort was, when we created that platform and we knew that the need for it is clearly there because lots of these experiments were being run, people were starting to publish, but they were all struggling with the analysis. The question became, you have a platform, you have users, how do you reach out to these users? How do you commercialize? How do you tell people that here is something that will make your life easier? And that is not, as we realized, until then we hadn't thought about this problem. Once you have a piece of technology, however powerful it might be, there is a lot of activity in taking it out and bringing it to the people's notice and getting it adopted, etc. So we call that the process of commercialization, of course. And when we started doing it, our first attempt was to, of course, there was very little happening. And so our first attempt was actually very interestingly in Japan, because in Japan, there is a lot of infrastructure, what are called the system integrators that bridge between the creator and the eventual consumer, but be the scientist or whoever who's trying to analyze data. And that infrastructure is so mature that you can very easily plug in there and go through that process of, through that ecosystem of system integrators eventually into the scientists. And that's where we got our first success. And a little bit in China and Australia, where there were these some ecosystem there. But of course, the main market was the Western markets. And there isn't any such infrastructure. It's a lot of, you have to go do, it's much less mature, so to say. You have to do your own commercialization. And there we came across our second realization that while it might take a certain amount of effort, creativity, cost, money, etc., to build technology, to do your primary innovation, the amount of money that it needs to take that innovation out and commercialize it may be an order of magnitude more than that. And that's something that we did not at that point have. So we thought, what can we do potentially to solve this problem? So we started looking for partnerships. So, and very quickly we struck up a partnership with Agilent. And Agilent said, look, we are selling microarrays. We need our users to adopt microarrays. You have a tool, we have the customer base. We can get into a partnership, but it has to be a tight partnership where we will own the IP jointly. And the market, the customers will be ours, the technology is yours, but everything else is done jointly. So we said, okay, I mean, it doesn't look like we have too much of an option to do this ourselves, because these tools are, you have to go and sell to so many scientists. The cost of doing that is very expensive. So we signed up with Agilent as our partner. And this had remarkable success. And then it also had a long-term sort of saturation, which I'll talk about next. But the success was there to see. If you do a Google search on some of these terms, these tools that we created together, so GeneSpring, Strand NGS, Avardis NGS, you'll see about 17,000 citations in Google Scholar, right? So it's a large number. So what that meant is that with this partnership, these products traveled widely to lots of scientists all across the world, and they started using them, publishing with them, and the number of such publications just rapidly build up, tens of thousands of publications, right? You can imagine 15, 20,000 publications. That's a large number. So that's the impact we wanted to create, right? I mean, you want to create technology, you want to then take it, have it being used and to further research or improve health or whatever the end goal that might be. And so this was remarkably successful, that we managed to very rapidly scale up to a large number of users, right? And then the ground started shifting. So this is, these tools were geared towards microarrays, and microarrays got adopted widely. People started using these tools, very successful. But then around 2009, sequencing costs started going down. As you can see from this curve, the inflection point where the curve starts going away from the straight line and dipping down is around 2009. Right? And those costs started dropping fast. and which meant that people started moving away from microarrays to sequencing, right? So, which is great because this is a new opportunity to create whole new sets of problems come up, whole new tools are needed, whole new analyses can be done, new results can be discovered, new applications can be built. So all of that was enabled, right? So we very rapidly started adapting our tools to sequencing analysis. We put all of that evolution happened. But what could not happen is we had an elephant on the other side. So Agilent was a big elephant on the other side, very slow to move. They had a microarray story, but not a sequencing story. They couldn't move. I mean, we were a nimble, sort of quickly move our products. So, all of that is fine. But the other end was a real elephant that just could not move. So the sequencing revolution happened, and our entire product line got stuck with Agilent because Agilent wouldn't move, right? And they had the customers while we had the technology. And that partnership was a strong partnership, but it could not evolve with the times. So that's another lesson learned, that while you can translate, but if you're building an institution that has to be resistant to the dynamics of the world, and all of us are together creating these dynamics, right? We're all working to create those dynamics. We want to create new products, do new science, new discoveries, and that will change, that will create a lot of dynamics. But then you have this inertia in the system that couldn't evolve to that dynamics. So this was a challenging period for us. Our core innovation over several years that could now evolve into new areas was completely stuck, right? We couldn't do anything about it. So we had to evolve beyond at that point of time. And also it was clear to us that while a lot of what we were doing until then was looking at just the data and the analysis in a research context, we didn't really have an impact on the eventual patients, right? On the eventual health of people. So we were still not translating it to the extent that we needed to. And the urge to take it beyond just publications and using these tools for publications to saying we want to have an impact on individual, on health. And second, that was one reason. The second reason was we also realized that when you're doing addressing only the informatics end of the problem, your ability to control the whole application is limited. You have to broaden yourselves to more parts of the application so that you can deliver a broader set of applications to your eventual, to make that eventual impact. So we said, okay, now we've got to expand from being only doing the analysis of the data to actually doing the entire end-to-end assay, because then you have something powerful that you can then tweak and evolve and innovate upon to create new applications. So that's where we expanded, set up labs. This was about 2012. Set up labs to do sequencing. And this was the earliest, right? Imagine 10 years, 12 years ago, most places in India did not have a sequencer. We set up the first sequencing lab, got it CAP accredited almost a decade ago. And the first sequencing sort of center in India. Very quickly, we realized that the biggest challenge in taking sequencing and bringing it to the benefit of patients and delivering healthcare applications was not the sequencing. It was everything that happens before the sequencing and everything that happens after the sequencing. Everything that happens before the sequencing is that you cannot, it's too expensive to just sequence, right? You've got to figure out what to sequence. You've got to enrich for that. You've got to optimize for that. You've got to cost optimize for that. And that process still continues. I mean, that challenge is quite substantial as newer and newer applications are coming in, to be able to cost optimize that so that you can deliver it at reasonable costs, particularly in markets like India. So everything that happens before sequencing was, of course, very important, but then everything that happens after sequencing was even more challenging and intriguing, in the sense that you get these, millions of variants out if you sequence everything, otherwise tens of thousands of variants when you sequence an individual. And then how do you dig through those to figure out what is relevant, what is not, what is the cause of the problem at hand? If a patient comes in to you and says, here is my problem, and I want you to figure out what the genetic diagnosis of that is, then you have to go from tens of thousands of variants down to those one or two that might be meaningful. How do you do that? And that was an amazingly intriguing process where we had to put in all of the analytical systems for that. That was a lot of sort of algorithm and software and data building. And it was very satisfying though, because very quickly we could take an individual patient and diagnose what their problem was. A decade ago, patients would go from doctor to doctor, they would go to the US, they would not get a diagnosis. And here we were able to, in 2014, be able to sort of start diagnosing these patients and getting, here is the character in your genome, that's the cause of that problem. A number of these things, right? And so it was so intriguing that I ended up writing a book with about 9 case studies of different detective stories that one had to solve with different patients. The first of those was my own color blindness, where I had been colorblind for a long time, but never knew why. And then, figured out through sequencing that 2 genes had joined together to create a hybrid between red and green colors that sort of made my ability to distinguish between those two lesser. And then many, even my family, my mother-in-law had blindness. We sort of solved that problem, etc. So very intriguing. So for the first time, we were able to take all this technology and apply it to a real patient and be able to derive value to a real patient, right? Which was great, which was amazingly exciting. What was challenging though, is that as we started doing this in India, we suddenly realized that the cost of doing this is so high and what patients can afford is so low, and the doctor's understanding, etc., to convince patients to pay value for this was so low that we were losing a lot of money doing this in India. While the science and the impact was tremendous, the economics was disastrous, so to say, right? And so at that point of time, we said, how do we do this, right? How do we take this, a natural market for the US? We were there with the informatics, the analytical tools already, and we said, how do we do that? By this time, remember, the analytical tools had started winding down because Agilent was still not moving with sequencing, and people were moving away from microarrays, and our presence in the tools market was starting to go down. So we said, let's do one thing. So we've created this entire diagnostic workflow. We've got the sequencers, we've got the lab, we've got the informatics. We know how to go from lots of variants into a single variant, how to do these diagnoses. Let's take that to the US. The point in the US being that the US market can potentially pay. Our cost of development, etc., will be far lower than what, and we have all the technology already that we can deploy there. So we built labs in the US. We said we'll focus on one problem there. We'll focus on this cancer sequencing problem. So we created this really interesting panel almost in 2014-15 to sequence tumors and report on these tumors. Published it and built up a CLIA lab in the US, started offering it. And of course, if you have to offer in the US, you have to have labs in the US because you can't be shipping samples to India. You have to have teams that reach out to clinicians in the US. So all of that started getting built. And there we ran into our next challenge. We suddenly realized that the way the whole ecosystem works in the US is our premise that we will have something that would be far less expensive than as a test would be than most companies could in the US, fell on deaf ears because the cost doesn't matter. The doctor orders, the patient takes the test, the cost is paid by somebody else. It's a third party, an insurance guy. And that guy will anyway need 3-4 years of dancing with before he'll pay for it. And nobody else pays. And so the way companies operate in the US, doctors order the test, patients take it. Insurance companies may choose to pay for it, may not. The testing companies just write it off. And they lose hundreds of millions of dollars in the process. Till about 10 years they take and they build large businesses. So that was an economics that we just didn't realize that's how it works. And we realized that we were certainly not, we didn't have the money to last, play that big level of game. So we had to exit from there very rapidly, saying this is too big a game for us to lose too much money out here. In the process, though, we realized that we had extended ourselves way too much. We had put in too much money, and if we are exiting all of that investment basically gets lost. And so we ran up loans. And that's what I call the big fiasco of 2016 for us, is where all of our investments that were done were lost. And it's like a game of snakes and ladders, right? You've spent 16 years building the business. You've done a lot of impact. You've got 15,000 publications where people have used your tools. You've got all these patients in India that are getting diagnosed because you're now able to offer them sequencing. And then, but you're now at a stage where you've got a snake that from the top is leading you all the way back to the bottom because you've run up loans, you don't have money to pay salaries. What do you do next? So, because we overextended ourselves in the US. So at this point, the big fight back began, where we sort of sold off our microarray IP saying, now it's anyway going down. We sold it back to Agilent and said we got some money from that. We paid off our loans with that. But our core IP was gone. We still had all of the sequencing and sequencing related IP. So we started with that and we started rebuilding in 2016. It was literally a rebuilding of the entire company, entire business. So I use this World War II analogy just to say it was literally like this. We were in shambles in 2016. We sort of rebuilt. And the rebuilding involved really taking all the technology and expertise pieces that we have out. And we didn't have time at this point to build new technology, etc. Right? We had to take what we had out. And so we basically, a few of us just said, we're going to just get into commercial mode. We're just going to go out, reach out. I started traveling every month to the US. We're going to reach out, connect with everybody who can be benefited and build up revenue streams from them. And we were somehow, luckily, I mean, this is not something that happens very often, but somehow over the next 3-4 years, we were able to pull ourselves out of that mess nicely by being able to, generate that business. So if you look at everything that had happened by this time, if you look at the different stories, what had happened was we had a great story with the analysis, great impact with the analysis tools, but we couldn't control the commercialization in the US. So we couldn't control our long-term destiny there. We had a great story with impact in India where we could impact patients, but we just couldn't make it economically viable. And so for these two reasons, our core ability to translate everything that we had, our creativity, was struggling. Even though we had local highs, we couldn't create a long-term, sustainable long-term model. But over the last 4 years, from 2016 to 20, perforce, because we were forced to do it, what we decided is that we will build our business in the US to be where we help solve genomics problems for lots of people. That generates revenues for us, that generates profitability for us. We use that in India to grow the sequencing business and the patient impact business to such point where it reaches a certain scale, by which point, technology costs, everything comes down, and then that will become viable on its own. So that together, we are not extending ourselves as a company, we are within our means, and we are able to continue our innovation. So this US funding India model came about in that last 4 years. And also, over those 4 years, we were able to build, or in the subsequent 4 years, or in the last 3 or 4 years, we were, and the reason I'm showing this picture is that you need a lot of different pillars to hold an institution, hold a temple up, but hold an institution up. You need commercial, you need technology, R&D, lots of different pieces, regulatory, right? And over the first 20 years of the company, one or the other piece was missing for us. If we had technology to commercialize in the US, we were dependent on an Agilent to do that for us. If we had the technology to commercialize it in India, we didn't have the capital. So you need capital, you need the commercial teams, you need the R&D teams, you need that in all, you need a commercial team in the US, a commercial team in India. Until all of that comes together and capital, everything comes together, you can't really build a sustainable organization that will be able to adapt to changes in the future. And then all of that finally came together after 2 decades because we were able to, ride through all of these different challenges, evolve, adapt, and continue to innovate, continue to learn from these and bring it together, right? So now we are at a state where all the pillars are there to hold ourselves up, and then we are able to sort of operate in both continents, subsidize one continent with the other, continue our innovation and bring that. So a lot of innovation is happening, which I won't talk about, but given that I'm at the end of my time, I'm going to just talk about the lessons learned from this story and what, as translators or as aspiring translators of science into impact, what do you need to keep in mind? The very first thing you need to keep in mind is there are many steps from science to impact. It's not, hey, I've created the technology, I've got this nice IP, I've got patented it, now somebody else should take it. And that mode can also work. You can get lucky. Your IP may be so powerful and so impactful and so directly applicable that you don't need to do anything else. But that's a rarity. Most often, IP is part of the problem. It solves part of the problem. It makes assumptions which may not be there, may not be valid in real life. It will be partial for the most part. So there are many steps from science to eventual impact, and treating the science as only the first step and respecting the process of generating that eventual impact as a multi-step process that has a lot of science and art to it is the first learning, right? The second learning is that this process of translation from science to impact, there are many unknowns. Just as there are many unknowns in your science, you do experiments to uncover those unknowns, right? To discover why those unknowns are happening or what are those unknowns about. There are as many, if not more, unknowns in the process of taking the science and converting into impact. These unknowns are of a different variety. They're of the variety of what is your eventual customer behavior? What is the economics? What is the regulatory? What is the competition doing? Are people doing things that will make your invention irrelevant 5 years from now? So if you're taking 3 years to commercialize your invention, anyway, there's only 2 years left, right? And so it's not, so all those unknowns that are there are even more than the unknowns in your core discovery. And therefore, just as in your core discovery, you do experiments and there's a method, a scientific method to getting to controlling those unknowns, there is a method to discovering and experimenting with all of these more complex unknowns and getting to the, controlling those unknowns, right? And that method is the method of translation. And that method itself has a number of ingredients. One, you have to know what all the unknowns might be. You may not know what those unknowns are, but you need to know which areas they might be. They'll be in the competition, they'll be in the understanding of the customer or the consumer on what they really want and whether your core invention can actually translate into something that within a certain economics will be able to reach out to them, etc. There will be the process of commercialization. I mean, the customer may be all willing to buy what you have. You may be all willing to, you may have all the right offerings, but the friction on the channel between those two has its own complexities that one has to unravel, right? So to experiment with all those unknowns and exactly as you do in a lab experiment, to experiment out there and feel and control all those unknowns, you need a number of things. In a lab, you need only 1 or 2 or 3 disciplines. Now you need multiple disciplines. You need these well-balanced teams, right? Teams that understand the commercial world, the economic world, the maybe the supply chain world, the technical world, the regulatory world, because there are unknowns in all of those. And if you leave any one of those unknowns, as several of our examples are history, if you leave one of those unknowns, like in our US journey, we did not understand the payer behavior well. And because we didn't understand the payer behavior well, our economics just went out of the toss, right? And I gave you that example. So if you miss one unknown, you can come down like a snake all the way, you would have climbed up a lot of ladders and then one snake is all you need to come down. So well-balanced teams that cover all of the unknowns is the key. Once you have teams of all these different types, they will all head in different directions by themselves. So that's what I call entropy. Everybody has a way of looking at the elephant. Somebody look at the tail, somebody look at a head. In any conversation, you watch that conversation, you'll see this guy is only talking about the tail, that guy is only talking about the head. Somebody has to integrate that picture. And so you need leadership, you need to control that entropy on an ongoing basis. And this is a non-trivial task. I mean, this is, and then, of course, as I said, the world is dynamic. As I said, we created a great impact using microarrays, and then sequencing took off and our elephant just couldn't react in time, right? So the world is dynamic and time is risk. So speed and adaptability is key. You have to constantly watch what the world is doing and evolve. So, and then along the way, all of this is not going to come for free. You need to have the money to be able to do this. So you will need to refuel many times along the way. Even if you get a grant initially, there are so many dynamics here that you'll have to refuel. And your thought process has to be, how do I refuel? How do I've gotten to this petrol pump, I've gotten my fuel in. The next petrol pump is 100 kilometers away. What do I need to do to get to that next petrol pump? Who will give me that money? How do I set their expectations? How do I build their confidence? And that is an art in itself, right? Because, I mean, it's very hard to predict when you will get to that next petrol pump. You don't know in real life whether it's 100 kilometers away, 20 kilometers away, where is it? So there are many challenges, as I said. And because these challenges are there, as the ecosystem builds up, maturity builds up, the method also builds up. Just as we have the scientific method in the lab, there's a method here of experimenting with these real life variables. That method, unfortunately, we don't talk about much. We don't systematize it much. Maybe some business schools write here and there about it, but they don't understand science well enough to be able to connect it. And so what the connection happens when you have this ecosystem of translators who have done this and who are able to engage on both ends of the spectrum and bring that method in. There is a solid method in here. And that method, unfortunately, we learn the hard way. But all of this is worth it because the eventual reward when you're able to take a patient who's struggling for a diagnosis and give them a diagnosis, when you're able to generate tens of thousands of publications through the tools that you've had, that is satisfying at some level, right? And there's just so much more to go because so many more health problems to be solved. So that is what makes it all worth it. And best wishes to this institution and all the people who are seeking to translate their discoveries into the real world, and I'm sure you'll all do well. And some of these lessons are hopefully helpful as you go through your process. Thank you. Questions, or do you have time for questions? Or okay, I'm happy to answer any questions. Yeah. So you have to have your invention, technology, whatever is it, and then you have to go out and talk to people about it, right? You got to see, nowadays you have social media tools that are far more, but then with every tool also comes a lot more people pushing, so the noise also increases. So you have to figure out how to get your signal above that noise in the commercial end of things. So you use various tricks. You hire people who sort of in from the ecosystem who have that network. You sort of get advisors who have that network, you start connecting with them, and then if your technology is strong enough and the need is strong enough on the other side, connections will happen relatively fast. If either of those is not strong enough, it will take longer or you'll have to evolve faster. But that's how essentially we got to the first point. Yeah.
Really amazing talk. So how are you able to have the team together? There were multiple dips in the process. Was it very difficult, challenging? How was your experience?
It is, you've got to constantly lay out the vision, right? And say that while we are in a dip, things are not so good, but we are still where we are heading towards in the longer term is still rewarding. We'll last through this, etc. Right? And there is a path. Not that people didn't leave. Many, very, very good team members also left, but we were able to keep the core team intact, and that requires. Why do teams stay together, right? I mean, teams stay together because they see the difference between an enterprise like this and a large enterprise with bureaucracy where other people are making decisions, where things is that here they can be the masters of their destiny. The only thing that stops them being masters of their destiny is all the unknowns in on the dynamics of the ecosystem and the world itself, not some bureaucracy. And once you sort of bring teams to that level where they are the masters of their destiny and they can do something about it, I think that is a very strong driver, I find, in that overrides a lot of things, including salaries being not for periods of time not cutting edge, other people chasing them, the Googles of the world chasing them, saying, come work with us, etc. Right? This notion of being masters of your own destiny is a powerful motivator.
Hi, amazing description of an amazing journey. So I just wanted to know, in case of US market, exploring the US market, which was more critical or more difficult, having a clear lab or getting the payer behavior sorted?
Well, undoubtedly the second. The clear lab is a deterministic. You can predict. You start today. 1 year from now, you'll have everything in place. What you cannot predict is everything that you need to do to get, and that process is so complicated. I can tell you, the only way to run that process in the US is either go through FDA approval, which is like another 2-3 years of a lot of expense, or you just start the commercialization and eat the losses for a few years. And all these companies that we were competing with were losing $150 million every year, not batting an eyelid on it. And that's not something that in India we have the wherewithal to do. Rarely any company, I mean, we have the e-commerce companies that have now shown us that path also, but not in these sorts of scientific worlds where people are willing to take that sort of trust. So undoubtedly the payer, and that economics took us a while to realize the complexity of that economics.
Thank you. Very inspiring talk. So since it's a technology-driven company, and having co-founder in technology-driven company is very important. So what their challenges on that front? Finding like-minded co-founders. Yeah.
Yes. So two parts to this I will answer. Having a team is absolutely important. I mean, there's no way you can do all of this yourself, even if you had the skills, right? I mean, the chances of one person having all the diverse skills that are needed are anyway low. But even if you had the skills, there's so many ups and downs that you need sounding boards. It's a very lonely journey as an entrepreneur if you realize it, because only a small team realizes all the issues. Everybody else is not able to really appreciate that same level, because their mind has shifted from being an employee to saying that everything depends on me. I've got to make it work. If it fails, it's on me. If it's if monthly salaries have to be paid, I have to be responsible. It's not that it'll come from somewhere else. That mind change only a few people make, and once you make that mind change, your whole approach to everything changes, right? I mean, it's your baby. You have to make it work whichever way. There's no pointing at anybody else. There's no blame games. There's anything can go wrong, and there's nobody else responsible for you other than yourself, right? So that mindset is a lonely mindset, and only a few people get into that mindset. And having a few people there with that mindset, so you can brainstorm and go back and forth, is very important. So the team is very important in that sense. We were lucky enough that we were 4 of us who started, but then I think we made one among various things. We started 2 different companies. You would have heard of the Simputer effort, perhaps, if 2 decades ago. So two of us went off into that. So there were only 2 of us, but 2 was good. We continue to be there together in different capacities, but that was very important. But then we were able to build a core team thereafter in 2 or 3 years. Many of that whom are still there so that core team was extremely important. The other flip side of the story is the core team that we built was very unidimensional in its thinking and in its skill set. We realized that, and if we had a more rounded out team that could look at things in many different ways, that we built over the years, much later in our journey, we were able to build. But if from the beginning we had that commercial mindset, a deeper analytical understanding. Commercial, we often think of commercial as a bad word, right? It's about selling. It's not about selling. It's about a different ecosystem whose behavior has to be studied and which has its own rules and which has its own methods that have to be incorporated in your mind, and you have to start thinking and computing on that world. And so that is a different world which we did not have, and I think we paid the price for it and we built it eventually.
Very inspiring talk, sir. So my question is, like, we are hearing some of the best startups who had the success for some time, and that this term always comes, it takes 2 decades. to reach the success. And I believe 20 years back, the startup was very new. And for the last few years, the startup has come in India in an enormous way. And in 2023, there are around 92,000 startups had registered, and 50% are in the life science. So how you advise somebody would like to, enter into the spin up at this stage, and how you see the sustainability of the startups in the longer run?
Yeah. So first question is, if you're passionate about it, you have an idea, just do it, right? That's the Nike tagline. Just do it. Do not overthink. I mean, everything that I've laid out, unfortunately, it's about overthinking that process. But then, having been through the process, I might as well down all of these things so that it's not lost. But when you're starting up, do not overthink start, right? Number one step. Number two, once you started, learn as much as possible from everybody else, their failures. Don't let it discourage you, because every path is different. One of my realizations consistently has been, we've had all these advisors over the years, right? We go to different people, some are academic advisors, some are, industry, various types. Almost every time I've realized that within 6 months, that person has nothing more to contribute to what you have, right? Because your journey is so new, it's so different. I mean, you can't blame that person. That guy has his own journey. He's learned, he's very accomplished in whatever he or she is doing, right? Every journey is different. And what you have to do is to learn from many, many such people, right? So talk to as many people who have traversed the path, people who are traversing the path, learn from them. Don't get overly influenced, learn from them, take tidbits from them, and then juxtapose that on your journey, your ecosystem, your setting, and see what it tells you. And then, let your heart drive those decisions, because time and again, we've had many people say this or that, but we can't see how that applies to our world, right? And so it's not as if these recipes are ready-made. I think there is a method that is ready-made, though. That method you can learn. I think there is something to learn in that method. Applying that method, just like we say, the scientific method is a method, right? But if you're running your experiment versus somebody else running their experiment, will be very different thought processes. But the method is a method that, to that extent, you can learn. I think there's just so many problems to be solved that if you take anybody with a health problem today, chances are, I would say, put that figure at 90%, you go to a doctor, you will not get an answer, right? So there are just so many problems to be solved today that unless more and more of this happens, we will probably not, make a dent. So the opportunity is tremendous. Challenges are no fewer. Particularly in a country like India, where the healthcare ecosystem is very immature, it's a little harder. But you have to chart your path and believe in the fact that if you do the right set of things and right set of dynamics, then somewhere or the other, you'll be able to find a feasible solution that will drive your idea forward towards its eventual impact. You will have to build quite some dexterity for that, though. I mean, 90% of companies may not succeed, but if you have a good team, if you are able to nimbly adapt, you have all the multidisciplinary skills that are needed, then the chances increase.
That's very illuminating, Dr. Ramesh. Thank you for giving that. I have many questions, but I'll ask you just one. So if you were today asked to advise the government of India, for example, you work both in the public sector as well as the private. So what are the things that you would ask them to facilitate to allow entrepreneurship to take off in this country?
Yeah, so there is a cultural thing, and then there are, of course, things have changed a lot over the last several years. I'm seeing, there's more and more government programs to encourage entrepreneurs. But there's a cultural thing, the cultural thing about industry versus academia, industry being seen as primarily a profit motive world, academia being seen as primarily a social good. That has to change, number one, right? most industry loses a lot of money. I mean, the mature guys, the Tatas and all, of course, they'll Reliances, they'll make hand over fist, etc. That's a different world. I mean, if with that money, they're not able to solve all the health problems that we have, then what is the point, right? I mean, they are not able to, right? I mean, today, I mean, if you have cancer, they're not, they have all the money in the world, but money is not able to solve the problem. So there's something else here, right? And that only translation, youthful entrepreneurship, whatever you call it, something that is what will make headways there. So that world loses money hand over fist. Most investors will lose money on most ideas. One or two companies, if an investor invests in 10, one company will hit it through the roof for them, and they will be able to, make returns, and that whole ecosystem will continue to be fueled. Those 10 companies will lose money, So this notion, the government has to start thinking about entrepreneurship, not as a profit mechanism, but as a social good. Because as I said, most ideas will fall by the wayside, right? Number one. So number two, in life sciences, now this is very different in technology. Technology life cycles are short, right? 5 years. You can start building a piece of software, and in 5 years, you can take it quite some distance. Here in the life sciences, the life cycles are much longer. I mean, I would say at least 10 years. And if you make 1 or 2 mistakes, then 10 can become 20 easily. So recognize that 10-year-old company could also be, for instance, a startup, right? Because those ideas are still, you need continued support to get there. It's not, these life cycles are not very short. And you want to be able to incubate more companies, but you want to take promising companies and take them to the next level and take them to the next level. And then drop them into the free water at some point of time. At some point of time, they all have to swim for themselves, right? So that process has to be, that period has to be longer in the healthcare life sciences space. The third is, I've seen this happen in countries across the world, in the Western countries particularly. There are large government programs. A lot of funding goes into those programs. In India, those programs almost exclusively get reserved for the public sector, the academic sector, right? Startups don't play too big a role. And to give you examples, right, if you look at Genomics England, the sequence 200,000 genomes, the infrastructure that they created of private and public enterprise. So the deliverable from that was what? That deliverable was an ecosystem of public and private enterprises that were then able to take that engine forward. Right? In India, when we invest in these large programs, I don't think we think enough about what is the ecosystem of private enterprise that will be a deliverable at the end of this process. We only think of, are we building capability, or we largely think of, are we building capability in our academic institutions, which is very important. Are we building, enough publication record, very important. But are we building enough private ecosystem that has now emerged from the dark as a result of that platform and the investments made in that platform, that they can then translate all of these things that we're not thinking about? So those are the three things I would say that we should think hard about.
So Ramesh, fantastic. Continuing on the question that Karthik asked, I'll ask it perhaps in a more pointed way. Sometimes in Bangalore, they say, one of the great reasons for the growth of IT was nobody in the government actually thought IT was going to become that big. And nobody was trying to create very large public ecosystems that would eat up the funds and create competition to the young entrepreneurs who started it. Now, of course, investments required were less in that particular ecosystem at that phase in time. Today, again, the biotech boat is rising. How far, that previous analogy, that things actually do better when there is less thought about them and there is general growth occurring all around, would you say is true for the newer areas which are investment heavy?
So the IT industry is a remarkable success, but it's also an unfortunate model, because if you look at that model's success, it was based on essentially a price arbitrage of smart demographic that we built through our investment in education, costs of a corresponding demographic in Western economies being very, very high, and the demand for IT going through, because whatever you do, right? If you do biotech, you do e-commerce, you do your selling vegetables on the street, you will need IT. So IT just transcends every industry, and the demand, the volume of effort needed there is so large, and everybody changes their mind. Today I want a button here, tomorrow I want a button there. So some human has to come and do it. Just too much there, right? So I think we just got lucky there that we had that large demographic. The price, the cost differences were substantial. Will such an opportunity present itself on a platter again? I doubt, because while IT required skill, that skill is going to be far less, or was far less, than for us to capitalize on future opportunities like that. Those skill levels are going to be higher, and we are not going to be able to train nearly as many people to capitalize on those skill levels. The markets, the market naturally was in the US. US companies were paying, Indian companies were serving them, led to beautiful growth, and led to this great platform on top of which now we are innovating, right? Which is great. But for example, in the biotech world, is that likely to happen? Do we have the skill levels by which a US company that's taking the risk, right? A big pharma company spending billions of dollars that's taking the risk will shovel billions of dollars worth of investments into India. I don't think we have the skill levels for that yet, and the numbers for that, right? So I think we have to do something differently now. We have to look at local markets. We have to look at building the skill levels through these large initiatives. And once we have platforms like this, we'll have to pick our battles perhaps, but once we have platforms like this where we are the best, we've got so much going, then that engine will get unlocked, hopefully.
Hi. Thank you for the nice talk. So I have a very different question from the scientific temperament, in a sense, because you work in India, you work in US. So you are coming from a very different background, and then you are applying on a very different background. So I just want to ask, how people from this different background can convince each other? In the sense, there are some routine things which both of them knows that, okay, these are the things we can take help from a computer scientist, or these are the things we take help from a biologist. But somebody want to do some innovation in each of the field. Then how you can convince the people from the other field that, okay, this is something innovative and you have to do it, or you can help me and something like that?
Yeah, I mean, the answer is primarily you'll have to learn the language of the other field. So my point is always there are no walls now, right? It's, yes, our programs force us to, and we need structure in our educational programs. So for 4 years, we'll do a BTech in something. For 2 years, maybe even 5 years, let's say we'll do a PhD in something. But that is just 5, 7, 10 years of our life at most, right? And then the rest, 50 years of our working career, let's say. I mean, why should that hold us back into saying, okay, I can only do that, right? I mean, there's so much more time you're spending. If you're learning at least the same rate, I mean, you should be learning at least the same rate, because you're wiser now. Then you should be able to learn, whatever is needed to learn the other guy's language, start talking to them in that language. You'll also have to learn the unifying language. If you're in academia, the unifying language will be whatever, publications. If you're in industry, it'll be the commercial element of things. And that unifying language also you'll have to learn and, put it in those terms. Then everybody understands. And so, in other words, don't be, just because you did biology for the first 10 years of your educational career doesn't mean that that's all you should do. Just evolve. I mean, it's all about evolution now.
First of all, thanks, and quite intrigued in terms of engaging discussion. So my question is regarding to your entrepreneurial journey altogether. So if given a chance to time travel back to, 16 years before, and you still have to travel that journey again, which 2 events you think, distinguished events as per the prime concern, which you feel as an importance, could have been dealt differently as per the person you are, and you go back and then change the things? Those 2 events, if you can describe.
I mean, in the context of the company you're saying, or in general in my evolution?
overall, I think, in terms of what you feel it as a prime importance, both in terms of the company, maybe the ROI or the kind of different kind of portfolios, or anything which comes to your mind?
Yeah, I mean, the thing on the top of my mind is clearly what I wish I had done all along the way was to understand all the unknowns much better, much faster. And unknowns are there every day, new unknowns show up because the world is changing, right? I mean, it's changing so fast in the areas that we are in the life sciences. And just so every day in the morning you wake up, you get 700 papers in your feed. I mean, you go through the abstracts of them quickly to say what's happening. I mean, you can see 5 different directions that could potentially impact your future process. So there are so many unknowns here that I wish I had known this method. The method is to control the unknowns. To control the unknowns, you've got to feverishly work on understanding the unknowns and figuring out how you can control them as quickly as possible. Assumptions I've made along the way saying, hey, let's do this and this will work out. And only to get egg in my face and then look back and say, these unknowns were obvious. Why did you not look at these 5 things? I mean, right? So that is my biggest learning. Feverishly work on unknowns and try to make them the known. And that has manifested in many different ways over and over again. But at some level, the fun and the forward journey is to ignore everything. I mean, that's humans have been made this way, right? Little children, you tell them walk in a straight line, they'll go here, they'll go meander, because part of it is to just meander aimlessly. That's how discovery happens. But then you can fall off a cliff by meander. So that balance is an interesting thing that all of us, each of us, has to strike. But some amount of learning, controlling unknowns is very important.
Thank you.
I'm going to ask a more general question, maybe not your domain, but in an entrepreneur world, at least for low middle income countries, why is it that diseases which have more public health importance, particularly around maternal child health, there isn't so much excitement? I mean, I know it's not your domain, but it's,
no. I mean, it's
coming from
it's all of our domains in that sense. But why is there not so much excitement?
And larger government programs, there's huge amount of funding going there.
Sets to make programs that are sustainable. Eventually, the government has to be a capital providing organization, right? The government provides capital, goes for 5-10 years, and in that process, an engine ecosystem builds up that the economies of that ecosystem then solve that problem and continue solving that problem forever, right? So we have a number of challenges. We have the challenge of the fact that we are not a rich country. There's poverty. There's not enough money to perhaps solve all of these problems. But the reason why there isn't excitement is that I think one element of that is simply that we have not thought in this way, that we've got to build sustainable ecosystems with one-time investments. And so let's get as much of as many minds into that ecosystem so that there's a chance of it becoming sustainable. There is still an element of control where we say we want to control it rather than we want to make it sustainable, right? So COVID times, beautiful example, right? I mean, do you want to enable everybody to be involved in COVID research so many people can sequence and foresee the Delta type waves, etc., or do you want to control sequencing because potentially it can fall into bad hands and bad things can happen? So generally our feeling is we should control a little bit more because bad things can happen. If our thought process is that, okay, here is a maternal health problem, we've got to solve this. We have 5 years and whatever, 1,000 crores, 10,000 crores, whatever, ascribed to that. At the end of that, there has to be an economics that sets in, where there's private ecosystems, there's public health clinics, and between all of them, there's a sustainable, viable ecosystem, right? Perhaps it's non-trivial in a country like India because then, primarily you'll have to solve the employment problem to eventually solve this problem. And that is a much bigger discussion, right? But just looking at this problem alone, if this thought process sets in, but then in certain areas, the excitement will build because then you'll have an outlay of capital, you'll have, you can invite matching outlays from industry, you'll have private industry participating. That will build up that excitement. And then solutions might come, which are not coming otherwise because so many people are applying their minds now. Because once you have a lot more people involved in the process, hopefully, all you need is one Sachin Tendulkar somewhere, right? I mean, and only one of those shows up amongst a billion. So by just involving 100 people, you may not find, but you involve 10,000 people, 100,000 people, somehow you might find that person, that problem may get solved, right? No. So I don't have an easy answer to your question. At the end of the day, primarily, we have to solve a lot of economic problems. But I think thinking about creating platforms where you have a certain finite amount of investment. At the end of that, you expect things to become self-sustaining through the participation and incentivization of both private and public sector right from the beginning, has a better chance of success than what we're doing today, most likely, is my guess. Again, we have to experiment. Like everything that I've laid out, anything, all these are unknowns. You have to experiment with all of these things, figure out what works, what doesn't, tweak those experiments. The scientific method applies in every realm of life is what I find. So just have to apply that.
One last question before we, so this will offer a segue into the next session as well. So at any time in your journey, did you feel handicapped? You said lots of unknowns, right? One of the unknowns that we as clinicians feel is that there is an incomplete understanding when you don't involve all the parties. So did you, in your journey, at any point in time, feel that it would have been better if you had involved a clinician in the beginning?
No, tremendously. I mean, the constant problem on an ongoing basis is every idea starts with somebody, right? Somebody has to champion. That champion will come from a certain angle that will best reflect their skills and their training. But to convert it to a full solution and a full something that can make impact, there's just so many unknowns which come from all these other disciplines, including different technical disciplines. I mean, in our line of work, I see at least, I mean, there is molecular biology, there is statistics, there is algorithms and software, there is the clinical world, and then there is all this whole commercial thing, and then there's the regulatory. I mean, there's just so many disciplines that have to come together, right? So constantly we feel ourselves constrained that we know 3 out of these 10 very well, and the remaining 7 we are taking guesses on. We are going and talking here and there to people, but those people are not so invested deeply that they can understand your problem to the extent that you need them to, right? So people who can crack that problem will be more successful, I can tell you that much. So you have to somehow engage a lot. I think you have to engage a lot more, and most engagements will be superficial. You can sort of keep them, filter out of them the few that are going deeper and deeper and deeper, and then see how you can really put together all facets of that problem. But yes, that is the biggest problem to be solved, I would say.
Thank you, sir. That was an inspirational talk which has rekindled the scientific enthusiasm in all of us. Now, I request our Executive Director to kindly felicitate Dr. Ramesh Hariharan. Excuse me. Felicitation.
Welcome to the Clinician Scientist Conclave, a gathering where the art of healing converges with the precision of scientific inquiry in a symphony of innovation and compassion. Amidst the ambience of focused inquiry and shared purpose, the Clinician Scientist Conclave stands as a testament to the synergy between bedside practice and laboratory investigation. It is a sanctuary where medical intuition meets rigorous experimentation, forging a path towards a future where every patient's journey is illuminated by cutting-edge science and compassionate care. Now, I request Dr. Nitya Vadva, Senior Professor, BRIC THSTI, to kindly conduct the Clinician Scientist Conclave.
Thank you. Good morning, to all the esteemed guests, colleagues, and students. It is my absolute pleasure to welcome you today to today's panel discussion on a topic that lies in the heart of advancing human health and medicine, the integration of basic and clinical sciences for impactful biomedical research. In the realm of biomedical research, the confluence of basic and clinical sciences is not just a frontier, but a necessity. Basic science lays the foundation, uncovering the intricate mechanisms of life and disease. Clinical science, on the other hand, translates these discoveries into tangible medical advancements that improve patient care and outcomes. Today's discussion brings together a distinguished panel of experts who have been trailblazers in bridging these two vital domains. I would like to invite on stage Professor Anurag Agarwal. Professor Agarwal is the Dean of Biosciences and Health Research, Trivedi School of Biosciences, Ashoka University, Delhi, India, and former director of the Institute of Genomics and Integrative Biology. After completing graduate medical education at the All India Institute of Medical Sciences in New Delhi, he further trained in internal medicine, pulmonary disease, and critical care at Houston, USA, followed by a PhD in physiology. His primary research is in respiratory biology, and broader interests are in the new vision of health and healthcare seen through the lenses of emerging technologies. He serves on numerous national and global advisory boards, most recently chairing the World Health Organization Technical Advisory Group for Viral Evolution. He received the Shanti Swaroop Bhatnagar Prize in 2014 and is a fellow of the Indian National Sciences Academics. Welcome, sir. Professor Govind Makharia. He's a professor in the Department of Gastroenterology and Human Nutrition at the All India Institute of Medical Sciences. He has trained 102 fellows and 9 PhD students in gastroenterology and edited a book on celiac disease. Dr. Makharia is presently the president of the Indian Society of Gastroenterology and has been involved in various leadership roles in both global and regional professional organizations. As an educator, he has initiated and nurtured many education programs, namely the Young Clinician Program, Evidence-Based Gastroenterology, Gastroenterology Young Masters, Train the Trainers, and master classes. And he's a member of many national academic committees and institutions and has received many awards. Professor G. Karthikeyan. Professor Karthikeyan is the Executive Director of THSTI Faridabad. He is a clinical cardiologist and a professor of cardiology at the All India Institute of Medical Sciences. He completed his training at AIIMS New Delhi and at McMaster University, Canada. His research is mainly focused on cardiovascular diseases affecting the low and middle income countries, including valvular heart disease, particularly rheumatic heart disease, mechanical valve thrombosis, anticoagulation, and indigenous drug-eluting stents. He is one of the key leads of the Invictus International Research Program. Professor Jayanta Bhattacharya. He is the Dean at Translational Health Science and Technology Institute and was previously the Director of IRV and Program Director of the THSTI IRV HIV vaccine program. He has more than 25 years of research experience in infectious disease biology, and he played a major role in helping the National AIDS Control Organization, Government of India, towards formulating and implementing rapid detection of HIV from dried blood spots obtained from infants and children and played a central role in implementing the same in national programs, as well as establishing different regional laboratories with capacity building. He's contributed significantly towards understanding in genetic and functional diversities of HIV grade C, the major circulating subtype globally, and in developing broadly neutralizing monoclonal antibodies as products for prevention and treatment against HIV and SARS-CoV-2, and very recently, the first indigenous potent human monoclonal antibodies to deadly Nipah virus. Welcome. Professor Amit Awasthi. He is a senior professor at THSTI and heads the immunobiology and immunology core laboratory. He's an internationally renowned immunologist with significant research contributions in the field of T-cell biology in inflammatory and infectious diseases. He has been conferred with many prestigious awards, including the Innovative Young Biotechnologist Award and G.N. Ramachandran National Bioscience Award presented by the Department of Biotechnology, the DRI Award for Excellence in Drug Discovery Research, G.P. Talwar Young Scientist and G.P. Talwar Mid-Career Award by Indian Immunology Society, and the Young Scientist Platinum Jubilee by National Academy of Sciences of India. He is a founding member of Immune Oncology Society of India and an executive council member of Federation and Immunological Societies of Asia, Oceania. Once again, thank you to all the panelists. And we're going to do this is first, there are going to be two talks, one by Dr. Makharia and then by Professor Awasthi, and then we'll have the panel discussion. So could I request Dr. Govind Makharia to please make a brief presentation on his viewpoints from the clinician point.
Thank you, Dr. Badwa. Thank you, Dr. Karthikeyan, for kind invite, and very good morning, everyone. I think this is a wonderful opportunity to discuss or integrate clinical and fundamental science for advancement of health sciences. So once I see a patient every time, it's not only me, there are 100 other people work with me that point of time. If I meet a patient, there are ward sisters, there are lab technicians, there are labs, there are pathologists, there are radiologists, and all together we make a difference to the patient. Not one person. And this we learned from the very beginning, that one person cannot treat a patient completely. It's a team of people who works together to treat patients successfully. And that's a very fundamental point of doing anything good to anything. This system was understood a long time back, and it was a good integration that in hospitals, there has to be all departments work together to provide a holistic care to the patient. But once you come to science, look at all these things which happened to our country recently, and all of them was a teamwork. It's not one person. It's all a teamwork which led to great success to our country. But look at what happens many times, that we work in silos. And Dr. Hariran just said that entropy, that team is important. Everyone has a role to play, and all role comes to integration to a solution. What we do generally is we live in our own domain. We're working in silos. So this is my domain, I work in domain and close there. Just lead to failure, and the story doesn't move forward. What we need to go is like an integration. Means that everybody works towards a common goal. The Chandrayaan was a common goal, and everybody worked around that with success. Unless we do that, it's going to be a failure. Therefore, for success, we need to have a collaboration with institutions and with people with different abilities and expertise. And this is the fundamental of doing a good research. I'll start with a couple of clinical scenarios and give you small stories, and how the question arises in the clinic and how we together in the fundamental science can answer those questions. So this is celiac disease, we all know. This is a disease occurs because of allergy to gluten. A certain number of people around the world cannot digest gluten, and therefore they have a small intestinal damage, and that leads to multiple health issues. and it can affect any part of or many organ of our body. So in early part, say 1940s or 30s, we didn't know how to treat this disease. And the treatment was only giving this patient bananas. This was the only treatment available in, say, about 8 years or 9 years back. This person, he's a pediatrician, the William Carroll Dickey, once he was sitting in his clinic, one of the patient's mother told him that my child is good if I don't give him bread. And that this doctor took that simple narration from the mother and worked for 10 years in his hospital. Just 5 patients, 10 years, and he could prove really that indeed the gluten or the wheat is the cause for this disease. And over time, we confirmed it by doing more biology, that how it happens. Therefore, the essentials, what we learned from this very humbling story of William Carroll Dick, that humble and kindness is the prime of discovery. Open-mindedness, self-conviction, and more important, desire and dream, all together required for doing a good science. So celiac disease, as I said, was thought to be very uncommon, but by multiple studies in the country, we could show that celiac disease is an important disease. And the question comes, how does it occur? And we know that this happens with two important factors. One is the gluten protein, and second one is that genetic susceptibility. And if both of them there, this disease can occur. What will happen? Gluten is a large protein, so each protein has to get digested to get absorbed, but this protein is not digested. And therefore, there remain peptides. So these peptides become immunogenic in those people who have a genetic susceptibility. And these peptides leads to T cell activation and then damage to multiple organs. And therefore, if you can give a cartoon like this, suppose this is a peptide. So once patient ingests gluten, this peptide, which are partly digested, they pass through the intercellular junctions. So these are intersites, these are lining of a small intestine. So between 2 cells, these peptides enter. There's a small protein here that's called tissue transglutaminase. This deamidate, this peptide is deamidated, become negative charge, and that's presented to APC recognize them and present to T cells through HLA, and binding of HLA occurs. And it's also important that the binding of this peptide with HLA is the key for presenting this to T cells. And subsequently to that, there's a T cell activation and this T cell clone expansion followed by circulation of these T cells around the different organs and can lead to a damage. So these T cells, once become active, they produce a lot of cytokines, B cell activation, antibody production, and damage. So now the T cells become the center. These T cells, which are gliadin-reactive T cells, like COVID T cells, they are very specific for celiac disease. The question comes that these T cells, if I can recognize, we can make a diagnosis, and also we can do a lot of pathophysiological studies. And these T cells can be detected by tetramers. And these T cells, which are the center of celiac disease, if you stop gluten, there's no T cell activation, you treat them effectively. At the same time, this T cell can be used for diagnostics. And this T cell diagnostics are so important. Therefore, we need to have our own infrastructure to create this T cell detection. And with Amit here at this institute, we came to him and asked that can we detect these T cells. So we are doing this T cell detection, or this T cell can help us make more diagnosis, and it has a lot of other implications. Therefore, a critical question arising in the clinic, and with a collaboration with a fundamental scientist, we can do much, much better. Second question comes, I'm again taking one more story from celiac disease, or one more question for celiac disease. Once you make a diagnosis of celiac disease, you look at biopsy from a small intestine. And this biopsy is invasive. This invasive biopsy is difficult to do because it requires expertise. And as I said, celiac affects 1 in 100. It means we need to diagnose celiac disease at small town or small city where there may not be industry available. Therefore, can we have tests which can detect intracyte protein and tells us that, yes, we can diagnose celiac without endoscopy by doing a non-invasive biomarker. Non-invasive biomarker probably one solution for this entity. And we have shown that citrulline, which is a protein, which is the amino acid, is from a small intestine, intracyte. It synthesized in the intracyte. If there's a mass of intracyte goes down because of the disease, the citrulline level will go down. And we proved that in celiac disease, the citrulline level goes down. The question is, citrulline is detected by HPLC. Again, HPLC is not available everywhere. So this can't be a chemical test. It is a test that's required high expertise. Therefore, we thought, do we do something? So one day we were sitting with BLV Prasad, which Anurag you introduced, and I was talking to him that I want to do citrulline, but HPLC is too difficult. This can't teach labs. He said, let me think over it. And over a couple of months, he came back that he found a small chemical, which is called phenylhydroxine. It can bind with citrulline, and with using fluorophore, we can detect citrulline levels. This part is being done. So just cross-talking with your colleague or friend give you a solution sometimes. And this is one example of that. Further, we want to have new protein. I mean, intracyte is a specific cell. It absorbs. It has a lot of proteins. If the cell die because of whatever cause, these proteins will be there in the circulation. Can we detect these proteins in the circulation and make that use of that protein as a diagnostic? And like ALT, like SGPT, which we do often for liver disease. Liver cell die is ALT release, blood levels increases. That's the diagnostic, common diagnostic. Same that can there be a protein from intracyte which on damage of cell releases circulation and detect without endoscopy. So this work is done with the Santanu Gupta at IGIB, and we found almost 12 proteins which are potential to be a diagnostic marker. And this process is undergoing a phase of validation stage. So we hope that some of these proteins, either alone or in combination, might give us an opportunity to detect enteropathy without doing an endoscopy. Then another small example I want to give that which led us to a good question. But celiac disease occurs in those people who have HLA DQ2 or DQ8 haplotype. And the whole world believes that DQ2, DQ8 is essential for celiac disease. Once in our multiple studies and a large multi-center study of over 23,000 people, we observed that of all those who have celiac disease, 30% of them do not have a DQ2, DQ8 haplotype. 30%. Globally, 5%. Why? Because the disease has happened, it means the antigen have been presented to T cells. Therefore, why it is so? And we observed in a study that it could be DQ4, DQ7, DQ8, or DQ9. So the question is, why? DQ2 and 5 are essential, but we are finding DQ4, DQ7. So there is always homology. And to show that homology, we tie up with other colleague at IIIT, and now we're looking at the model of HLA, that how the binding of this protein happens with this HLA molecule. Therefore, this kind of integration, a question arising from clinical bedside, can raise a good question, and collaboration can lead to answer to some of these questions. Some of these questions could be big and can change or could be of value in human health. With that, I'll use a couple of minutes time to through the SWOT analysis of Indian science. What happens, how we practice today, and how should we do? We have a huge burden of diseases, so no doubt about it. So our needs are that we need to have diagnostics, we need to have therapeutics, we need to have surveillance strategies, we need to have a diagnostic algorithm for various diseases, and also we need to have devices. These are the scope of work. But, and we have a strength of our institutions all over the country. I think we have a fantastic institutions. And this being one example, there's a great institution, Karthik. We have committed scientists, we have wonderful infrastructure, and we have wonderful questions. But why we are not able to answer these questions? Why we remain, why we just publish, but we don't have solutions? And these are because of our weakness. Our weakness is that our mindset is project mode, is not mission mode. The Chandrayaan was a mission. Everyone worked together for a mission. We work in silos in a project mode, 3 years, 5 years, and then that's end of it. Lack of connect between clinical science and fundamental science. We work in parallel way. Parallel things never result into solutions. Most of these are basically initiated. They're not somebody looking at from country perspective, what my country needs. All what we do in cross-section, whatever I will believe I do that. It may not be valuable to the country, may not be valuable to the country, may not be valuable to the country. Lack of collaboration and also restrictive collaboration with industry. There has to be connect with the industry because the whole ecosystem has to be connected, as Dr. Hariharan already just said that. Therefore, we have great opportunity, we have great questions. We have to move from project mode to mission mode. And we need to prioritize. And there are many, many such issues. There are multiple threats, and the threat is that our mindset. I think I like to place just one mindset, that our mindset threat is that we are all inclined towards our own self. Our own question is my lab does this. This has to change someday. With that, I'll stop.
Thank you, Dr. Makharia. Now I'd like to invite Professor Awasthi to give his viewpoint from a biologist perspective on the importance of this integration.
Thank you, Nitya. As Dr. Makharia has already very nicely alluded, if we really need to solve a public problem, a public health issues, I think there has to be a marriage between a clinician and the basic scientist. And this has started happening in India, but it was not the case before. And luckily, I was fortunate to have got the taste of such thing back in my postdoc days when I was working in a fundamental lab, which is dealing the question of fundamental immunology, how T cell behave in various preclinical models. But I was really fortunate to be surrounded by all big clinicians who was really giving the perspective of what we are doing in the animal or preclinical research to ask the question. So this is something when I came back, and this was the mindset I already had made back in my those days, and I was really fortunate to landed in THSTI. That was Dr. Bhan was very instrumental in creating this niche and the center where such people can, basic scientists can interact with the clinician and really ask the question which is relevant for the public health issues. I mean, it's very important to understand the structure of DNA, and I think that is the revolution in the recombinant DNA technology happened after the discovery of double helix. But we have to really capitalize those basic research things and put them in the clinical perspective. So my take here is understanding immunology in disease for advancing medical research, a collaboration between a clinician and the basic scientist. So this is, I will give you a couple of examples of my journey and how the clinical physicians or clinical scientists were really instrumental in the way of my journey, and that's how we able to address some of the key questions. So basically, my lab is really revolving around these major 4 areas where we have a preclinical animal models to understand the fundamental questions of immunology, which we cannot really do in using human settings because there's a limitations. You cannot really manipulate, human, make genetic deficiencies or those kind of things, which you can really very nicely, you can replicate that thing in the mouse model. And then you can creating these model and you ask these fundamental questions using different models of autoimmune disease, cancer, viruses, how viruses actually modulate your immune system and so on. And then you come back to human. So you start the question from the human disease, and then you go around to pinpoint these things using fundamental, knowledge generation and come back to identify these nodes, what we through different routes. And then you can see, can we use this as a druggable target, or can we create a vaccine? How, immune response is behaving in this kind of particular setting. And ultimately, this is how we can put together things in our labs. I'll give you two major example, which we are working from last, 10 years when I came back to India. And one of the things I was fortunate to got associated with All India Institute of Medical Sciences, with the gastro department, with Vineet and Dr. Govind Makharia, Dr. Garg, and everybody. So the first thing which I picked, because this was easy, low hanging fruit for me, because my interest was understanding the T cell response. And one of the good thing was the inflammatory bowel disease is one of the disease which is primarily driven by T cell. So cholinergic T cells can cause intestinal inflammations. So this marriage kind of happened between the clinician and the basic scientist, because there is a resource. You have a lot of samples, you have biopsies, you can really go endoscopy, you sit there, you get the sample, and then you come back to lab and you dissect it out, what is there, what is at the cellular level and the molecular level, how these changes are happening in the patient, and that is ultimately leading to the disease progression. Can we really modify them? Can we understand them? So this is one thing. And second thing is, during COVID-19, that the collaboration with clinicians, like, with Dr. Sinjini Bhatnagar team, Dr. Garg, and everybody, we work together to understand not only the virus, how immunopathology is being induced by the virus, and that knowledge actually, taken us to the journey from deciding antiviral drug to the vaccines. So these are the two things. So for example, when we go to a clinic, we understand the disease. So you have two kind of major inflammatory bowel disease, divided in two categories: a Crohn's disease, a patchy disease of the small intestine. And then the second one is the ulcerative colitis, which is a continuous disease of the colon. So when we go into the clinic, we like to see, we see this, and clinicians see these lesions, right, in both the diseases. But what is happening within these lesions is the question what we really, as a basic scientist, like to understand, right? So that is important, and that's how we see how these lesions and what are the component in these lesions, what are the effector T cells, what are the immune cells are accumulated, and maybe they are actually feeding the inflammation and making this whole thing as a disease. So we understand in collaboration with AIMS team, and we identify some subset of this inflammatory or regulatory T cells in these disease setting, and these are the few articles what we actually published during. And I give you some good examples. When we started, we wanted to understand the new subset of T cells what we identified back in those days. And with Vineet's team, we really understand how these T cells actually evolved during inflammatory bowel disease in the patient of ulcerative colitis. And that's the work was done by Ritika Rampal. She was the PhD student of Dr. Ahuja, and she was really working in my lab first 2 years. So she was trained in my lab. She used to get samples, and we were working together, and that's how we come up with these few publications. And then subsequently, the other thing what we were really working on, this dietary potassium. So this is important, like, if you eat really, bananas, it's really giving good response, and you have really anti-inflammatory response was shown in the inflammatory bowel disease. And we published a couple of, clinical studies, one with Vineet Ahuja and one with the MGS team back in Boston. So this is something clinical thing gave me insight. What is the molecular mechanism by which this anti-inflammatory response is being evolved when we give this dietary potassium to the patient? And then we dig into it as a molecular mechanism. We identify if you really put the potassium or dietary potassium in some form, whether it's a form of banana or it's a form of coconut water, we can really induce anti-inflammatory T cell response. And that's how the disease is being prevented. And so I will not really go into the granularity of this molecular response, as we have already published. But then we continued this journey with the clinician. This is important. So sometimes we like to collaborate only for the sake of getting the grant. That's, I think, not the right collaboration. Or when sometimes you like to collaborate just to get samples from the clinician. That's also not the right, mindset. I think the collaboration has to continue longer time, irrespective whether you receive grant or you don't receive grant, or you get publication or you don't get publication. Somebody get last author, somebody get first author. Let's not get into this small thing. Let's have a bigger picture of the collaboration. So the collaboration, what we set it up in 2012 is continued. And then this is the coconut water trial, what AIMS has published very recently. This somehow got delayed because of the COVID-19. And what this trial actually suggests that if you have inflammatory bowel disease, and if you really get this coconut water it's a blinded trial, you get a kind of better anti-inflammatory immune response. So since we started this work long time ago, and this is the clinical outcome is published recently, so we decided why we can't use the resources of THSTI, understanding the molecular mechanism of this, and using clinical samples and clinical material from AIMS to have perfect disease mechanism. So this is what we started. I'll not really go into each of this data, but what I can mention that whatever the coconut water trial has been done in AIMS, the similar thing we have done in the preclinical settings, and we found exactly the same result in the controlled disease model, that giving coconut water is actually having the remission of this inflammation in mouse model. And really we went one step further, dissecting whether it's really potassium of the coconut water or maybe some other minerals, right? Some other, so then we remove the potassium from the coconut water, and then that coconut water is not really having any beneficial effect on the inflammation. But as an immunologist, if I go and say to you that coconut water is doing that, some of the immunologists will laugh at me and say, what is this? You are saying give it salt or give some lasan or something, some mirchi, and this is not science. So we really went into the molecular mechanism, and we identify the metabolites which is being changed by this dietary pattern what we have given to the mice. And one of the metabolites actually came out in these settings is the cholic acid. So then we replaced potassium with the cholic acid, and we found the same result, that cholic acid is able to suppress inflammation, inflammatory bowel disease. Now we identify a metabolites. Can we really design a drug around this metabolite, which is now being produced in our system? And that's what we did. Using the computational approach of Dr. Sthana, which is in THSTI. So this is kind of collaboration and a different expertise. So we went after the cholic acid structure we identified. We went to Asthana and said, can we identify the target? And we identify a target that how cholic acid is suppressing at the molecular level the interaction between these different amino acids in the ROR gamma T, which is a Th17 or effector T cells, which drives the disease, is suppressing this inflammation. Now, once we identify this mechanism, we wanted to have a new drug. So then we went to a medicinal chemist back in Punjab University. This is one of our collaborator, long-time collaborator. And luckily, he had a series of molecules sitting in his fridge. Almost maybe 100 molecules he was having in his refrigerator. And I called, I said, do you have some of these molecules? Is it possible we can make these molecules for, better, new druggable target? And he said, I already have these molecules. We got these molecules. We did the analysis in the animal models and in vitro, and we identify a couple of key molecules which can become a drug. That journey started from the clinical side, the potassium or coconut water, and now we have a new drug from that whole pathway. So the idea is that you start something and then you identify the key nodes, and these nodes need to be targeted to have better therapeutics. In case of, this is the example of inflammatory bowel disease. The second example is the respiratory viral illness, where we have multiple collaborations, and one of the collaboration what started right here in THSTI with Dr. Sinjani, Dr. Garg, and everybody came together because they were having the idea of this effectiveness of vaccine, and I was having the idea how T cell response during this time. So the idea that we merged together, and this happened because we were constantly talking every day, couple of hours. right? Sitting in the boardroom or over the phone in the night, can we do this? Can we do that? So this continuous, brainstorming happened. And that's how we come up with this idea during the Delta wave that, the virus actually evolved from Wuhan to Delta. And the vaccine which is generated against Wuhan strain is not sufficient to neutralize because the antibody is not able to bind and neutralize Delta virus. And then the vaccine is still effective because the severity of the disease was reduced in the people who were vaccinated. So what is the mechanism? Because antibody response is not sufficient. Then we went into the T cell response and we say that T cell response is preserved irrespective of the mutation what virus is acquiring. And that's why you are the severity of the disease is much, much lesser. And similarly, one more collaboration we did with the ECIC hospital where we got these samples of active COVID and we identify the immune response, right? Right from the innate immune response to T cell response, how the immune response is being shaped during the active COVID-19. And this is that paper we started when there was no vaccine was available. So our idea was to identify the correlate of protection in this case. I'll give you one more last example. This is again a collaboration what we did in All India Institute of Medical Sciences with Dr. Samir Bakshi. He was interested how this vaccine is going to work in the cancer patient, right? And so he has a cohort and I have the assays and the understanding of the T cell response and immune response. So then we put together this program and the vaccinated individual, the cancer, the children's, we got the samples and we evaluated their immune response. And we come up with the thing that, maybe the cancer patient or cancer individuals, the having lesser antibody, but still they will be protected because their T cell response is kind of preserved. So I think with this, I would like to just sum up and say that, the understanding of clinician real problem and having two people who are really expert in the subject, that the person who understand the disease and the person who understand the molecular or cellular aspect of it would be really good. But when we say this, I think it's not sufficient by saying this. I think this needs to be implemented by our funding agency, where there should be some grant where a physician and the basic scientists must be there to drive this program. And that's what with this, I would like to end my talk. Thank you very much.
Thank you, Amit. Just give us a few moments. Just give us a few moments while we put some chairs over here for the panel. Thank you for.
With you at the focal point of the concavation.
Huh? I just thought you could put a little more where we could see each other. So thank you, Dr. Makharia and Dr. Avasti, for your insightful presentations. And so we've kind of all, I think, understood the importance of integrating basic and clinical sciences in biomedical research and we've heard from one basic biologist on that. I'll just ask the panelists if they have any other views on this and would like to give their perspective with maybe some specific examples from their work where the integration of basic and clinical sciences led to a significant breakthrough.
Unfortunately or fortunately, we are all saying the same thing. So I just want to be the devil's advocate and raise a counter view to this. I don't know how many of you So I had written an op-ed in a newspaper and advocated that we should de-emphasize basic science and or research for the sake of research, but do it more in a programmatic fashion. So I got several very startled responses from some of my teachers. So one of them actually said, "What happens to research for the sake of research? This is what we've been doing." And I think many people have echoed that sentiment as well, especially when it comes to a conclave of basic scientists, largely basic fundamental scientists. There is a case to be made for fundamental science in itself also. So I just want to raise that issue so that there is a debate and around it. So just want to raise that.
Yeah, thank you.
Yes, Dr.
That actually gives a perfect platform where I wanted to go. Ramesh is perhaps not here, he stepped out. Ramesh made a very interesting statement that we don't understand 90% of disease anyway. I'm going to preface this by saying that any one of us who's a physician and trained, in our own house, in our own lives, 90% of stuff that bothers us, we go wrong, we can't put a label on it. The reality of this world is everything's a continuum. There are certain well-defined areas where we all in our field learn to put labels. It's equally true for basic scientists. It's equally true, in fact, within T-cell biology as well. And within those labels, we feel comfort. Now, the challenge of doing things by focusing very hard on the few labels that we know and understand is it becomes the equivalent of what is called the streetlight effect, where a person loses their keys to their car and is found searching under a light. And somebody comes and asks them, "Did you lose your keys here?" He says, "No." "Then why are you searching here?" "Because here there is light." So whenever you constrain to what is known towards further discovery, you discover more and more in narrower and narrower areas, which will not really change science or advance human health that much in the long run. So what are the solutions? You have to first understand where the lights are, where the lights are not, and you have to plant more lights. Now, where I slightly start disagreeing, so I disagree with everyone, so should be equal opportunity, is two people aren't enough. They say it takes a village to raise a child. I would say it takes a town to cure any disease. Two people, a clinician who understands their label and a basic scientist who understands their label, if they come together to solve something, it is assumed that the solution lies within those labels. I would actually argue it needs to be a broader pattern of questioning. And I think Govind had this wonderfully in his slide about open-mindedness on the question itself. And the second part, Govind, you had was confidence. That's where I would go to what Ramesh said was the method. The only thing you can agree upon, irrespective of whether you're a basic scientist or a clinician, is the method. Open-mindedness on what is the question itself. There's a very nice 2x2 graph that a friend of mine shows. There are known questions and unknown questions. There are known answers and unknown answers. Question is known, answer is known. It's really for the government to solve the problem for you. You aren't really required, maybe implementation research at best. If the question is known, the answer is unknown. That's one good part where a clinician gives input and a basic researcher tries to generate the answer, or a clinician researcher. There are technologies that are known, but where they can be applied is unknown. So for example, in the corporate space, you didn't even know that you had a food delivery service until Zomato came. This is industry-driven research. There is a solution. People haven't figured out the problem to which to apply it. Then comes the wicked area. Neither the problem is defined well, nor are the solutions. Asthma would be a great example. What is asthma? In 1950s, COPD included asthma and chronic bronchiectasis. Why? Because doctors had only one treatment called bronchodilators. If you only have one drug called a bronchodilator, well, asthma is the same, COPD is the same, a bronchiectasis is the same. You're going to give just that drug. New drugs come, and asthma splits off from COPD. But the labels remain in the clinical world. Then later on, mouse models come, all built on allergic asthma. Most of them go to human trial and start failing because human asthma is a big mixture. Actually, the guy with asthma doesn't care in the slightest whether it's allergy driving it or something else. He only cares that he wheezes. So asthma itself is a heterogeneous syndrome, not a disease. So this open-mindedness comes with entire communities of people from a variety of areas. And here I would make a plug for physicians who do science for a living to actually be able to bridge the two groups well. And that too is required. Very long answer to something very simple, but I promise to give the rest of the time to others.
Thank you.
I think Karthik wrote a very nice article, intriguing, that how little money you have in our country, how to use that more efficiently, kind of, and a very nice article. Again, the question comes that whatever we do has to have a purpose. And what's the purpose? Purpose is to train somebody, purpose is to train yourself, have more abilities. But what is that ability if I know for myself, and I can't make a difference to anyone, either to students or to the people I live with, or to my own country. So everything has to be a purpose, and purpose. If you start thinking of purpose, and let's do our work into a purpose. That whatever I do, what useful, how much useful, and I got this insight that, and this is how you choose your research question. That once you take a research question, if your hypothesis is right, you get an answer 3-5 years down the line. If the answer is there 5 years down the line, then so what? What do you do out of this? Even if you're right, you may be wrong many times, but if you're right, what do you do after that? So what's the next pathway? The next pathway guides you, is this work worth doing or not? If it's not, then why do it? Is this investing your own time or 25 years of your science life? This may not be that smart. And second point was, again, the question people raised on your note was that science doing for science. Again, they could be debated. People may have point of view, but purpose has to be there. There has to be some purpose of training, purpose of building institutions, purpose of, but bigger purpose that you're solving a problem. If you're working health sciences, then probably health science problem for your own country or for your own locality. That should be the ultimate purpose.
So just so I'm continuing to play the devil's advocate. So if you, so it was very difficult for me to say that something has to be top down. I mean, we are all, scientists are all liberals at heart. So when you say that something has to be imposed, somebody has to tell you, look, this is the way you, this is the question that you have to try and answer. It seems kind of illiberal, and that is not the way of science. So my only argument against that, I mean, when people raised objections to that, my only argument against that is that we don't have the luxury of looking at everything. I mean, it's a very utilitarian approach to science. It may not be very glamorous. It may not be the right thing to do in the long run. I mean, in the long run, if you don't produce enough good quality science, then I don't think you can really make progress. You'll have to rely on borrowed science then. But in the short run or in the medium term, we need to make progress to maybe acquire the luxury of being able to do science. So I think that's the only argument I can make for going this way. I mean, going a top-down approach or a focused approach rather than laissez-faire kind of science. So I mean, that's the thing that I can say.
Dr. Jain.
I largely agree on these points that you said. I think one should leave, it's up to anybody who's a basic scientist or a clinician who thinks what is the right questions, and as long as they're aligned, they should take a call, which is the direction they should go. Of course, the suggestions are very important because there are many times you probably don't know who would be your right partner along the way. So that would be my.
Dr. Avasthi, do you have?
I think both the things are important. I think the curiosity-driven science is also important because we are creating intellectual to the country. We are creating thinkers for different areas is important. But at the same time, when we are dealing the country like India, where we have larger health issues, then certainly, as Dr. Karthikeyan has alluded in his, this article, that there's a mission mode, funding should be in a mission mode where, multiple partners are actually tackling a problem from different angles. And obviously, it cannot be, I mean, the funding agency can have a funding program, but the funding agency's role is not to put people together. I mean, they can say, we have a program, you can apply, and then that's up to the investigator. They come together based on the expertise, based on the complementarity to address that problem. Thank you.
Yeah, yes.
I will come back in again. So I don't think we have the luxury of not having the luxury. I mean, it's a complicated statement. If I were to look at my very limited knowledge of how, say, Strand would start or algorithm got built, there would be no driving human disease purpose to it. Dr. Gokhale speaks about the middle income trap. The middle income trap is when you, as a low income country, keep doing research to trying to solve your problems. They don't actually generate any money, any great commerce. You remain poor, and because you remain poor, your problems never get solved anyway. The risk of tuberculosis is better correlated to socioeconomic status and to ventilation and the quality of your house than to any research that you do. If the country gets richer, tuberculosis will get better. How do you get richer is by creating value. You can only create value by finding things that don't exist and are known to other people. If you aren't doing something truly innovative and radical, you will not generate global value on the global chain. The place where we go wrong is, I think, we don't understand currency. What is the currency of your field is different for different people. Any situation where your currency and incentives are wrong, just publishing a paper will get you something. Then you do research for not for research sake. You do research for publication sake. It's not even curiosity research. It is publication-oriented research, which is an artifact of a promotion system that has been put into place. Genuine curiosity-driven research for the sake of genuine research, because you want to advance human knowledge, will lead to companies, will lead to outputs, will lead to IPs, and eventually, many things in medicine will get simply better because India becomes a richer country. Just my thought.
Thank you.
Yes. Just continue what Anurag said, that all those countries who have a limited resource of science funding, who decides that how much money goes where? And I think there's a lack of that understanding amongst our health science leaders, that if I have 100 crores, where do I put my 100 crores, which at end of 5 years, they accrue some kind of great science in terms of countries' science capabilities, may not be product, or a scientific product. The 100 crores, I put 50 crores to this outcome, which brings scientific mighty of a country. At the same time, some product, some useful thing comes out of it. Second, the 50 crores is for research things, like building institutions, because we have to build institutions also. Unless you build an institution, how do you get science? Therefore, these institutions may not, the outcome may not be a product. Fantastic if they can relate to the product, that's fantastic. But the goal will not be achieved every time. So we have to invest in our institutions and train our scientists or students that they train into fundamental science step by step, right technique, which don't happen to most universities, unfortunately. But the whole idea of a university that you train your students that they can do, when they grow up, they do more science and good science. So there has to be somebody who decides and distribute money as your father does at home, that you put this money to this objective and see that this objective is achieved, at least partly, and other objective is achieved partly. We live in cross-section. Something comes, I write a project, is funded because I write the project, it's funded. It has to be anyway, it has to be good. But what's the outcome? Who judges that outcome? How is the outcome going to go? The cross-section things probably, Karthik, I think this is also very important, that somebody has to own that how this money is used and what outcomes.
Okay. So just to what Anurag said about, I think that's the liberal aspect of science. So you want to let science build capital on its own. So I think we are all agreed on that. The only thinking is for when resources are really constrained, you have different strategies for short-term and long-term investment. So I think the mix of investment in translational versus basic would differ when your initial pot of money available is very small compared to, the whole of the West, Europe and America, they had colonial rich. I mean, Europe had colonial riches, right? They had the luxury of doing whatever they felt like. You just had people doing all sorts of research, and some of it really paid off and they really advanced. So that is a model which is very viable when you have a lot of money. But I think the mix should be, say, 60-40 or 70-30 in favor of translation in a situation like ours, because we've just not been able to make any headway either this way or that way. Because in order to make big advances in basic science, you need a very large cohort of people working on high quality basic science, which we still don't have. So I think what we can do, the lowest hanging fruit would be to invest a little more in translational science so that we achieve that capacity to do more work, maybe grow the economy as well, and then do other things.
Okay, so I've heard these words like more open-minded when one is doing research. So what is the that you all would like to give the early career researchers who are interested in pursuing research? Let's say the interdisciplinary part will come later in research in biomedical sciences, because if you start if you're doing your own research and not interacting with, so how early should that interaction with everyone, with the whole team start, in the sense, if you're a basic scientist with a clinician, and if you're a clinician doing research with a basic scientist, what would you advise the earlier career researchers? How soon should that interaction start for you to be open-minded in your thoughts about whatever research that you are doing?
I mean, if you're an early career researcher and you're now starting to ask yourself the question of when should I start engaging, you're already too late.
Okay.
Ideally, you should have been engaging even before you asked yourself the question. I would say connect with everybody who can help you frame the questions better and reduce your unknowns. Again, I'm borrowing a lot from what Ramesh said. But there are known unknowns, and then there are, of course, the unknown unknowns you can't do anything about. As far as you can interact with people who reduce the number of unknowns in your system, do that. Also, for every question that you ask, learn when to exit if you think it's going in the wrong direction to what you had hoped for, and be very clear on that, and then have multiple things going side by side. What I think, and I say this to everybody at least who joins my lab, is that start by imagining half of what you know is wrong, and you don't know which half. If you start with that perspective, you'll do better. In my own even clinical world, when I joined medical school, they would fail you if you gave beta blockers to a patient with heart failure. By the time I finished residency, they would fail you if you did not give beta blockers to a patient with heart failure. Everything changes. Keep your mind open, understand your outcomes, but also understand the limitations of the measures you use as outcomes. So continuously keep refining the questions, reduce the unknowns, and keep building on it, and work with all the type of people who can help you get to that level of depth of understanding what it is you're trying to do. My experience, anybody I have met who has built genuine depth of understanding of their area of work, almost always does well.
I want to add to Dr. Naga what said. I think the fundamental thing after you do that first thing for early career, that science is very addicting. It gives a lot of pleasure. Good publication, good PhD student. You are in that kind of kind of a pleasure mode. That you good science, you're very happy about it, whole team is happy about it, and keep going. And that becomes my lab. My lab does this, and that's a trap. And the trap sometimes may be punishable in terms of that to yourself, not to anybody else. That again, we are not thinking, I'm going forward, but I have not set a goal. So I'm going all my life there, 25, 30 years, and I'm not set a goal. So I'm getting publication, great PhD students, great honors, great awards, but this goal has not been achieved. Setting a goal is, I think, it's very important for each one of us, at whatever age we are. What is my goal? What am I going to do? So my goal is I'm going to 10 great PhD students. That is my success. I'm fine with it. I don't care what anybody says. My goal is to create 10 great PhD students who do great science. My goal is to get 10 great papers who will change science around me and around my country or around the world. Or my objective is to create one product, diagnostic therapeutics, or understanding disease biology, so solving a problem. So setting a goal probably for each one of us is very important. And I know that this how it has helped me. This I cite my own example. Setting a goal about celiac disease is a pleasure. And this becomes and you keep changing. So setting goal probably to mind is very, very important for anybody.
I would actually, I agree with Anurag when he said that you have to start off with that uncertainty, and that uncertainty has to start very, very early. I think I don't know, I can't talk about the basic science education system, but in the clinical education system, we are imbibed from the beginning with some sort of pseudo certainty. I mean, we just think that, okay, this is like this. And like Anurag said, you would get failed for saying something with some degree of conviction, or if you didn't say something with some degree of conviction, that would be counted against you. But I think that conviction is the problem. I think if we start off at a very early stage in the medical training curriculum itself, that there is so much of uncertainty, that would really help. But also from a larger perspective, I just wonder, because there are so many medical schools now coming up in science campuses or IIT campus. So I don't know if that would really help develop that mindset at some point in time.
So thank you. Yeah.
Yeah, I think I agree with both of you. But with respect to the people who comes to the basic science lab, I think the perspectives are changing right now. I mean, if I think about ourselves, when we actually landed to a lab to do a PhD program, the way we used to think and the way today the students are thinking are totally different. And in fact, what I see happening nowadays, even though you don't force them to go to what's so-called as immersion program, they tend to actually, because of the, those days, you didn't have the internet, you didn't have access to, the whole world, what's happening around. And I think they start, I mean, I see my son that he started thinking, I would like to be something, I started thinking, I think that's the stage that they should start thinking about. When you say early, how early? That thinking is still there, but it certainly needs some sort of guidance to shape it up. I think that is something that I would actually advocate. At what stage one should actually give the proper guidance. I mean, let's take an example of what happens in typical labs. We get even graduate students come for 3 months, incubation thing, and what you say that we just ask them necessarily not to work on something which is already defined project. I think by the time they leave, they understand that if I actually end up doing a PhD in a different lab, what kind of mindset I should be thinking on that line the moment I go into that lab.
One interesting experiment that's happening right now, and Karthik, you alluded to it, is engineering schools opening medical schools, and how that's going to go is going to be interesting. If there is any one more field with even greater conviction of what is right and wrong, it's probably engineering than medicine. So I think when engineers get into medicine with a sense of the human body as a machine, which doesn't always work that well, it's going to be interesting to see how they see it. I would actually argue that probably the best model will be when all people, including liberal science, come together to work on problems without worrying about campuses on which to open hospitals. Because really what people are doing is everybody feels we don't get samples. Let's build a captive hospital on our grounds. Then we will use the samples from those hospitals to do some great research because we already have the scientists. I am not convinced at all about that model. I think there have been plenty of hospitals, there have been plenty of scientists. What we need, and along the lines of what we are saying, is a greater acknowledgement of what Ramesh started with. There is so much unknown, and part of it is unknown because of the way we approach things. But we can all agree on great scientific method. We follow the rigor, we will all do great.
So what you're suggesting is that there has to be some kind of a, so do you suggest that there should be some kind of a policy change at the institutional level and at a more, say a government level or somewhere there, for, to promote more integrated biomedical research?
I'm going to give Govind's example here.
Yeah.
I'm going to maybe embarrass him a bit. So when I came back from America to India, I was used to something like 3-4 months clinical, you pay off your clinical salary. Remaining time, your protected time. Somewhere along that time, I met Govind. Every Sunday, he would be in his place, working dedicatedly, doing fundamental, groundbreaking work on celiac disease. And I honestly asked myself the question, could I do this? I mean, a full-time clinical load. I mean, I don't think he sees a single less patient than any other clinician who does not do research, then spends the entire other time, free time available, pursuing research because of his passion. But if only super passionate people who are willing to work 24/7 were going to do clinical research, you would also lose a variety of other people who are curiosity driven and would love to explore things, are bright, but don't have the ecosystem in which to use their brightness. But at the end of the day, they want to go back home and also have a other life. I think we need a better ecosystem. AIMS, maybe Govind himself can maybe talk about how AIMS has tried to address these problems and why they don't work. Why some people with demonstrated capacity for doing research can't be given more protected time to do it? And what would it take from funding organizations to make it possible at a place like AIMS? I know it's not about money alone. I've been through this discussion multiple times, but I think Govind and Karthik both are the right people to perhaps talk about this, because there's somebody needs to talk about this.
Certainly. I mean, I think mindsets have to change in terms of protected time for research that you're talking about. All those things have to change. But I think there is also this, see, you rightly pointed out that why people are setting up medical schools and engineering campuses. The question, the reason for that is not the right reason, right? So I think there's a more fundamental issue to be addressed here. We really need to this is it's a short circuiting a problem. People really, if you talk to engineers, that's, I don't know if there are any engineers here. It's not a general statement, but some of them, at least, they think of, so I know I can create an algorithm for, say, detecting diabetes complications. They have no clue about any of these things that they are saying, but they know that they can create an algorithm. Now, that's not a solution that you're looking for. So it has to be much deeper than that. The engagement should be deeper from both ends, and it has to develop from very early on, I think. And I think it has to change at the medical school level. So we are all already constrained in our own, I decided that I would do cardiology when I was in second year. So that's really not the way to go. I mean, we need to really be more open-minded about things. And
I think you rightly said, Dr. Nayak, that it's very important to have a time to protect it, to do certain kind of mindful work. What happens that there's no time, if there's no protected time, you get tired over time. Over years, you get tired. Your ideas become slightly more slow. You're not able to see things all together in a manner of steps, how to go, and then you probably start losing interest with time. And still you proportion become bigger, but again, you don't have great ideas. And that's a problem. That's a problem. Because as you become bigger, you become more audible. People listen to you, and they listen to you, the ideas which are still back behind ears. And that's the problem. I think is a continuous thought process to go. So all the science leader should have that kind of a mindset. And we are getting trapped. We are getting trapped. The every age and working continuously, you get tired. Your ideas get tired, and that might affect many others. It not only yours is fine, but might affect others.
I'm just going to put Dr. Srinath Reddy on the spot. Sorry, sir. But on this question of how do we change this whole thing of how do we make research happen for the sake of progress in medicine? How do we do this at a policy level? What do you think?
You did put me in the spot because I've not been really party to the conversation earlier, to the stream of thought that is developed over there. So I've just walked in here, and therefore I was just catching up on the comments that Anurag and others were making. So the question that you're posing is, how do you ensure that research is actually becoming a very live and vibrant stream during medical education? Is that what you're asking me? Of course, I believe it should be entirely possible to do that right from the undergraduate level in order to really pose it as problems to be looked at in terms of identifying the causes and the solutions. And if we can actually stimulate them into that mode, rather than actually provide it entirely as a very prescriptive mode of education, then it becomes possible to do that. And certainly at the postgraduate level, it should be even more possible to do that. I think, when I was appearing for my initial faculty appointment at the All India Institute of Medical Sciences, I was interviewed after my DM by Professor K.G. Nair. And one of the things he quizzed me was, of course, on what my research interests were. But he also said, how will you teach mitral stenosis to undergraduates, postgraduates, and DM students? Facing a faculty appointment interview and getting a question like that is certainly not the easiest thing in the world to respond to. So I did not know what to answer, but I came up with this answer. May not be right, may be wrong, but I said to undergraduates as facts, to postgraduates as concepts, and to postdoctoral, to doctoral students as controversies, DM students as controversies. Reflecting, I'd got the job, but reflecting on it, I think it was not a very right answer. Because I don't think even to undergraduates, it should be dinned down as facts. It should be taught as questions to be explored. And certainly you can talk about what are recorded as the facts at that time, but certainly raise questions even there, because I don't think it should ever become prescriptive at the undergraduate level, because then that trend will continue later on, even in further on. So I think the ability to raise questions and then say that, okay, at this current state of science, this is the best possible answer we have. This may not remain so over the years. We may actually start revising our opinions or even refining our opinions based on research, and therefore we need to continue to do research in order to explore this avenue. And then, of course, how to apply the knowledge much more fruitfully. That again requires a further bent of mind. So I think that entire process has to begin at the undergraduate level, and getting them to actually explore not as individuals, but as groups is going to be very important. After all science is going to be team science, now and in the future. So they have to learn to work as teams, cross-fertilize each other's ideas, and then start exploring that And I think that peer-to-peer learning is also going to be very important in the future. I don't think we should leave any medical student, undergraduate or postgraduate, as a solo warrior to explore.
Thank you, sir.
Thank you. So, I mean, in the interest of time, I don't know, maybe there's just one last question, and I don't know whether we have time for the for questions from the audience. No, okay. So how do you envision the future of biomedical research with greater integration of these two fields? Is the last question.
Let's call it health research. And the way I see the future, in fact, very interestingly, on the way here, I was writing something for India Today, is that health is a very simple word that is really complex at the back end. Its definition changes from the time you're born to your die, from past to the present, from strata to strata. And it's as much social sciences as it is natural sciences. And that's, I think, what Dr. Reddy said so much better, that it's going to be a team effort. And we will need to take health sciences out of medical colleges into other places as well. And we are going to need to bring other sciences into medical colleges as well. One day AIMS will have to have a department for medical AI. Within the department of medical AI, they would have to start thinking about ethics, justice, social sciences, anthropology, and computer sciences, of course. That's just incidental to everything. Similarly, if engineering colleges want to get into health research, having a hospital will not be enough. And in the end, everybody will realize we are very good at what we do. Let's find other partners who are good at what they do. And let's not bother so much about ownership of areas and simply work together. So I think this concept of larger health research, in many ways, I'm saying only things I've already seen people like Dr. Reddy do. So there's nothing new that I'm adding, but I thought I would say it anyway.
Thank you. Anyone else on?
I fully agree with you. I think one of the things that I wanted to say earlier, that I have been part of a very first HIV vaccine trial in India back in early 2000, in Pune, and I had the opportunity to see how, this is an aspect I wanted to bring, the importance of having a social scientist and natural scientist in this part of the journey as a team. And many a times I have seen, people with a socio-behavioral background had acquired the skill set of speaking science that we do. That's not normal. You don't expect a social scientist speaking science, laboratory science. And that only happens because you create an ecosystem where they understand what the science going around in delivering a public intervention solution. So I believe that, but the other question that I had, anyway, in the interest of time, I didn't have the opportunity to say, when you speak about clinicians, basic scientists, integrations, the kind of thing generally we do is the same, we want to ask the same questions in the laboratory, whether it's yours or it's mine. I may have some kind of equipment or infrastructure. I may not have, therefore I am complementing myself. How about clinicians who don't work in the lab? And how do we exactly integrate there? Because we do a lot of work in this field where we work with clinicians who actually don't have a lab, but they do understand the science and how best we can actually integrate, understand the language and deliver the solution together. So that's something that I want to.
I can completely agree with both of you on that. Only that, if we have to create a triple problem kind of situation, the problem, and this problem has taken by 10 person with the 10 different abilities. Only then we solve a problem. Our infrastructure is now the way we created around the world, the hospitals, then there are science institutions, fundamental science, AI labs, they're all in segregation. Either there's a way that in our own existing system, we create an ecosystem that we have crosstalk. And that's the best thing to do, because in the event infrastructure, people can crosstalk. And that's we need to have a kind of a environment creation, talking to people that I have this question, so this question can go here, this is solved here, then go there, kind of, or continuous, problem-oriented. Second one is if you want to create a university now, or I want to create a department of gastroenterology. So what I will see that I create a department of gastroenterology or hospital gastroenterology, which are clinical scientists, clinicians, so we treat patients. We have a epidemiologist, who do research on epidemiology. We have fundamental science labs, but in every specialty on fundamental science, and they work together. The whole work is like a chain and work together. That's a dream, but hope that dream will come true someday, somewhere in the world, that we have all the beats together required or work together, either in the beat form in the system or a new system.
Thank you, Dr. Makaria. That was, we hope that that happens very soon. On that note, thank you to everyone for a very enlightening discussion. We'd just like to felicitate you with a small token of, and I'll request Dr. Karthikeyan to please do the honors. Director? Okay, you're good.
I request our Executive Director to felicitate Professor Anurag Agarwal. And I request our Executive Director to felicitate Professor Govind Makharia. Now, I request Professor Amit Avasti to felicitate Professor Karthikeyan. Now, I request Professor Amit Avasti to felicitate Professor Jayanta Bhattacharya. Now, I request Dr. Nithya Vadva to felicitate Professor Amit Avasti. Thank you, ma'am. Now we shall break for the lunch and assemble back in the auditorium by 2 p.m. for a brainstorming session on quiz competition. Lunch is arranged outside in the auditorium complex. Thank you.
रख दो कैमरा हेलो खाना खाओ खाना है
Preeti Singh. Welcome back to the post-lunch session. Hope you enjoyed the lunch. May I request all of you to turn off your cellular devices, and for those joining online, to please put yourself on mute as we will be starting the quiz competition momentarily.
Hello.
Not coming. Okay.
One team is not coming. She just announced.
Huh. Quiz competitions are an effective tool for enhancing one's critical thinking skills. By participating in a quiz competition, students can develop their analytical, creative, problem solving, communication, and confidence building skills. Let's move forward for an exciting session of questioning or knowledge. Now, I request Devuti and Jayanti to kindly conduct the quiz competition. Over to you.
Good afternoon, everyone. Welcome to the quiz competition on the occasion of 15th Foundation Day of THSTI. I am Devuti Palan, an SIB fellow here.
And I'm Dr. Jayanti, an SIB fellow as well. We are your quiz moderators for this competition, and this is the basic overview of the quiz. There will be 3 rounds. Round 1 being MCQ from current affairs, round 2 MCQ from science, and round 3 an audio-visual round. Each team identifies a person who gives the answer. Only the first answer will be considered. There will be 2 questions per team as round.
These are the teams who have qualified the prelims. Rules for the round 1. Each correct and complete answer will receive 10 points without options or 5 points with options. There will be negative marking of 5 points with the wrong answer. Each team will get 15 seconds to answer each question. Unanswered questions will not be forwarded to other teams. The team with the lowest score will be eliminated at the end of the round. And in case of tie, all teams will move to the next round.
Now, instructions for the special audience. So there will be 2 questions for the audience, especially after the end of each round. Use of mobile phones to answer the questions will not be accepted. Kindly avoid verbal prompting of answers during the quiz. And one audience member gets to answer just once, unless there is no one raising hands.
Let's begin with our round 1. Team A ready? Do you have your mic?
Yes.
The question will be displayed on the screen. And if you want to see the options, you have to respond quickly. Yeah, MCQ based questions. Are you clear? If you have any doubts, you can ask us right now.
But if you answer without options, you get 10 points.
And we request audience to please do not prompt in between. You wait for your questions. We have some interesting questions for you as well. Team A ready?
Terrico and
Time up.
Trees.
Wrong answer.
Medicine.
The correct answer is Svante Pabu. You could have asked for the options if you feel like.
Next question will be for Team B. Team B, do you have your mic?
Yes.
Yeah. Ready, right?
Yeah. Options. A. Option A.
No, the correct answer is Harmanpreet Kaur in 2023.
Team C, next question is for Team C. Do you have your mic?
Yes, yes.
Ready?
Yes.
Options, please. A. Tamil Nadu.
No, the correct answer is Kerala.
Team C, do you have your mic? The next question will be for Team C. Ready?
D.
Okay.
It's Team D.
That name was given to you in your prelims. Right now, it's A, B, C, D, E.
So we are D.
Yeah.
Okay.
Ready?
Yeah. Options, please.
Time up.
C. Option C. The answer is correct, but since
Time up. Yeah. Next question for the next team. You guys ready?
Yes. Options. D. Andes.
Correct answer. So we come back to Team A. Ready?
Yes. Options.
No. Time up.
The correct answer is 1986.
Next team ready for the question?
Yeah. Options.
Time up.
So the correct answer is Dinesh K. Tripathi.
Next team ready?
Yes. Options, please. Pass.
Time up.
So the correct answer is Thailand.
Next team ready?
Yes. Options, please.
Time up.
Okay, the answer is the state of Tamil Nadu.
Next team ready?
Yes. Options. Ministry of Home Affairs.
Wrong answer.
The correct answer is Ministry of Education.
We will announce the end of round 1 and results in 5 minutes. In the meantime, we will have questions for audience.
आप ऐसे आगे मत करो
हेलो,
थोड़ा-
हेलो।
हाँ हाँ कोई बात अच्छा हमें जंप ही करना है वो एक टीम नहीं आई ना
Audience. So the first question for open to the audience is Sentinel nuclear missile was developed by which country? Okay. Mic to the faculty member here.
Russia. Wow.
I said Israel.
Sorry to inform you that's not the right answer.
Wrong answer. Russia.
Wrong answer. We have the right answer there, but you have used your devices, it seems. Kindly avoid using your cellular devices to find out the answers. Correct answer. So I know it seems you are sneaky, but everything is visible from here. So we can see your devices if you're using any. We have one more question for the audience. USA. NASA recently captured an epic view of TASH using the Mars Odyssey orbiter. Any space enthusiast here? Anyone? It was quite talked about in the news also. This particular name of the entity. No, the answer is not the Mars.
It's Hugo volcano. Volcano.
No.
Here. Volcano.
The correct answer is Olympus Mons.
Okay, you have to go back to the question. इनको half marks दे देते हैं. We'll give them five marks. We'll give you 5 marks. We'll get 10 marks. 10 marks.
So there was some dispute regarding the scoring. So the scores as of now stands as Team A with 10 points, Team B with minus 5 points, Team C with minus 5 points, Team D at love 0, Team E is also at 0.
Minus 10 and plus 10.
I would request you follow the scoring given it is minding, it's not up to dispute. Please follow the sanctity of the game. Should we do?
We'll move on to the next round.
Moving to the second round, there will be 2 questions per team in this round. Each correct and complete answer will receive 10 points. And without options, or 5 points with options. There will be negative marking of 5 points with wrong answer. Each team will get 15 seconds to answer each question, and unanswered question will not be forwarded. 2 teams with the lowest score will be eliminated at the round 1. And then in case of a tie, team with more correct answers in round 2 will be moved forward because this will be science-based questions. All teams ready for round 2?
Yes.
No. But the timer starts with the question displayed on the screen. The question will be displayed on the screen and the timer starts. If you wish to see the options, you have to prompt quickly. Ready for your first question? Okay. Time up. The correct answer is option A. Next team, ready? Ready for your question?
Yeah. Options. D, medulla oblongata.
Correct answer. Next team, ready with the further question?
Yes. Options, please.
No, the correct answer is option A, Carl Barry Sharpless and Frederick Sanger. Next team, ready for the question? Yes. Options. 33. Option B. Correct answer, but audience, we request, please do not prompt in between. You can clap, They gave the right answer. Next team, ready?
Yes. Options. Dr. Christian Barnard.
Wrong answer. The correct answer is Dr. Jonas Salk. The audience is quite on their toes. We come back to the team A. You guys ready?
Options. C.
Correct answer. Next team ready?
Options.
Yes, absolutely right. The answer is Prokaryote. Next team ready?
Yes. Options.
Time up. The correct answer is option A, mutation of the HGPRT gene. Next team ready? Yes. Options. Time up. The correct answer is IIT Kanpur. Next team ready?
Yes.
Audience, audience, please do not prompt in between.
A, sheep.
Wrong answer. The correct answer is C, buffalo calf. We went here. We'll have 2 questions for the audience now. In the meantime, we'll calculate the scores and we'll give you the results for this round.
No, the cursor is here. You need to drag here.
Click here. Question for the audience. So the first transgenic plant to be produced is BT cotton. You're going for the low-hanging fruit. Think. Tobacco. Cellular device wins again. Tobacco. Correct answer. Tobacco is correct answer. Ready for your next question, audience? The youngest scientist who was awarded the Nobel Prize in Physics was? Oh. Correct answer. We will wait for 2 minutes and then we'll declare the results, announce the results. It looks like the scoring is quite neck to neck. It's taking quite some time for us to calculate. So after the results, the two teams will have to leave us at this round. So those two teams are Team C and Team E. But a round of applause, please, for their participation. So the three teams advancing to the round 3 of the competition are Team A, Team B, and Team D. We'll have round 3. Okay. So the scores for, okay, you want to know the scores? Team A is 15, Team B is plus 5, and Team D is 5. So the leading scorer is Team A. Team A. Now we'll have an audio-visual round. We request the audience to wait for their question. So for this round, we'll have again 2 questions per team, and the rules are as we have discussed earlier. All teams ready for this round? We'll start with Team A. Audience, please. Audience. No, you don't get options in audiovisual round. And audience, we appreciate your support. Please do not answer if you know.
Rocketry.
Correct answer. Yeah, the correct answer. Is rocketry the Nambi effect on Dr. Nambi Narayan? Next team ready for your question? So this will be an audio which will be played to you. You have to recognize this.
Yeah.
Give me your mic.
I know, I can hear it. I heard it.
So do you have the answer?
Brooklyn Nine-Nine.
Yes, correct answer. Next team ready for a question? Yes. Options. You do not get options in audio visual round. All of you have seen this movie. Recount your days of silence. But to answer the year is so difficult, right? So, audience? I heard 1994 there.
94.
Volunteers who raised the hand first, volunteers?
1994.
No, we know the right answer is 1994. We heard it from you, but who raised it first? So, it will be from this block. You'll have your chances. There are more questions for you as well. But round of applause. Team A ready for your question?
Yes.
We'll skip one slide, okay? Your question is after that slide. This is your question.
Jwala Goswami.
You're nearly there.
Jwala Datta.
Wrong answer. Only one answer is considered. Disqualified. Yeah, only one answer is considered and wrong answer. Audience? Yeah. It's nice to see you recognizing female cricketers as well. Round of applause for audience. Next question for the next team.
The first. What is that?
Next question for the next team. Time's up. So if any of you have read the newspaper today morning, you'd know the answer. Spain, the Euro Cup winner. Yeah, next. Next question for the next team. Yes.
Which team who won the first F1 title?
Time up. So let's throw it up for the audience. Any answers? You please first raise your hand and then answer. It will be easier. Yeah. No, the team with which he won the first title. Yeah. I'll give you a hint. It's a British team. Notably orange.
Ferrari.
Mercedes.
Audience, we want to hear it from the audience.
Mercedes.
Wrong answer. Lando Norris tries for this team.
Ferrari.
One. The participants here have answered it. McLaren. Yeah. So the scores will be calculated and we'll announce the results soon. In the meantime, now we have actual question for the audience. Yes. Now is your time to shine, audience. You've been prompting too much.
Athens, 1986.
Correct. We have one more question. Like we have two more questions. So audience, try different options and see, let's see who wins. So the audio clip, please bear with us. Chandni Chowk to China. Okay. It's not Bahubali.
चांदनी चौक तो चाइना है क्या?
No. So a hint, the name of the movie, the word is alliterated thrice. Correct. Correct answer. This movie represented us at Oscars. You should all know. So we have our results. Are you guys excited to hear who's the winner?
No.
Okay, I think the answer is quite evident in guesses. So yes, we have a winner, Team A, for the quiz competition. But commiserations to the Team B and C. The prizes and will be taken care by the committee, and that will be released later on. Yeah.
Thank you, Devuti and Jayanti, for an enthralling session of quiz competition. Now we move on to the next session of the day.
To deliver the Foundation Day lecture, we have with us our guest of honor, Professor K. Srinath Reddy. Professor Reddy is a cardiologist and epidemiologist by training. He is the founder president of the Public Health Foundation of India and is currently serving as the honorary distinguished professor of PHFI. Under his leadership, PHFI established 5 Indian institutes of public health to advance multidisciplinary education and research for multisectoral application. He is the recipient of Padma Bhushan, the prestigious civilian award conferred by the President of India. Several other honors received by him are the WHO Director General's Award, the Luther Terry Medal of American Cancer Association for his outstanding contributions to global tobacco control, and the Queen Elizabeth Medal for health promotion. He's also the first Indian to be elected as an international member of the US National Academy of Medicine. Dr. Reddy has more than 500 publications in peer-reviewed Indian and international journals. With such a glorious career, Dr. Reddy is a true inspiration for young aspirants like us. Sir, we have the proud privilege of welcoming you to deliver the 15th BRIC THSTI Foundation Day lecture.
Good afternoon. It's indeed a privilege to be here as a part of this gathering, to be celebrating the Foundation Day with you. I'm grateful to Professor Karthik for the gracious invitation extended. I have known of THSTI for several years, having served initially in the Scientific Advisory Committee, but for several years have not had the privilege of visiting this institution. So Professor Karthik has provided me an opportunity again to renew my connection, and I'm grateful for that. Indeed, after that very exciting round of quiz, a lecture may be not the best thing in the post-lunch timing, but nevertheless, I will redeem myself by assuring you that I will not pulverize you with a PowerPoint. I will therefore try and speak without necessarily referring to any slides, but try and address the general theme of translational research from the context of how we should advance that in India for the benefit of not only India, but of the global community, and where THSTI definitely ought to lead the way. When Professor Karthik asked me, or rather I asked him what I should speak on, he left the choice to me, but then obviously it had to be tied in with the theme of the day and of the institution's mandate. So I said I'll speak on science to society, the translational pathway. Firstly, of course, why translational research? It's very clear that when we're really talking about science, we must recognize, and it's my firm belief that it is so, that science is sterile if it lacks a social purpose. And at the same time, public policy will crumble on clay feet if it does not stand on the basis of strong science. So it's absolutely important that we do connect science to public good through public policy. And therefore, when we are really going through the process of science from discovery to development to delivery, then we must recognize that science transitions into technology, but ultimately will have to be implemented through effective systems, in this case, health systems. And therefore, I believe that science discovers, technology develops, and health systems deliver. And we need to maintain a very integral connection between those in case we have to ultimately promote societal good. But we also recognize that there are different elements here, and all of them need to function in tandem and wherever possible in close concert. We recognize the importance of exploratory research, wherein we try and identify various elements which may be of potential use, but at least contribute to our understanding of some basic elements of human physiology and functioning of various body systems. Then we undertake, that is essentially the basic science research, which leads us to that kind of an exploratory work. Then we move to elucidatory research to try and develop some hypotheses and test them out. And that's essentially clinical research. And finally, we have to move into implementation research, which is again, essentially, in this case, both clinical and health systems research. So we must recognize that ultimately, public health will have to come into the field of translational research, and that's where I come from, both as a clinician and as an exponent of public health. I also believe that public policy will crumble on clay feet if it does not stand on the basis of strong science, and therefore we must connect to public policy wherever we can. Therefore, we'll have to ensure that as we move from purpose to process and to products, we must ensure that this seamless continuity is maintained throughout our effort. Now, in terms of how we actually ensure that there is a robust research methodology that energizes each of these elements, that again is absolutely critical. Because when we really talk about purpose being not only scientific curiosity, but a social purpose, and the process being essentially very sound research methodology, we must definitely ensure that the research methodology addresses the right questions with the right way of investigation. And that is where we know that we have to contend in our quest for truth with chance, which is the eternal alternate hypothesis, with bias, which is systematic error, and confounding, which again is a form of bias. But at the same time, we also recognize that there is a considerable degree of variance, and we have to accommodate that variance. And now in research, we also recognize there is an element of fraud, which we have seen increasingly in recent years, and we have to keep our eyes open and be alert to that. And to try and tease out our best approximation of what might be truth at that point in time through this process of rigorous research. Then we also recognize that it has to be translated into products, which can find useful application. But we also know that we should not mistake science for technology, because essentially science yields an idea, whereas technology yields a product. Science creates knowledge, whereas technology creates tools. And therefore, it's absolutely important, while we pursue the process of translation, we do not mistake one for the other. We do need to promote robust science. There is no alternative to it. At the same time, we must ensure that the products of science, when translated into technologies, must also find useful application. So then comes the question, when we create the scientific knowledge or craft those technologies, the question comes in is essentially how best to use them and how to maximize their benefit. And there comes the whole area of applied research or translational research. And therefore, translational research is the ultimate pathway by which robust science finds social applications. And therefore, this particular institute definitely stands very high in the list of priorities when we look at how best to move science to social benefit. But there are also, as you know, multiple translational barriers. I do not have to really discuss them. But translating scientific knowledge into products is the first translational barrier. The second translational barrier is in moving those products into useful application. And that we have seen many a time is a more difficult barrier to cross. We have seen so many products which have actually not been applied to the benefit of those intended. And there has been technology pileup, whether it is medicated mosquito bed nets or vaccines, not reaching the intended beneficiaries. And therefore, those who have reveled in the creation of technologies and felt that they could solve all the problems have also realized that you need strong health systems and strong public health mechanisms, pathways, in order to ensure that the products are usefully delivered. And therefore, we will have to really integrate all of those elements as we plan it. So what we need are essentially to define the pathways where scientific knowledge can be transformed into transnational tools, transformational tools, and ensure that they can be efficiently used for the benefit of those intended patients or populations. But what we may have to ask ourselves again is, when knowledge is being created worldwide and in so many places, so many institutions across laboratories, hospitals, and universities across the world, why do we need to invest in translational knowledge? Is not knowledge automatically portable? Is not something that is discovered in the West something that we can apply readily? Should we really replicate and duplicate research? These kind of questions are often asked. But I think there is an absolute importance analytics. It is the prior probabilities that also determine what the posterior probabilities are. And for this, it is important for us to even look at simple examples. Professor Karthik, of course, is a wonderful cardiologist, but he would very well know that the significance of a positive stress test in a 60-year-old male diabetic who is a smoker is very different from a similar positive test operational steerability, and political viability, which means that we need interdisciplinary or multidisciplinary research. Scientific credibility comes from biomedical research, clinical research, and epidemiological research. Financial feasibility comes in from health economics and research. Operational steerability comes from health systems and implementation research. And political viability comes from social science research. Now, do we have institutions which can actually bring in all of these streams of knowledge and combine those streams of research? Unfortunately, we do not in most places. Our university should be the knowledge commons where this should be happening, but it does not readily happen even there. We have very much functioned in silos. Now we need to move from silos to synergies. And that is where we must bring in multiple streams of knowledge and research capacity in order to generate this confluence of knowledge which can enable policy and programs. So we should not blame the policymaker if the policymaker does not avidly take up the research we believe we have delivered and is going to be very useful. Every researcher believes that his or her research is going to transform the world. But we must also recognize that policy has its own imperatives and compulsions. And if we have to enable the policymaker to not only understand and appreciate the research, but also apply it for public good, then we have to tell what is the magnitude of the problem? Why is it important? How many people are likely to be suffering? Are there equity considerations? Are there vulnerable populations that need to be attended to? What is the nature of intervention that's required? What is the evidence that the intervention is effective? And what does it cost? And what will it deliver in terms of benefits? And in what time frame? And are the systems ready to implement it? Who are the people likely to support it? Who are the people likely to oppose it? These are all the legitimate questions that a policymaker will ask. So when you're moving from the laboratory into the clinical arena or into the community public health practice, you still have to answer these questions or anticipate these questions and then hope that your research cumulatively will deliver these answers to the minister who is going to ask these questions. So translation will require all of that to be done. And that's why multidisciplinary research is absolutely important. And we do need this. So, as I said, we are also dealing with multiple complex adaptive systems. We have, and we've all been trained in research to follow simple linear models. In fact, most of our research looks at A leads, must find a solution to that linear progression from A to B. But we now recognize research and in particular translational research. But nevertheless, let's make the case. It's very clear that we cannot have research functioning in silos, and we cannot have bits and pieces research if we really want research to yield transformational products. Therefore, we do need a much greater input into our research, an input and investment into our research enterprise. And for this, partnerships are critical. We need partnerships which are interdisciplinary, which are interinstitutional, and in fact, going beyond academic and research institutes into other wide variety of societal partners, not just the academia, but the industry, health professionals, NGOs and community-based organizations, health system managers. All of them have to become participants in this enterprise, either to provide the inputs or to absorb the outputs. And unless we do that, we are not going to see the transnational research pathway deliver the kind of results that we hope it will. So we need these kind of partnerships to catalyze those coalitions, to channel their common efforts, to create transformational products, and even to consume them in a manner that yields the best benefit. So we therefore need to bring in all of these elements together, and not to forget the media and the public as well, because it is now becoming importantly clear that communication is a very important element of even translation, of ensuring that we communicate with the public or the potential consumers what the product is about and what the benefits are likely to be, and so that there are no misgivings, there is no misinformation, and there is no resistance to their application and absorption. We have seen this with vaccines. We have seen how the lack of vaccine confidence can be a tremendous barrier to the uptake of useful vaccines. You cannot take the public for granted. So you have to communicate with the public, and you have to make the media a useful partner. So this again becomes a very important element as well. Therefore, acceptance and adoption of innovative and impactful technologies requires all of these partnerships. Can we do it? I believe, yes, we can. Certainly, we have shown our capacity to do that in many areas. Why not in translational research for health? And it's not as though we have not had innovators and explorers. I know Dr. Brahmachari is most often talked about, but what about Rustam Jal Vakil? Professor Karthik is here. Here was somebody who brought in Revolfia from Revolfia serpentina, Recepine, as the very first antihypertensive drug in the world. There was no drug for treatment of blood pressure before Recepine, and that was the contribution from India. There is no reason why we cannot do it again. We have, of course, the opportunity to explore all the wealth of Ayurveda and ensure that the right kind of scientific investigative technologies are applied so that we can get the best benefits out of that. Then we also recognize now in the world of medicine, we are also looking at repurposing of drugs. There are so many drugs developed for a particular purpose, but which can be applied for other purposes. We should investigate so many other pathways. Nobody should be able to tell us today that metformin is used only for diabetic, because there are so many other physiological pathways that are influenced by diabetes, including aging. So why are we not capable of investigating many of these pathways and ensuring that we can also set the pace for global transformational research? So I believe that we do require multiple stakeholders to be engaged in the process. But we also need political commitment. And we need both for political decisions and for political direction to ensure that science can be applied for social benefit through translational research. Patricia Farah is a Cambridge academic who has written a brilliant book called The 4,000-Year History of Science. Let me quote from her. Incidentally, she starts a book, the very first chapter the very first paragraph is a citation from Rig Veda, interestingly, when she traces the whole 4,000-year history of science. But let me quote Patricia Farah. Science is not just a final product such as a theorem, chemical, or instrument, but is an integral component of society, interwoven with industry, business, warfare, government, and medicine. And she goes on to say, taking advantage of scientific discoveries entails making decisions about how to use them, political decisions. So she points out that it's absolutely important that policy and political decision-making make the appropriate use of what science has produced and what, not only in terms of knowledge, but in terms of products, especially the technologies. And therefore, it's very important that policymakers do engage with scientists, and scientists also influence policy. Without that happening, much of the scientific knowledge will remain locked up in medical journals or other scientific publications and will not create products. Therefore, partnerships are going to be very important to ensure that science is translated into products which can be utilized. At the same time, to ensure that they are appropriately applied to benefit everybody who can potentially benefit from that is the second translational pathway. And we must also ensure that health systems and public health, as well as many other partners in the implementation pathway, are brought in to make sure that the secondary translational barrier does not thwart the progress, even if the first translational barrier from science to technology has been crossed. So I believe that this particular institution is certainly going to be laying the path for that, providing both the vigor and rigor for translational research. Of course, one of the most important things that I keep reminding myself and my colleagues in Public Health Foundation of India, there are two things I keep reminding them. One is that we may not be able to achieve everything that we want in the most perfect manner. But if the perfect is not possible, perfect the possible. Whatever is possible, try and do it the best way possible. And then try and alter systems, gather resources, build teams to make even the perfect possible. But do not give it up as an unimplementable idea in the beginning. The second is what Louis Pasteur told his research fellows whenever there is an idea to be explored. Keep your enthusiasm, but let strict verification be its constant companion. So you need the rigor and vigor of science to be combined with a very open-minded imagination in order to produce social change. Thank you.
Thank you, sir, for such an illuminating talk. I now request Professor Karthikeyan to kindly come up on the stage and felicitate Dr. Reddy. Sir?
So I was remiss in not introducing Dr. Reddy before he spoke. I was just outside at that time. So Dr. Reddy has been a leading light for all of us in clinical medicine. And he's been a teacher and a mentor as well. So as you can see, the breadth of his knowledge and the breadth and depth of his knowledge, we are truly honored, sir, that you agreed to speak here. And thanks for reminding us of the complexities involved in translation. We were simply thinking about in terms of basic science versus clinical science, but there is so much more to it. And as always, you were articulate and alliterative, so we enjoyed that quite a lot. So normally Foundation Day lectures are lectures, nobody asks questions, but I think you raised many issues, and if you're okay with it, we can open the floor for any questions that they'd like to ask Dr. Reddy.
Usually in meetings like this, we quickly exhaust the clemency of the clock and the tyranny of the timekeepers gets into motion. But since now we seem to have the luxury of time because some of the dignitaries are a bit late in coming, I think Karthik has decided, Karthik has went to the stage. So I'm ready, I'm open.
Venture capitalists in biotechnology and pharmaceutical companies have been investing for the past 20 years. My question to you, sir, is one of the key ingredients that is required to bring about change is culture. And the culture defines the character of an ecosystem. The culture also defines the achievements of an individual. Like say, for example, the same Indians who have gone abroad have excelled because of the system and the culture that is available. What do you think we need to do to bring in that cultural tectonic shift in our thinking so that our scientists deserve greater merit, greater respect, and greater remuneration in the field of translational research? Thank you.
Well, I believe that it's absolutely important for us to have very well-structured systems with predictable mode of functioning, which are not necessarily subjected to vagaries of decision-making and which can be bent in multiple ways, either by prejudice or because of other patronage or whatever. So that is very important. Unless we can instill that confidence, people will not feel vested in their institutions and institutions will not grow. While doing that, at the same time, it's absolutely important for us, as I said, to create teams. Because one of the problems is individuals may function well, but unless they're collaborative, they're not likely to become great problem solvers. And we must create the institutional culture for that kind of a teamwork. It's absolutely important. And unless they feel that they can solve problems together, they will not be able to come together even when there is a challenge that requires them to do that. I do not know whether I'm speaking the correct number or not. In Massachusetts Institute of Technology, I think building 29 or building 22 or whatever, some building with that name, became very famous. And that became the model elsewhere too. That is where they could not initially accommodate the growing number of scientists and faculty members in their disciplinary buildings. So all this supernumerary later entrants were all put in one common building. And suddenly they started talking to each other in the cafeterias and elsewhere. And then suddenly a lot of new ideas and new products started emerging. So they said, oh, we must now recreate these models in other universities. So I think that we must create that culture of collaboration as well, where people can actually have cross-disciplinary linkages. Then, of course, we must ensure that people with good ideas, with proven skills and commitment are given the required resources and support to function. and given the challenges which they can address. You mentioned people being very successful abroad, Indians being successful abroad. I usually attribute two reasons to that. One is, of course, that they have very good systems there and very good structures to support them, apart from a lot of financial resources. But they do even better than their American counterparts because they've learned to survive with multiple challenges in India. They've learned the jugaad. They know lateral thinking. So when an American suddenly is flummoxed, when the predictable pathway doesn't seem to be functioning, our Indians can find the lateral route. So they're very capable of doing that. So I think it's that adaptability that comes through very well. So that systems plus the ability to adapt and innovate is also important. No, have they, are you ready now to take over? Or will you manufacture some questions? We can call the quiz masters back. Right.
Thank you, sir.
Thanks.
Good afternoon, everyone. I, Rohit Kachwaha, on the advent of 15th Foundation of THSTI, would like to invite Professor M. Srinivas to kindly deliver the Foundation Day address. Professor Srinivas has had an illustrious career with more than 3 decades of experience in various positions in the academic, administrative, and research fields. He joined the Institute of AIMS in 1994 and ascended the academic ladder and became Professor of Pediatric Surgery. He has more than 200 publications and has trained a large number of undergraduate and postgraduate medical students. His administrative experience includes the development of new medical colleges, wherein he initiated setting up more than 10 new medical colleges and initiated postgraduate and super specialty courses in these institutions. Professor Srinivas received a prestigious fellowship from the Children's Research Center in Dublin, Ireland, and returned with a rich experience to teach and train in India. He is currently the director at the All India Institute of Medical Sciences, New Delhi. I request Professor Srinivas to kindly come to the podium and deliver the Foundation Day address. Welcome, sir.
Thank you, Dr. Karthik. The more the delay of the dignitaries, more people keep ready for the Foundation Day lectures. I was not told. On the pretext of calling for felicitation to Professor Reddy, he gave the question answers. Good evening. Well, Professor Reddy has been a teacher for all of us. And after the foundation is done very well. So there is no foundation again lecture. So the only thing is that we were just discussing with him that how best, now Dr. Pramod has also come. How do we bring the institutions, what Sir told, the most important thing which was striking, whenever Professor Reddy speaks, a lot of things goes to our mind that, how do we change ourselves? How do we change our institutions? He thinks not just the institute, the country, public health, beyond that. And that's the reason why he's not only in India, but he has been a teacher across the world. And I was just wondering that how do we bring the people together? The most important thing is we were looking at our own institutions, that how do we build the teams? how we can connect the people together. It is connecting the people is the most important thing. It is within the institution, say within the All India Institute of Medical Sciences and the THSTI. And then once we bring the people together, the groups will form, the basic science and the clinical science is what sir was telling. If we can bring them together, put the higher purpose, what sir was telling you, the challenge, the jugaad and everything, the challenge, the reason we excel, the reason where we can deliver is when we have a higher purpose, the goal has to be set in. When you put the higher goals, what sir was telling about the policy and advocacy and everything, we have big goals, for example, even during the COVID time, Professor Pramod Garg is here, this institution had a mandate that how you will get your own vaccine, our own vaccine for the country. So when you have a higher goal, people will sit together, whether it is clinical scientists, whether it's the basic scientists, they will sit together, they will work together, things will happen, that will definitely happen. So that's where we need to keep the institutions and the leadership of the institutions and the government and the policymakers and advocacy people. They need to set the goals that these are the institutions which are there in the clinical sciences, these are the people in the basic sciences. They have to sit together and work on that and do it. And we have wonderful opportunities, at least for All Indian Medical Sciences and the THSTI. We look forward for the wonderful opportunity of working together in synergy. What sir was saying, silos versus synergy, that's where we can work together. There are a lot of complementary things. In fact, we were discussing with the present director here and the former director here, both of them are here. How do we bring the people together, in the form of bringing the students over here? It could be a sort of observership, a fellowship, a combined PhD programs, which we were thinking of it, how we can bring the people here. And the people can be PI and co-PI within the institutions. Again, that will bring the people and the labs and work together so that they can deliver and answer to the needs of the country and the institutions. And then the other concept which we're thinking about, the visiting professors and the adjunct faculty members. Faculty from here can be there and faculty from there can be here. That is something which we are working on that, and then we are sure that we have been thinking of that. Now I think we will sit together and then work out that how best we can bring these things in place so that we can work together and deliver more than what we are thinking. Another advantage of the THSTI is that you have the strong basic sciences and the clinical trials which we can do here. That is something which this institution alone cannot deliver. Because you need a strong people of clinicians cutting across all the what sir was telling, multi-sectorial, multidisciplinary. That is something which the AIIMS Delhi can definitely give it to the THSTI. We will be in a position to support and help not only from the level of the design and the clinical trials, but also during the clinical trials and all the things. The building is ready here, and then I think they are shortly going to start all those things where the clinical trial units, the human trial units is going to happen. And that is something which we will definitely take part, and so that we can work together and then see to it that we come out with whatever we need to look into that, especially for the newer molecules, newer vaccines and everything. Another advantage of THSTI is that the industry collaboration, unlike the All Indian Medical Sciences, the THSTI has got that mandate and they are doing excellent work, reaching out to the industry and the people who have already had research on those areas. Unfortunately, we don't have at All Indian Medical Sciences, and we are planning to have that kind of the units there. And until then, that definitely we will work together and see to it that the gap which we have at AIIMS New Delhi is filled up by the THSTI. In a nutshell, I can say that it's a win-win situation for both of us, two organizations and the people sitting together. Definitely, we will have a lot of impact, because what sir was telling, the social good, public good, that is something which definitely will come out of these two big institutions, because we have strength in our areas, and the weaknesses whichever we have in the individual institutions is filled up by the other person. That is something which will definitely add on to our strength of the two institutions working together. Definitely we look forward, and then this is my request to sir, because he has contact with the people and the Honorable Ministry is also going to come now. We will definitely speak to them and tell them that how we can bring these institutions together so that we can work together and then deliver for the public good. Thank you very much. I can take questions. Before you say that you are being felicitated and then look into the watch and then they say the airport, the flight is delayed, I will take definitely questions. I'll take questions, let them. Yeah. You have time? Yeah, please. By the way, he was our neighbor at Asian Village. Whatever Professor Reddy has not asked, I'm not going to record that. Whatever is answered is answered. Anything with respect to how we can bring together, that is something which we can do together because we are there together now. Bringing together the institutions. Yes, please. Yeah, as I just said that, the first thing is that we should come here, our students should come, our PhD scholars should come because there are excellent facilities. Last time when I visited, I was very surprised that so much of things are there and then how we could have utilized this. I always look into that. So that is something which we can, and there is enough space for them to go around and then hustle bustles of the South Delhi. They can be here for some time and then interact with your people. There's some of the people who are so good in those specific areas. Definitely it will inspire and ignite those young minds. And then whatever they are doing, they will continue to be there. And then once the students and the faculty and the research scholars come, then definitely the faculty will come over here. We are already talking about how we can get the adjunct faculty, the visiting professors and all that. And then once it happens, then the research will go on. The PI will become co-PI, co-PI will become PI, both sides. It will go on and then we will be in a better position to take care of whatever the resources we have optimally in these two institutions.
So since industry representation is here,
Yes,
one of them,
yes.
I think let me ask on behalf of them. If AIMS and THSTI comes together, how would they get benefited? Like, for example, if they were to advance some of these leads, either originating from THSTI or NCR biocluster, so that those leads can see end of the light. Any thoughts on?
See, basically either you come with a molecule or a vaccine or a gadget or equipment, That's where the industry. Unfortunately, most of the best of the institutions, the medical institutions in this country have not had that kind of access to the industry. We were not that good in this thing because one, we are lost in our own clinical work and the support system within the administration, the government system was also not, that much of supportive to the people to go reach out to them in the form of, say, the protected time for the clinicians to go into the research, the time which they can spend it to the reaching out to the industry, take the patents, have a Section 8 company or something like that, and be part and parcel of those things. That we did not do, let's say, institutions like IITs have done. The medical institutions in, at least government institutions have not done that. That's where I think the thing which we lack at AIMS Delhi is something which you already have done it. The connect between the THSTI and industry is definitely more than what the AIMS has got with the industry. And that's where they can come to through you. And then we have all the necessary infrastructure here for the clinical trials. So definitely, if All India Institute, which institution else can do in this country? The testing, validation, all those things. I don't think any other medical institution has got that kind of infrastructure, the people, the expertise and experience, and what sir was telling them, all the disciplines coming out, all the multidisciplinary specialties and super specialties and basic sciences also come together and then be part and parcel of testing, validating, giving the feedback to the industry, and then getting the different versions, higher versions, and course correcting all that. That's something which we can do together. That is something which I see a huge potential where the AIMS and the THSTI can collaborate with together.
Thank you.
Hello, I'm Sachet Shukla from Jivanu Therapeutics. And from an industrial perspective, one important component in all of this is the regulatory aspect. So anytime if we have a new therapy and, navigating the space with CDSCO and the regulatory authorities, for a startup, it's really a very big burden to bear. So any thoughts that you have on that and how we could work together with AIMS and THSTI to take that forward? Thank you.
As I said that, there are issues, institutions like AIMS, we could not reach out to the industry as much as what we could have done it. But of late, the COVID has taught us many things. With the COVID things, many things have changed. Our understanding has changed. And we also know that we need to be very fast and then cut down on those these things. And it should not be a big problem if there is a potential molecule or a gadget or equipment or a vaccine or whatever you are bringing from there through the medium of the THSTI, the basic scientist of this place and that place, AIMS New Delhi. Definitely we can go for the clinical, whatever the trials, there should not be a problem. Fortunately, we being in health ministry, we are in a better position to talk to the people and also get through the ministry for those regulatory things also, whether it is from the ICMR, whether it is the equipment testing units of the ICMR or the CDSO. I think we'll be in a better position to interact with them and get those things done.
Hi, this is Bhavesh Chaudhary from Serum Institute of India. Very nice talk, Dr. Srinivas. Yesterday's talk from Dr. Paul, I think we are talking about speed, and you know better than us that the clinical trials, the period, and phase 1, phase 2, phase 3 takes a long time. As a clinician, as a future way forward, what's your thought how we can abbreviate that? Sometimes the efficacy is needed, but we don't have the disease specifications and the populations. What's your thought? I mean, I want to know the philosophy, what you think about it. Thank you so much.
You're talking about the clinical trials?
Yeah. Clinical trials prospect for the different kinds of indication. For example, in COVID, we have done in 2 years, kind of stuff, 1.5 years. But that is just a very emergency case, but it's good, lessons for us. But in future, if we need to develop as a prototype model, as a clinician of this country, how you think that it's better abbreviated and what preparation we should do now?
Whatever we could do in COVID will not happen with the non-COVID drug. Let us be very honest to that. There was a lot of things which we jumped. It was very faster. And then the regulatory people also have a lot of leniency in that time. And that kind of things we can't expect in the present scenario. But what is definitely what I'm saying is that, it is industry with the THSTI and AIMS, which we can do better than what AIMS with industry can do alone, or the THSA with industry can do alone. That's what I'm saying. Once we have the person, who are there here, for example, if the look into the clinical trial which they have built up here and all, even if we want to do it from Alliance Medical Sciences, it will be little difficult for us because we don't have that kind of infrastructure now. There are clinical trials happening in AIMS that continue to happening, and it has always happened. But if they're looking for something which is a totally dedicated institution, building a new molecule, human trials, that's what we can do over here. And with the visiting and adjunct faculty, and then bringing a synergy between institutions, what we're talking, we'll definitely be able to facilitate that, what you're talking about. Definitely there will be a faster resolution of the things because once the people are there, and it will happen, there should not be much of a problem. I don't foresee it.
Hi, sir. This is Saurabh. And the question is very simple in terms of what challenges you foresee in coming future in terms of the medical arena, what you are feeling in terms of like pandemic came to the world or in India, for Indian perspective, what you foresee in next 2 years for the medical arena?
Well, I think Professor Reddy is the right person to talk about this. Now the question has been put to us. You see, look into the present scenario. Even if you go to any of our ICUs, even in the government institutions, most of our equipment and gadget is imported, which is not right. It's not that we don't have that kind of brains or technology or something to do about it. And the problem with that is the more we get these imported items and have that comfort zone of the things, we never bother to have that Indian component or indigenous component or the focus to say that how we can become Atmanirbhar Bharat. That was lacking. Now with the government policies of bringing the 20% and then the Make in India component and all that, it helps in two ways. One, we have known that we were in the comfort zone, we need to come out of the comfort zone. That realization has happened to most of us because we were just importing right and left all the things from abroad. Second thing is that it is also given the companies an incentive, not only the local collaborator or the distributor or a person who's associated with the companies and the multinational companies, that if they want to have the big volumes, because India consumes a big volume, then there's a chance and a potential that they can put their own industry over here to whatever possible extent and bring that local component so that the local people also get the thing. And it is possible here, whether it is with respect to the expertise, whether it is with respect to the engineers or the clinicians, basic scientists, depending upon whether it is equipment or a molecule or a vaccine. So it is possible. So it's a win-win situation. By saying that the country should go for Atmanirbhar Bharat, we have realized that it's a time for us with the focus from the government and the policy advocacy which is happening, that we are rather forced to go into that side of thing. It has a flip side to that, because what happens if we don't have that kind of quality and standards in our own thing, and we start claiming that, this is the best thing equipment, which is equivalent to what is the equipment which is imported from outside, we fail the system. That's what is sometimes happening, and we have had issues with our procurement and purchases in the institutions and the government institutions also, because now it has become everything transparent, GEM, Make in India component. We are also the victims of the same sometimes. So it ultimately, the policy is good. It is, we drive that. If we are honest enough to that, and if we keep our interest and our country's interest up, of course, not compromising the quality and the standards, we will be able to deliver that. There should not be a problem. At least something, thinking is gone. Some initiative has gone on this, and the people are thinking now that how we can have the Make in India component and make this thing successful. Where is the flight? I have to understand.
Hopefully soon. Hi, this is Abhinav. I'm from a company called Sphera Pharma. We are into small molecule-based drug discovery. My question is, centers like, and specifically AIIMS New Delhi, your focal point for a lot of referral cases across from across the country, which is very relevant for rare disease drug discovery. Can we, as industry, look at AIIMS to create some sort of a clinician interaction platform where companies like us can actually talk to clinicians and gain insights, not just when we are ready to get into clinical trial, but much earlier on from a preclinical development space, improve our trial designs and improve our preclinical strategies to get better shots at discovering drugs for rare diseases in India for India.
Definitely, I think there cannot be a better institution than All India Medical Sciences New Delhi for this, because it's, look into the load. We get almost 15,000 OPD patients per day. And you would name the rarest of rare disease, it becomes not rare in names. You look into the numbers. It may be rare for the textbook or the incidence wise, but because the entire country refers, the people come from not only AIIMS but also SAARC countries. Definitely the rarest of rare disease is also sometimes common disease for us. And there are groups which are working on those rare diseases. You can name the disease. There is a clinic which is running on that. Definitely you're most welcome to speak to the clinicians. Have the brainstorming with them that what are the challenges which they have and what is that which we need to do with respect to the diagnosis of that particular disease, or the therapeutic point of view, what are the molecules which are available, what are the things which we are getting from abroad, what is that you are planning to do, is there anything which they can help in the say animal experiments or something. That again, we can do that. THST can do that. We can do together also. What is that we can do together? And then once it comes to that again, we can do together the clinical trials, in all the components, whether it is from the understanding of the disease, whether it is diagnosis, whether it is prognostication, therapeutics, clinical trials, in all parts of that, we are there. We are there to help the people, the industry people. Nothing prevents you reaching out to us. There are, most of the rare diseases have got the clinics now. You name the rare disease, there is a rare disease clinic in AIIMS. You can reach out to them and speak to them. What are the needs and what are the expectations of us from you? And then you can tell us that what is that we need to do for you. Backbenches. Flight is not arrived, I know that. Otherwise, you would have gone out. Who is sitting here means flight is not arrived.
Anything?
Thank you, sir. I now request Professor Karthikeyan to kindly felicitate Dr. Srinivas.
For the speech or the question?
For the question.
Oh, see, he just got.
It'll keep on growing. So I would like you to stay back on the stage for the prize distribution. And I would also request Professor Srinath Reddy to kindly join us on the stage for the prize distribution. So at THSTI, we have a custom of conducting scientific colloquiums every year. And this year, we have 5 students receiving Best Scientific Colloquium Presentation prize. And the PhD students are Mr. Vaibhav Nayan, I request Mr. Vaibhav to kindly come up on the stage. Ms. Meenal Chawla, Ms. Samriddhi Mehta, so yeah, this is Ms. Meenal Chawla, Ms. Samriddhi Mehta, Ms. Saloni, and Ms. Taruna. So that's Mr. Vaibhav. We also had poster presentation competition, and the competition was basically divided into 2 categories. The first category was the scientist category, and the winner, so there are 3 winners, so there's no first, second, third, but the first person that I'll be calling out is Dr. Suchitra Gopinath. Next up, we have Dr. Rajdeep Dalal. And Dr. Daisy Talukdar. Dr. Daisy. Moving on to the PhD student category, we have Bhavna Verma, Rohit Kachwaha, and Jayendra Jyoti Kundu.
As we all know by now, I mean, the barbs notwithstanding, we know by now that the dignitaries are delayed. But I think we did have some useful discussions in their absence. So I can ask the compeer to come here and decide what we do next.
To keep up the zeal of the event, we have Dr. Manas presenting a cultural event. He'll be performing a few songs. So I welcome Mr. Manas.
Hello.
We request everyone to please be seated on their seats.
की ग्रेट होने दो
Hello.
अरे सर
Hello.
इसमें जोर है
Hello.
सेम बराबर है सर
तो
हम उसी हिसाब से गाएंगे
I request everyone to please kindly be seated.
आ रहे हैं क्या बेंच से आ रहे हैं चलो प्ले करो सब बैठ जाएंगे प्ले करो प्ले थोड़ा और थोड़ा
Hello. कैसे देखे सपने लगी कैसी है
बात तेरी सच्ची लागे बोल तेरे बोल तेरे मीठे मीठे बात तेरी सच्ची लागे मने मने कितनी अच्छी लागे शान से तेरी गजब की सरब की लड़ी से दूर बड़ी से साथी लागे मने मने कितनी अच्छी लागे साथ के मौसम में तन्हाई के आलम में मैं से निकल आया होटल भी उतार तो जीरो में दो साथ में अभी जिंदा तो जीरो में दो साथ के मौसम में तन्हाई के आलम में साथ के मौसम में तन्हाई के आलम में अभी जिंदा Thank you.
Thank you, Manas sir, for such an enlightening performance. I request you all to kindly be seated.
ये बैटरी रख लो।
I request you all to please be seated since we are about to start with the cultural program now.
For our next performance, get ready to be dazzled by the incredible moves of our very own MSc student and Kathak enthusiast, Ms. Soumya Chauhan. Please welcome Ms. Soumya, who promises us to spin into world wind of elegance. Thank you. Guys, everyone please assemble in the auditorium as we are almost ready to begin with our Kathak dance. She is just about to come and perform.
Guys, can we have a loud round of applause for Soumya?
रंगी परोड़ आवी खुशियों संगे लावी अलगाए हयो हाय हाय आशानी के अलवेदी गुनगुनाए हाय हाय हाय
हे शुभारंभ हो शुभारंभ मंगल बेला आई सपनों की बहरी दिल की बाकी रे शहनाई शहनाई
रास रचीलो साज सजीलो शुभ घड़ी चेरावी आचा आचा टमटमाता शोळा चेरावी
नीचे लगी रब ने तस्वीर बनी आज हवा पानी को मिला तो फिर तस्वीर सजी झरे 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 बिजली की बानी कोई कोई अनछुई तुझ में है कुदरत सारी खोई कुछ में है कुदरत सारी खोई खोई तुझ में है कुदरत सारी खोई खोई
Guys, wasn't that such an amazing performance? She definitely made us all dance on our feet. And thank you very much, Soumya, for such an energetic performance.
While we are waiting for Honorable Minister to join us, I would request if someone from the audience wants to come up on the stage and perform. We'd like to request you all to welcome Mr. Sachin from CDSA, who will be performing a few songs for entertaining you.
Hello, hello, hello. इसमें ये ऑन द स्पॉट है, तो अगर कुछ इशू हो जाए, थोड़ा बहुत ऊपर नीचे हो जाए तो चलेगा। म्यूजिक थोड़ा एक पार्टी तो से दूर तक निकाल दूर तक निकाल कुछ साथ पास दिखा दो तो ये सिलसिले दूर तक निगाह झुलते हुए
मेरी सांसों में बसी खुशबू तेरी ये तेरे प्यार की है जादूगी फासले हुए तो सिलसिले हुए दूर तक मेरा दिल है तेरी पनाह छुपा तुझे न जाऊं तेरी तस्वीर है निगाहों में दूर तक रोशनी है राहों में अगर रोशनी काफी हुए प्यार के नजारे दीप में जले हाय किताब तो ये सिलसिले दूर तक निगाहों में खिले देखा है किताब तो ये सिलसिले दूर तक निगाहों में खिले
Guys, for now, for our next performance, get ready to be transported into heart of Haryana. Okay. Yes, sir. Guys, please everybody, I request everybody to assemble in the auditorium first. We request everyone to kindly assemble in the auditorium since Honorable Minister will be joining us any moment now.
It is an immense pleasure and proud privilege to have our Honorable Minister of Science and Technology and Earth Sciences, Dr. Jitendra Singh Ji, who is gracing the occasion today. We render our heartiest gratitude to you, sir, on behalf of the entire THSTI family. A medical degree from Stanley Medical College, Chennai, and MD in medicine from Government Medical College, Jammu, have paved his path to become one of the distinguished endocrinologists in the country. His notable research work on stress diabetes in Kashmiri migrants and his contributions towards guidelines for management of diabetes in pregnancy have been highly acclaimed by the World Health Organization. From a successful physician, his journey towards thought leadership core group in the government has been a truly exciting and inspiring one. नमस्कार, सभी अतिथि गणों का THSTI फरीदाबाद के MK भान सभागार में स्वागत है। ट्रांसलेशनल हेल्थ साइंस एंड टेक्नोलॉजी इंस्टीट्यूट THSTI DBT का एक प्रमुख संस्थान मानव स्वास्थ्य में सुधार के लिए अत्याधुनिक क्लिनिकल, ट्रांसलेशनल एवं बुनियादी अनुसंधान में लगा हुआ है। मातृ एवं शिशु स्वास्थ्य, टीके, क्षय रोग, विषाणु विज्ञान इसके प्रमुख क्षेत्र हैं। THSTI ने प्रबंधन प्रोटोकॉल को बदलने की क्षमता के साथ समय से पहले जन्म के कारणों को समझने के लिए 12,000 अधिक गर्भवती महिलाओं और 5,000 से अधिक शिशुओं का सबसे बड़ा समूह विकसित किया है। THSTI ने एक SARS-CoV-2 टीका विकसित किया है, जिसके नैदानिक परीक्षण के पहले चरण में जाने की संभावना है। THSTI एक पैन बीटा कोरोना वायरस वैक्सीन के विकास में प्रयत्नशील है, जिसके लिए THSTI को $12 मिलियन की फंडिंग प्रदान करी गई है। THSTI ने एंटी-टीबी लीड मॉलिक्यूल्स विकसित किए हैं। THSTI के वैज्ञानिक गैर अल्कोहलिक फैटी लिवर रोग के लिए भी उपचार विकसित करने के लिए प्रयत्नशील हैं, जिनमें उन्होंने छोटे अणुओं एवं जड़ी बूटी आधारित सूत्रों से लीड्स की पहचान की है। THSTI परिवार की ओर से हम माननीय डॉक्टर जितेंद्र सिंह जी का MK भान सभागार में स्वागत करते हैं। माननीय डॉक्टर जितेंद्र सिंह जी, इस महत्वपूर्ण क्षण को और अधिक गरिमामय बनाने के लिए आपका यहाँ उपस्थित होना हम सभी के लिए सम्मान एवं गर्व की बात है। आपकी उपस्थिति हमें ऊर्जा एवं प्रेरणा प्रदान करती है। हम आपके इस सहयोग और समर्थन के लिए हृदय से आभारी हैं। प्रोफेसर कार्तिकेयन जी से नम्र निवेदन है कि वह माननीय डॉक्टर जितेंद्र सिंह जी एवं डॉक्टर राजेश सुधीर गोखले जी का मंच पर आने में साथ दें। आप सभी से अनुरोध है कि दीप प्रज्वलन करें। दीप प्रज्वलन के इस पावन अवसर पर आप सभी का हार्दिक धन्यवाद।
I now request Dr. Jayanta Bhattacharya, Dean, BRIC THSTI, to kindly deliver the welcome address.
Good afternoon, all. It's a privilege to welcome our distinguished dignitaries to THSTI's 15th Foundation Day celebration. We are extremely honored by the presence of Dr. Jitendra Singh Ji, Honorable Minister of State, Independent in Charge, Ministry of Science and Technology, Ministry of Earth Sciences, Ministry of State for PMO, Ministry of our Personnel, Public Grievances and Pensions, Department of Atomic Energy, and Department of Space, Government of India. And we are also equally honored to have Dr. Rajesh S. Gokhale, Director General, BIC, and Secretary, Department of Biotechnology, to celebrate with us THSTI's 15th Foundation Day. I also take this opportunity to extend a warm welcome to Professor Srinath Reddy, Honorary Distinguished Professor and ex-President of the Public Health Foundation of India, and Dr. Srinivas, Director, All India Institute of Medical Sciences, New Delhi, for joining us in our celebrations. BRIC THSTI, though a relatively young institution within the DBT family, has emerged as a vibrant hub for translational research. Our collaborative environment fosters the convergence of diverse expertise from physicians and biologists to chemists and mathematicians to transform groundbreaking ideas into impactful healthcare solutions. In just 15 years, we have established a reputation for scientific excellence and cutting-edge infrastructure. Today, we are privileged to witness the inauguration of few more new facilities by our Honorable Minister, Dr. Jitendra Singh Ji, which we, I'm sure, will further strengthen THSTR capability for translational research. I hope this Foundation Day will be a memorable occasion for all, filled with inspiration and a renewed commitment to our mission of bioinnovation. Once again, a warm welcome to all of you present here. Thank you very much.
Thank you, sir. Yesterday at THSTI, we hosted Synchan, Synergy in Science built through elements of collaboration for high quality data and expertise in NCR Biotech Cluster. Synchan was a bright opportunity for collaboration between the institute and different industry partners. Our outreach to industries has been incredibly successful, leading to an exciting collaboration between BRIC THSTI faculty and our industry partners. To celebrate this achievement and further solidify these partnerships, we are signing letter of intents and agreements for collaboration with our industry partners. We'll now be announcing the names of these companies and request their representatives to kindly come up on the stage. First up, we have Serum Institute of India Private Limited. I request Mr. Parag Deshmukh, Dr. Bhavesh Chaudhary, Dr. Sriram Raj Pathak, Dr. Jehan Vapiwala, and Dr. Saurabh Kalmakar to kindly come up on the stage for agreement of research and development of product against Nipah and agreement of research and development of product against respiratory viruses. From Genova Biopharmaceuticals Limited, we welcome on the stage Dr. Swarendu Kaviraj for agreement on research development on vaccines against Nipah and influenza. From Govind Capital 2, Singapore, we welcome on stage Dr. Anand Govind Daluri for the letter of intent between Quantum Bioinnovations Fund and THSTI to invest in biotech spin-offs with the aim to promote startup entrepreneurship and commercialization of biotech projects. From Metlin Biotech India Private Limited, we welcome on stage Dr. Shivakumar Natarajan and Dr. Purudvidhar for letter of intent to advance research in cell and gene therapy through innovative technologies. From Polyamnia Nanotech Private Limited, we welcome to the stage Mr. Krishnan and Mr. Saurabh Sachdev for the letter of intent for collaborative research to reduce hospital-acquired infections. From Omni BRX Biotechnologies Private Limited, we welcome to the stage Mr. Ravindra Patel and Mr. Jagdish for letter of intent for process development, testing, and validation for CAR T cell therapy manufacturing. From Lysine Biotech Private Limited, we welcome to the stage Dr. Ananda, Dr. Das Gupta, Amit Lahoti for the letter of intent to collaborate for CAR T cell therapy. From Jivanu Therapeutics Private Limited, we welcome to the stage Dr. Sachet Ashok Shukla for the letter of intent to collaborate for manufacturing and commercialization of specific bacteria with therapeutic implications. From Ruvenil Biomedical OPC Private Limited, we welcome to the stage Dr. Rajkumar Halder for letter of intent to collaborate on bio and microbiome therapeutics. From Argan Life Sciences Limited, we welcome to the stage Mr. Vikas Katyal, Mr. Rahul Verma for the letter of intent for biologics product development and manufacturing technical collaboration. From Sphera Pharma Limited, we welcome to the stage Dr. Abhinav, Dr. Saumidatta, and Dr. Yashika Mattu for letter of intent for co-development of mTOR inhibitors for NAFLD-associated HCC. From Pursued Life Sciences, we welcome on the stage Dr. Ajay Soni for the letter of intent to collaborate for drug discovery. Thank you, sir. Professor Ganesan Karthikeyan is a clinical cardiologist. He is a professor of cardiology at All India Institute of Medical Sciences, New Delhi, India. He completed his training at AIIMS, New Delhi, and at McMaster University, Canada. He is a fellow of Academy of Medical Sciences and National Academy of Sciences, India. His research is focused on cardiovascular diseases affecting low and middle income countries, including rheumatic heart disease, mechanical valve thrombosis, anticoagulation, and indigenous drug-eluting stents. It's an absolute honor for all of us to have such an accomplished icon as our leader. I now request Professor Karthikeyan to deliver the inaugural address and kindly present the annual report.
Good evening, everyone, and the Honorable Minister. Thank you very much for gracing this occasion. Dr. Gokhale, for staying with us for such a long time. He was here yesterday as well. My teachers, Dr. Srinath Reddy, Dr. Srinivas, Director of AIIMS, Dr. Garg, former Director of THSTI, and my colleagues, my friends, and students. I won't take too much time. We've spent a lot of time. So I just want to highlight here some of the major work that has been done at THSTI over the last 1 year or so. If I miss out somebody's work, it does not mean that it is not significant. It is just that I just want to, in the interest of time, we'll only talk about the main things. This is something which we hope to change. So you will see that there is a medical research center and medical translation research facility, which the sites are marked already. And hopefully the next year when we show that, we'll have the actual buildings there. Everybody knows that there are several research verticals here at THSTI. All of them work separately and also in collaboration. And we'll just highlight some of the major work that has happened in these within these research verticals. One of the most important developments in the last year has been the development of novel human monoclonal antibodies to Nipah virus. Several candidates are there and they are undergoing testing in collaboration with ICMR at NIV. And we hope to have a product which is useful for this very deadly disease. Simultaneously, the work that has been going on HIV, it's a forgotten disease, but it's still there in this country. And the prevalence serovariant in this country, we have developed monoclonal antibodies, our scientists here, and they are again in a state where they can be translated into a product which can be useful. THSTI has become, only a couple of months ago, was declared as being part of the prestigious WHO's COVINET reference laboratories. Very few laboratories in the world are accorded this privilege. That is a testament to the work, the high quality of work that is done here. We have been consistently doing a lot of high-end vaccine-related work here at THSTI. Professor Garg was instrumental in creating the VDDC, the Vaccine Design and Development Center, earlier. And we have pre-GMP level facilities for production of protein-based vaccines and biotherapeutics. We already have established industry collaborations with major vaccine manufacturers in the country, including Serum and Genova, Panacea. Two of the, so we have focused on the major viral diseases that are important in this country. And I'm happy to say that the Beta Corona platform and the influenza tetralite, which is actually going to create an affordable influenza vaccine for this country, are both entering phase 1 trials later this year. this is something for which the Peter Corona, the platform for which we have received a patent. We've also worked on other promising candidates for Mpox, monkeypox, and the MERS virus as well. There have been several other key research findings which are important in how we think about disease and how we manage disease, both in SARS-CoV and other febrile illnesses in this country. So many of the researchers have made important contributions to this field. The Tuberculosis Research Center has been active for many years now, and one of the most important contributions is the development of CRISPR knockout variants of MTB. Many genes have been knocked out, and we have a library of these variants which are in great demand around the world, not only in India, but around the world as well. More recently, our group here have identified a class of drug candidates which look promising for both XDR and MDR-TB as well. And we hope to take these things forward in our collaboration with industry in the near future. The microbiome work that is going on here is quite unique. There is no other laboratory or in the country which is doing this level of work. So we have more than 6,000 isolates drawn from several hospitals across the country. So we have a very representative sample of microbial cultures. And we have now genetically defined all of these human microbial culture collection, which is now being used. It is going to be a repository which is going to be accessible to everybody, anybody who wants to use it for research purposes. So we are setting up this, and we've already set up this. We hope that the Honorable Minister will inaugurate this facility while he's here today. In the field of drug discovery, there is a whole unit which looks at small molecule drug leads, and there have been several small molecules which have been developed for NAFLD, which has been an area of interest for many years, even before Dr. Garg was here at the helm of things. And there is a promising dry blood spot method for detection of metabolites which are related to NAFLD, again, which is related to this disease. So that may prove to be useful for rapid diagnosis of NAFLD. The in vitro diagnostic group has developed several point of care tests which were very useful during the COVID pandemic. But they've also developed several IVD POC devices for the diagnosis of dengue and other febrile illnesses. The future is that we will have rapid tests for multiple diseases in the same strip. So that will be quite useful for in the diagnosis of febrile illnesses. There are several other cutting edge work that is going on here, including development of biocompatible artificial material and human liver organoid models. At least a couple of our scientists are working on this, and these can be very useful in drug development and toxicology studies. This is again a very unique work that is going on here at THSTI because of our the maternal and child health division that has been doing great work here. So one of the offshoots of this is that all the images that are available now with us have lend themselves to artificial intelligence analysis, and the group has developed several uses for these, and some of these are validated. So it is easy for now for using these algorithms, we can actually detect accurately gestational age much better than what Western algorithms tell us. So, and there is also a blind sweep ultrasound imaging method, which can be used in the periphery by untrained ultrasonologists. So that can also be used. This is largely AI driven, and it can tell us about gestational age in the periphery, even when we don't have skilled ultrasound technicians. There is, again, more basic work going on in terms of the role of the placenta in determining preterm birth. So this is work that is done by several of our PhD students here under the umbrella of maternal and child health. This again, so there are several areas in THSTI which are not found in most other institutes. So we have the computational and mathematical biology unit, which dedicates itself to looking at model-based analysis of molecules, how they interact with each other. And it's not just a theoretical exercise. There have been several leads which have been developed and which have gone into phase 1 or phase 2 testing. And we hope to see more of this with the interest that industry has shown in yesterday's Syngenta. Scientists here have established a platform which lets anybody determine protein interactions and how two proteins may interface. And the result of this has been that this is a platform that has been developed locally, indigenously, and it is available for anybody to use. And as a result of that, several leads have been identified and patents have been applied for these. For example, we have detected autophagy inducers or intracellular NO enhancers. All of these can be useful as drug candidates for different diseases. So we hope to see again these things going into translation mode with our interaction with industry. This is another useful tool that has been developed here. This is work on the antimicrobial resistance that has happened here. So we now, our scientists here have developed tools, which is a web-based tool where you can enter the bacterial isolate that has been isolated in any culture specimen anywhere in this country, and you can get an idea of what it is likely to be resistant to. This is all entirely based on the proteomic profiling and genomic profiling of these organisms. And this is freely available to use. And all this work has been supported by the high-end facilities that we have. The experimental animal facility, which has now been refurbished completely, and it is a high-end facility which has got everything that people may want to do, including CT scans and procedure units here. And we have the ability to do breed nude mice and knockout mice as well. So this is something which the Honorable Minister had inaugurated earlier, and he's been very supportive in the development of this. And hopefully we'll be able to show him the new one. And I'm happy to say that our scientists have received a large grant for the centralized animal network lab, which is going to be housed within the animal facility. And this is going to enable us to really amp up whatever we are doing in terms of animal-related work. So this is a summary of the many other facilities that we have, and all of these have been supporting both our scientists and the other scientists in the campus, which includes the scientists from RCB and any others who want to work here in our laboratories. And the industry also worked with us during COVID. This is just a summary of the achievements which were the result of all the work. So we have more than 85 publications in very high-end journals, high-impact journals. And we've filed 9 patents, 2 of which have already been granted. So I'll just end here by, again, reminding you that we have the new MRC coming up, which is almost ready now. And we will soon have the translation research facility as well. And hopefully in the next year, this picture will look much better. Thank you very much.
धन्यवाद। यह हमारे लिए सौभाग्य की बात है कि भारत सरकार के जैव प्रौद्योगिकी विभाग के सचिव एवं डायरेक्टर जनरल बायोटेक्नोलॉजी रिसर्च एवं इनोवेशन काउंसिल ब्रिक डॉक्टर राजेश सुधीर गोखले जी हमारे बीच उपस्थित हैं। डॉक्टर गोखले जी ने इंडियन इंस्टीट्यूट ऑफ साइंस बेंगलोर से पीएचडी करने के पश्चात स्टैनफोर्ड विश्वविद्यालय से पोस्ट डॉक्टरल रिसर्च करी। इसके पश्चात डॉक्टर गोखले जी ने नेशनल इंस्टीट्यूट ऑफ इम्यूनोलॉजी में संकाय के रूप में कार्य किया। डॉक्टर गोखले जी ने निर्देशक के रूप में इंस्टीट्यूट ऑफ जीनोमिक्स और इंटीग्रेटिव बायोलॉजी का नेतृत्व किया है। डॉक्टर गोखले जी अपने क्षेत्र में कई सम्मानों से पुरस्कृत हैं। उनमें से भारतीय विज्ञान अकादमी, इंडियन नेशनल साइंस अकेडमी की फेलोशिप एवं विज्ञान और प्रौद्योगिकी का शांति स्वरूप भटनागर पुरस्कार शामिल है। डॉक्टर गोखले जी को क्षय रोग से संबंधित अनुसंधान के क्षेत्र में उनके कार्य के लिए इन्फोसिस पुरस्कार से भी सम्मानित किया गया है। आपसे आग्रह है कि मंच पर आकर हमारे साथ अपने विचार साझा करें।
Honorable Union Minister of Science and Technology, Dr. Jitendra Singh Ji, Honorable Dr. G. Karthikeyan, Executive Director, BRIC THSTI, Dr. M. Srinivasan, Director, AIMS, Dr. Srinath Reddy, Honorable Distinguished Professor and Ex-President of PHFI, Dr. Pramod Garg, former Director of THSTI, Dr. Shinjini Bhatnagar, Distinguished Professors, Scientists, Students, and Staff of BRIC THSTI, my DBT and IBRIC colleagues, industry leaders, ladies and gentlemen, it's my indeed a pleasure to be here today celebrating the THSTI Foundation Day. First of all, time flies. It always looks like, it's been 14 years for this institution. And it's one of those institutions that through inception, I have seen it in front of my eyes. And that's why I said time flies. I still remember a day when Dr. Bhan actually called for the first meeting for inception of THSTI at National Institute of Immunology. That time he had invited Professor Eric Lander, who founded Broad, and also Nobel laureate David Baltimore. In fact, both of them came and visited. And I still remember I was part of that group to do the discussion work at that time. And it's a great pleasure to see this organization come like this. Of course, it was exciting discussions with Professor David Baltimore is a very, of course, he's a Nobel laureate, but he's also a very thoughtful leader actually, and has mentored large number of people all around the world. And I think the thought process at that time was how to build institutions that can create fantastic capabilities of doing translational health science research actually. And I'm sure all of you would agree, since last couple of days, many of you are here actually, that this is what we set out to do 14 years back has in fact come true actually. So congratulations to all of you, each one of you, for this fantastic achievement. I have really to thank Honorable Minister for taking time. I think from his really busy schedule, he just arrived at the airport. He's been for last 2 days in Jammu and Kashmir, actually doing all sorts of meetings in different aspects. I think he never misses any meeting. And I'm really grateful for you, sir, for your constant support. And for the last 2 years since I've been here, DBT has done some fantastic things. We should give a big hand of applause for them. I think under your direction, sir, THSTI did fabulous stuff for vaccine development, as you are aware of it, actually. Starting from every single vaccine development activity happened over here, all the immunological assays and various activities, follow-up studies and all as well, actually. So I think without his continuous support, and I think his amazing capability to really just say, go out and do it, and I'll take care of everything, whatever it is, has been just outstanding, actually. And I think we are really grateful for having Minister who's so supportive and who's so knowledgeable in his own areas of ways. I must talk about some of the absolute fabulous contributions of THSTI, actually, being a foundation day to day, actually. Dr. Bhan's real vision was, when he set out, was to look at preterm birth as a mother and child as a big area of discovery platform and understanding how really clinically one can make impact for it actually. I must say THSTI has contributed in the health of kangaroo mother care, which is now recommended by the WHO to prevent premature birth outcomes for mortality actually. And I congratulate the team over here for doing fantastic work over the years actually. Several programs on vaccine development, developing monoclonal antibodies, drug discovery program, microbiome research. All of it has come because of on the shoulders of several of directors who have in fact, worked together in harmony and synchronization along with PhD students, postdoctoral fellows, scientists, and administrative staff who really takes care of these systems. And I'm really proud to say that this has become one of the most enchanting place to come by and think about it for the future of this country. I think there is a transition in every careers of life. And I think 15 years, as yesterday Dr. Paul also said, is a time when, things will start to go, in my opinion, more like a T20 game. And a T20 game, you need a little bit of test experience to play T20, otherwise you only play a blindfold game actually. So if you play T20 game, you have to start hitting up from the right from the day one and actually make an impactful to bring World Cup back home. And I think now THSTI all set, really appreciate and take this new journey with all you leaders from the industry who are here actually, who's wonderful signatories to various letter of intents and various activities that you've talked about. In fact, the collaborations with all of you together is the key to the success. I think let's do some groundbreaking, really research and do some fantastic stuff over here in next few years to come. Let's embrace this opportunity and enthusiasm. And I think this is the time that our country wants all of us to work together, collaborate and make it happen, not for individuals, but for the best of the country. And I think next 25 years of glory is what we are looking at actually. I would stop here again and congratulate Dr. Karthikeyan for his wonderful leadership that he has given in last 4 months. I think it's fantastic to see what all he's been able to do along with, Dr. Pramod Garg always helping him out in the activities, what has helped. And I think it's been a fantastic teamwork. So congratulations to all of you and my best wishes again. Thank you.
Thank you, sir, for the invigorating message. I now request Dr. Karthikeyan to kindly felicitate Dr. Gokhale.
THSTI परिवार के लिए यह अत्यंत खुशी और गर्व की बात है कि हमारे माननीय विज्ञान और प्रौद्योगिकी तथा पृथ्वी विज्ञान मंत्री डॉक्टर जितेंद्र सिंह जी आज इस अवसर की शोभा बढ़ा रहे हैं। हम संपूर्ण THSTI परिवार की ओर से आपके प्रति हार्दिक आभार व्यक्त करते हैं। डॉक्टर जितेंद्र सिंह जी ने स्टैनली मेडिकल कॉलेज चेन्नई से MBBS एवं गवर्नमेंट मेडिकल कॉलेज जम्मू से मेडिसिन में MD की डिग्री प्राप्त करी है। डॉक्टर जितेंद्र सिंह जी हमारे देश के प्रतिष्ठित एंडोक्राइनोलॉजिस्ट हैं। वर्ल्ड हेल्थ ऑर्गेनाइजेशन एवं अन्य अंतरराष्ट्रीय संस्थानों ने भी उनके उल्लेखनीय शोध कार्यों की प्रशंसा की है, जिसमें से कश्मीरी प्रवासियों में तनाव मधुमेह और गर्भावस्था में मधुमेह के प्रबंधन के लिए दिशा निर्देश प्रमुख हैं। एक सफल चिकित्सक से लेकर सरकार में विचार नेतृत्व कोर ग्रुप तक की उनकी यात्रा वास्तव में रोमांचक और प्रेरणादायक रही है। सार्वजनिक जीवन में भी विज्ञान एवं प्रौद्योगिकी के क्षेत्र में उन्होंने उल्लेखनीय कार्य किया है। सामाजिक जीवन के प्रति उनका समर्पण और दृढ़ संकल्प हमारे सबके लिए प्रेरणा का स्रोत है।
Sir, it's an honor for the entire BRIC THSTI family to have you here with us. So the leadership role that you played in both professional and public life have been very impressive. As the Honorable Minister, your dedication and determination in the upliftment of the society through science and technology has always been very encouraging to us and will stay as an immense source of inspiration for the younger generations. To make this year's Foundation Day even more memorable, I would like to invite our guest of honor, Dr. Jitendra Singh Ji, to grace the stage on the occasion and deliver the presidential address.
Good evening, everybody. Dr. Karthikeyan, Dr. Rajesh Gokhale, former Dr. Garg is there, Dr. Srinivas, members of the faculty, Dr. Srinath Reddy, and all the scholars and the members of the fraternity. I think at the outset, I must start by thanking these two youngsters. They spoke beautifully about me. So I was, and I'm sure I assured them I'll try to trust each word that they spoke about me. Because though it was sounding a little embarrassing, but they went on and on and on. But it sounded good, so let it be. Because sometimes, when you come of age and even your own peers, your contemporaries, even your family is no longer interested in knowing what you're doing. So it sounds like a deja vu happening. So that's made a pleasant evening in this very hard-pressed routine. I have to perform the pleasant task of formally congratulating each one of you and also myself and all of us for observing the 15th Foundation Day of the THSTI. And while Rajesh was very beautifully trying to trace the journey from the times when this concept was conceived by Dr. Bhan, what was occurring to my mind is that this has been not a very long journey in the terms of time span, but it's been a long journey in the times of the upward graph that has been recorded by this institute. And that is because 15 years is not much of an age span in the life of an institution. Tomorrow, if somebody chooses to write the history of a nation also, because we are just about 75 years old, I'm not sure whether we'll also get a footnote, because 75 is just like infancy. It's only in the age of a mortal being that when you are 70 or you're 60, then something inside you starts telling you that you are actually moving on. So in a short span of 15 years, many landmarks having been covered. Because what is important in the age of an institution is that the initial years may not be much of an achievement, but they now not only lay the foundation of what is to come, but they also create a perception. So if the 5-7 years initially go off otherwise, वो साहब खत्म हो गया, चला नहीं. We have seen that happening now. There are 21 AIMS institutes, and many of them have dubiously, inadvertently sometimes earned that reputation. Whereas some of them just come up in one or two ways. So it's not only that you have to work, you should also appear to be working, and you should also appear to be registering landmarks. And that I'm glad that this institute has been able to do. Not only there's been a, yeah, you can clap. So there's not only been a substantial work done, but it's also received a certain amount of recognition. And to that extent, also the Department of Biotechnology itself is not a very old department. But I'm glad they don't have a proper address. Because if you ask Rajesh where he sits, he'll say care of ITBP, which is not a very flattering reputation. In the political organization where I come from, we were taught in our early years the 3 essentials for success: Karyakarta, Kosh, and Karyalaya. So you should have an address, you should have somebody to open the door, and you should have a good treasurer. But having not been fully equipped with all the 3, they have still made a mark. In fact, I was just now what we were talking while sitting on the stage. I said, it's high time you should have a separate address. And I'm sure now that today is a landmark day, we'll start very seriously looking for that too. But in the short span of time, even the Department of Biotechnology has not only progressively risen, but has also seized every opportunity to register its distinction and its distinguished capacities. One of those opportunities came in the form of COVID. I mean, suddenly everybody started talking about biotechnology. Most of the television anchors also became biotechnologists. But that is how sometimes, you need one boost. Because for every successful life story, whether that of an institution or an individual, you need to have two prerequisites. A, that you have to consistently be growing, or you have to be consistently going. You can't go out of scene. You have to perform every day. Every morning you wake up to perform. So you can't live on the glory of the yesterday. And secondly, you have to have certain landmarks, which you can recall. So COVID came as an opportunity and proved to be a landmark with the kind of vaccine work that was done by this department. That does not mean that you start praying that you have COVID every now and then. But you will have some other opportunities as well. And I was discussing, of course, the kangaroo mother care, etc. And he was saying that we are now going to have some more inaugurations also, but I was just now talking to Dr. Karthikeyan about the Nipah monoclonal antibodies. I'm sure that is going to give you certain amount of exposure across the world. And one of the earliest countries to have the preclinical animal network, a number of other such initiatives. Dr. Rajesh was talking about Dr. Bhan having, envisaged the preterm research in case of a number of disease and other morbidities happening. I'm glad to tell him, of course, I also have to thank him. Very recently, I referred to him a research work on primordial diabetes that we have started, which is quite in tune with what Dr. Bhan had envisaged. It's going to be a little tough job, asking a woman who's just got pregnant to switch over to metformin or insulin and then suffer the risk of repeated bouts of hypoglycemia, with the promise that then she'll have a disease-free progeny. But there is one project, of course, which is, of course, clinically being collaborated by one of the leading hospitals, and the Department of Biotechnology is also into that. So I think when we move on, we could have an integration of all these institutions together. What has happened is that when Dr. Bhan talked of a certain set of diseases, a certain set of conditions which we needed to look into, maybe that was the signature of the times that he lived. What has happened today, and I think there's also something sometimes more challenging, that we have a biphasic spectrum of disease. In the sense that maybe sometimes in the 80s and 90s, we started telling ourselves and believing that we have got rid of the communicable disease, and we got rid of the diseases which were the cause of morbidity and most of the mortality in the earlier times. Like, for example, smallpox, tuberculosis, cholera, all those things, because with the advent of antibiotics and other antimicrobials, we could succeed to a great extent. Because there were times when many of these diseases were actually considered to be fatal. And tuberculosis, known for its fatality, not only became an accepted condition in the medical families, but even in the social and cultural ethos. So if you created a short story with a love triangle, the best way to get rid of one of the characters was to afflict him with tuberculosis or her with. So it became a part of the folklore. So maybe then somewhere in the 80s and 90s that we delighted ourselves saying that now we have got over that and we got into the diseases which are now being taken care of by Dr. Karthikeyan, the metabolic disorders, the type 2 diabetes mellitus, the vascular disease, the cardiovascular disease. And somebody also in a lighter way and said that now India has entered into the age of civilized disease. Because these were known to be the disease of the West. So we were known to be suffering from diseases which were to a great extent contributed by poor hygiene, poor living conditions, poor socioeconomic status, all those things. So said now, even if you die of a heart attack, you died of something which an elite would die of. So much of our concentration actually started happening that way. That was also the time when, in fact, when COVID happened, I was telling some of the colleagues, I said sometimes I feel that the teachers of my generation were guilty of having moved the focus from communicable diseases, infections, and then moved on to this. Because, and even our residents and our postgraduate, even DMs, boys and girls and scholars, were focusing more on this in the belief that the other part of it was now on the decline. And then suddenly COVID happened. But I must appreciate our young researchers and young medicos, the manner in which they tuned themselves and reoriented themselves to a challenge for which they were not fully trained. So that's why now I said, now we are beset with both these. Since on the one hand, we not only have the metabolic disorders, the metabolic problems, but we have them at a younger age also. Then the Indian phenotype is adding more of the risk element to it, because I mean there are a number of studies which you and I, all of us would be aware of. Even the Indian origin people settling in Europe or America for 3 generations continue to carry same risk over there, in spite of all the other factors having got over, because the phenotype remains the same. And then many of our factors, like for example, the central obesity is some one factor which is an independent risk factor, independent of the overall obesity. In fact, way back in 90s with Dr. Yagnik, Rajesh knows, with Yagnik we did a study to scan individuals comparatively, the Europeans and Indians. And we found that most of the Europeans were obese, apparently, but inside they had very little fat, whereas most of us looked otherwise slim and thin, but there was too much of fat around. And maybe we've learned to also have that sartorial habits. You can wear a kurta and you can camouflage your belly. You can wear an 8-meter sari. So, but that is the fact. So there's a reason. So on the one hand, we are having all those diseases now, which till about 40 years ago were known to be diseases of the West. And the irony is that we are having same disease at a younger age. So the challenge today is the heart attack in the young, type 2 diabetes mellitus in the young. The usual picture that came to the mind when you heard of somebody having an episode of a myocardial infarction or being detected as type 2 diabetes mellitus was that person must be in the late 40s or so. Now it's no longer so. You have in 30s, you have even in late 20s. At same time, we are also continuing to have the diseases which were there earlier, which we thought we had got over. So we again are beset with the problem of tuberculosis, as the program has been started by DBT now. And that is also one of the Prime Minister's call to have TB Mukt Bharat by 2025. And we also have all the other diseases. As he said, HIV may have been got over in many other parts of the country, but we still run the risk of it. So the amount of spectrum that we have to deal with is much, much larger than any other country. When I talk of the India, I mean the entire Oriental belt and the Indian subcontinent. At the same time, we also have a biphasic lifespan. So that is also adding to it. Now, we very proudly say India is the youngest country. Of course, it is. More than 70% population below the age of 40. And those in public life, find it quite pleasant to say so, and all to the boost of the youth energy. But the matter of fact is that though the overall percentage of the population must be more than 70% below the age of 40, but the absolute number of the elders is also on the increase. It's not decline. It's not that the number of youngsters has increased and therefore the elders are ready to quit. no longer so. They are also there around. So in 1947, we had the lifespan of just about 50, 55. Lifespan was just 50 to 55. Now it's gone over to 65 almost. It's all on the increase. So while that is creating that is an issue which goes beyond the realms of a medical or a health professional or a researcher, because it has its social, economic, and political dimensions as well. But as far as we are concerned, when we are dealing with the task of research in healthcare or health-related disorders, what is happening is that many of those diseases which were not happening earlier because people didn't live long enough. So many of the prostatic cancers which are happening so frequently now are happening because you didn't live long enough for that. So we are getting now see the paradox is that the diseases of the later age are also happening in the younger age. And at the same time, you are also living long enough to get the disease of the later age, which your elders or your parents or your ancestors didn't get. So you can see how challenging and how complicated this specter is. Amidst this, to decide what kind of research to carry forward, I think becomes all the more crucial. And for an institution which is mandated to carry out translational research, this becomes a continuous endeavor with the effort to move on from one endeavor to the other. So we need to, I think all the more reason that we need to have Indian data, like Indian solutions for Indian problems, Indian treatment for Indian diseases. And that is also where institutions like this, I am glad that now the younger faculty at least is trying to have some aptitude into translational research work. Most of the earlier ones, we sometimes had difficulty in finding the right kind of people because there's not much. In fact, now that we've started, I've started programs integrating the public stakeholders with some of our institutions, which of course is not so relevant here, but at least in the CSIR. And so one of them, we have made a mark in what is now come to be known as Aroma Mission, the new genre of agri startups. So I told the director, they said, why don't you have a program with the agri startups, with the farmers? And I was astonished. Most of them, the audience was 500 of them, they were entering that place for the first time. So I said, we'll not talk, we'll let them talk. And they made observations which were not only hilarious, also eye-opening. So one of them said, sir, हम यहाँ से गुजरते थे, अब लगता है तो अंदर कोई रिसर्च हो रही है, मेढे वेढे काटते हैं लोग. तो हम सोचते हमारा तो लेना देना नहीं है. I hope that kind of reputation doesn't exist outside the gate of this institution. And one of them, in a lighter way, said, हमें तो किसी ने ये भी कहा कि वहाँ इतने वो मार काट का काम होता रहता है अंदर के रात को भूत नाचते हैं. So the point is, and I'm not saying this out of context. This happened just about 6 months back. This anecdote I'm telling you about, and one of the leading institutes. So I'm also saying so that I'm glad that today we have had a good integration of these MOUs with the industry. At least they know what we are doing. And it's not only important from the point of making them understand that, look here, there are no medics being carted or no ghosts happening. It's not only that, it's also because unless we integrate the stakeholders, how are we going to move forward? The government can't be funding everything. It's not possible. Unfortunately, we have this gossip habit of saying, देखिए साहब अमेरिका ये कर गया हमने. So when we go for the morning walk, you would say, ना क्या किया जी, रॉकेट भेजा वो. अमेरिका ने तो 50 साल पहले भेजा था चांद पर. But they don't understand that NASA has more than 60-70% of resource from the non-government. And you are expecting for the government. And you are drawing a comparison which is not parallel because you are ignorant. So that is morning walker's opinion, because they don't want to feel bored walking 1 hour or so. And that carries the day. That becomes the talking point for the day, in the canteen, in the office. So if we involve these people, then I think we can get over that, and we can also elicit their cooperation. And as far as I'm glad under this government, Prime Minister Modi has been very supportive. We have been very openly engaging industry, and I'm very clear about this, that we need to have a very strong industry linkage even for a successful, sustainable startup movement. Otherwise, it starts up, but you don't know where it has ended up because you're not keeping a track of it. So you have to sustain that. And therefore, more and more of integration between public and private sector is something I'm sure if Dr. Bhan was around today, he would have also appreciated, approved of, because that is the way forward. And also particularly because now India is also being looked upon as one of the cost-effective or relatively cost-effective destinations for healthcare research and also for healthcare management, both preventive as well as therapeutic. And that's why now you have a huge amount of health tourism happening over here. So our responsibility increases manyfold. And also because now we are almost at par with any other country of the world. It's no longer that we were behind about a decade or so, and it reflects in every sphere of science. The other day I was addressing the students in one of the universities. I said the simplest example is that television happened in America in 1950s. Every household had. And the famous election of 1960, where President Kennedy turned the tables on Richard Nixon and won the election, happened because of the oratorial skills which he demonstrated in the TV debate, which the presidential candidates have, as you are having these days. But here we didn't know what is TV about, what is debate about, which happened in India about more than a decade and a half later with some Chitrahar or something. But today it is no longer so. And similarly, even in the medical field, any VIP being advised by one of his seniors for a heart bypass would try to arrange one paid trip to Houston. So Houston became famous in India for cardiac bypass. So the point is that it's no longer so. You have all that facility over here, both in terms of research as well as management. And it is being borne out by the fact that you have people from other countries also coming here. So I think in a scenario like this, where it is one of the better times happening from the point of view, and there's also a certain amount of dignity, esteem being accorded by the government, by the political leadership, to the researchers, to the scientific fraternity, and the meaning of the translational health science also has begun to dawn. Because I'm sure when it began, people must be wondering what is all this translation happening about? What are they going to translate into what? So with this kind of scenario and a supportive policy-making machinery available, this is one of the times when we can make the best of it. We have certain plans, as Rajesh was saying. Actually, I am more impatient than him because the minister's time is, term is limited always. So it's just incident I came back now. So I was pushing for one or more two things. Then he said, no, sir, wait, we need to have a cabinet note for that. I said, okay. So yes, we are in a hurry. We are in a hurry because we have already made ourselves at par with the rest of the world, and we look forward to move beyond this. And as these days, everybody keeps talking of India 2047. So naturally, that is the legacy. Many of us may or may not be around because, don't go by the color of the heads because every kind of color is available in the market now. but the truth is that that is the kind of legacy that we have to leave. So I think, and we are now have been demonstratively accepted. I was in Berlin, before I conclude that I was really esteemed. I called upon some medical startups, and I found most of the startup groups had Indian team members. And in a place like Berlin, where you don't have many of Indians, I mean, it's different in UK. So I asked one of the seniors there, I said, why do you prefer Indian boys and girls? So they gave me a series of good reasons. They're hardworking, they slog. I said, of course, we taught to do that. This, that. And in the end, they said, lastly, but not least, they don't take a weekend off. So we have certain advantages with which our parents gave us birth. I think now this is the time to vindicate that and prove that we are better than others. And I'm sure we have a bright future. And when we meet next time on this day, we would have moved much further than where we are today. Thank you very much.
Thank you, sir, for your valuable message. I now request Dr. Karthikeyan to kindly felicitate Dr. Jitendra Singh ji. Achievement and success are not by accident. Dedication and commitment serve as a vital link in the growth of an institution. So as a symbol of our appreciation for your dedication and loyalty, we present and commence the award ceremony. The first category in awards are the Dr. M.K. Bhan Group Award for the most impactful collaborations. And the winners are Dr. Ramandeep Singh, Dr. Nishit Agarwal, Dr. Dinesh Mahajan, Dr. Shailendra Asthana, Dr. Yashwant Kumar, Dr. Saqib Kidwai. They are receiving this award for discovery, design, and development of novel therapeutics against drug-sensitive and drug-resistant tuberculosis. We recognize this year the TB drug team in the anticipation that its team will take the identified potential TB drug candidates from bench to bedside and generate funding for doing the relevant research in the TB drug development. The next award is an award for a faculty for the best published paper. And the award goes to Dr. Amit Awasthi, Senior Professor, and Dr. Srikant Sadhu, Research Scientist. The next category is the best PhD student. And the award for a PhD student for best published paper is for Ms. Tanshi Mehrotra. Award for a PhD student for the 5 years all-round performance goes to Ms. Garima Arora, Ms. Upasna Madan, Ms. Shabnam Ansari, and Ms. Pragya. Award for a faculty for all-over contribution to the institutional development during the previous financial year goes to Dr. Pallavi Shetrapal, Associate Professor, and Dr. Sweeti Samal, Principal Scientist 2. Award for the administrative staff for all-over contribution to the institutional development during the previous financial year goes to Ms. Rajni Verma, Section Officer, and Mr. Sahil Monga, Data Entry Operator. The next award is for the technical staff for the all-over contribution to the institutional development during the previous financial year. And the award goes to Mr. Sandeep Goswami and Mr. Ajay Kumar. We now invite Dr. R.P. Gaikwari and Mr. Mahinder from High-Tech Biosciences to exchange the letter of intent for microbiome modulation and biotherapeutics.
I would now like to invite Dr. Nitya Vadva, Senior Professor, BRIC THSTI, to kindly deliver the vote of thanks.
Namaste. On behalf of BRIC THSTI, we would like to extend our heartfelt gratitude to our Honorable Minister, Dr. Jitendra Singh, for his gracious visit today, his encouraging words, and his unwavering support. Thank you so much, sir. We extend our sincere thanks to Dr. Rajesh Gokhale, Director General BRIC, Secretary, Department of Biotechnology, for his constant guidance and support. Thank you, sir. Our special thanks to Dr. Srinath Reddy, Honorable Distinguished Professor and ex-President of the Public Health Foundation of India, for delivering the Foundation Day address, and Dr. M. Srinivas, Director of the All India Institute of Medical Sciences, for his insightful address. We are deeply grateful to Professor Pramod Garg for his constant support and for being here today to celebrate on our 15-day Foundation Day. We are deeply grateful to Professor Arvind Sahu, the Executive Director of RCB, for his consistent support and cooperation. Thank you, sir. We are also grateful to Dr. Krishanu Rai, Director of NBRC, and Dr. Shubhra Chakrabarti, Director, NIPGR, for gracing our occasions. A heartfelt thank you to Professor Ganesh Karthikeyan, our Executive Director, for his invaluable guidance and supervision, without which this entire program would not have been possible. Thank you, sir. We extend our gratitude to all the respected delegates from the ministry, and from DBT, Dr. Jyoti Logani, Dr. Varshnya Singh, for being here today, and for taking time out to participate in this event. We thank our esteemed guests and partners from the industry who have taken time out and have been with us for over these 2 days for the outreach program and also in for our Foundation Day celebrations. We are also thankful to all our previous directors for their significant contributions to THSTI's journey. Our heartfelt thanks to all our mentors and collaborators, as well as to all the invitees who have joined us virtually. There's anyone. We sincerely thank all the committee members involved in organizing today's program, especially the staff and student volunteers who have worked tirelessly over the past few days. Lastly, on behalf of the organizing committee, I extend my deepest thanks to the entire BRIC THSTI family for making this program a grand success. Dhanyavad. Thank you.
The Honorable Minister will now proceed to inaugurate the facilities on the second floor.
We kindly request all the guests to stay here. Thank you, sir.
Thank you, sir.
I kindly request all the guests to please stay here because all the inauguration of the facilities will be telecasted live here. Please be seated.
The GEHUMIC is a unique collection of more than 125 bacterial and fungal species isolated from different sites of the human body and also from the environment. Multiple isolates of each microbial species have been identified based on their 16 sRNA gene sequences, tested for antimicrobial resistance, potency, and decoded into their whole genome sequences. A subset of microbes were used to explore their proteome under optimal growth conditions. The primary goal of this facility is to develop a repository of genetically defined microbial cultures, providing authentic microbial cultures to scientists in research institutes, universities, and industries for research and development. The user will have the opportunity to use these indigenous isolates for their own research, program, and commercialization following THSTI's terms and conditions. As we can see in the LCT, Honorable Minister Jitendra Singh Ji is heading towards genetically defined human-associated microbial culture collection center, GEHUMIC. GEHUMIC is a unique collection of more than 125 bacterial and fungal species isolated from different sites of the human body and also from the environment. Multiple isolates of each microbial species have been identified based on their 16S RNA gene sequences, tested for antimicrobial resistance potency, and decoded into their whole genome sequences. A subset of these microbes were also used to explore their proteome under optimal growth conditions. The primary goal of this facility is to develop a repository of genetically defined microbial culture collection, providing authentic microbial cultures to scientists and research institutes. As we can see in the LCD, Honorable Minister Jitendra Singh Ji is now heading to inaugurate the Experimental Animal Facility at THSTI. Experimental Animal Facility EAF at THSTI has been upgraded with the support of DBT with the goal to develop innovative and advanced animal model platform for preclinical research in the area of both communicable and non-communicable diseases. This facility is one of the largest small animal facilities in India that has contributed immensely towards developing animal model platforms for the research in autoimmune diseases, infectious diseases, cancer biology, gut inflammation, cardiovascular diseases, hemolytic diseases, neurosciences, muscle development, and disorders. The facility is equipped with state-of-the-art laboratories such as mouse germplasm cryopreservation laboratory, transgenic mouse production laboratory, histopathology, and quality control laboratory. The facility is equipped with modern instrumentation such as rodent CT scan, live imaging, clamps, and gamma radiation. The facility will serve as a nodal resource center for preclinical research that will foster national and international collaborations between academic institutions, hospitals, and industries.
With this, we finally come to an end of the celebration of BRIC THSTI's 15th Foundation Day. We hope that we'll meet you again next year. On the behalf of BRIC THSTI community, I would like to thank our guests, sponsors, and everyone who helped to make this day possible. We are grateful for your support and we look forward to continue working with you in the years to come. Thank you, everyone, and good evening.
बंद था गाइस अब हरियाणवी डांस के लिए हमारे सामने आ रही है मिस सोनिया हम्पाल सो प्लीज वेलकम हर ऑन द स्टेज विद अ बिग अप्लॉज The performance, I request everybody who is outside the auditorium to please come into auditorium. Thank you very much.
ਸਾਨੂੰ ਦਾ ਨਹੀਂ ਪਿਆਰਨਾ ਬਟੋਲ ਕੇ ਕੰਡਾ ਕੱਢੀ ਦਾ ਸਪੀਕਰਾਂ ਦੇ ਬੋਲ ਕੇ ਸਾਨੂੰ ਦਾ ਨਹੀਂ ਪਿਆਰਨਾ ਬਟੋਲ ਕੇ ਕੰਡਾ ਕੱਢੀ ਦਾ ਸਪੀਕਰਾਂ ਦੇ ਬੋਲ ਕੇ ਕਿ ਗੱਲ ਜ਼ਿੰਦਗੀ ਦੇ ਰੋੜੀ ਨੂੰ ਦੇ ਮੋਢਾ ਮਿਲ ਖੋਲ ਕੇ ਲਾ ਕੇ ਪਿੰਡ ਕਦੇ ਖੁਸ਼ੀ ਦਾ ਬਹਾਨਾ ਰਵੀ ਰਾਜ ਵੇ ਕਦੇ ਪੀਨਿਆਂ ਦਾ ਰੁਝ ਗਮ ਗੋਲ ਕੇ ਨਹੀਂ ਲਾ ਕੇ
Yeah. So guys, she can dance later after the last performance. After the last performance, anybody can come to the stage and showcase their talent once again. But for the last performance, let's welcome Mr. Sunny Raj, our administrative magician. So guys, let's lend our ears to his lyrical musings.
भारत माता की, भारत माता की। सबसे पहले मैं सभी को THSTI की 15 फाउंडेशन डे की तहे दिल से बधाई देता हूं और मैं हाथ जोड़कर अभिनंदन करता हूं उन सभी साइंटिस्ट, फैकल्टी, एडमिन एंड टेक्निकल स्टाफ, स्टूडेंट और हमारे मजबूत लीडरशिप को जिनके प्रयासों तथा नेतृत्व के कारण THSTI ने अपनी पहचान चारों दिशाओं में बनाई है। किसी ने सच ही कहा है, सपने उन्हीं के सच होते हैं जिनके सपनों में जान होती है। सपने उन्हीं के सच होते हैं जिनके सपनों में जान होती है। पंख कुछ भी नहीं होते, हौसलों से भी उड़ान होती है। मुश्किलें हारती हैं, संघर्ष करने वाले जीतते हैं। तो आज की यह मेरी कविता का शीर्षक है, विपत्ति जब आती है कायर को ही कहलाती है। और यह याद दिलाती है कि उन जब हम मुश्किलों में आते हैं तो हमें ऐसा लगता है कि हम हार मान जाएंगे, लेकिन ये कविता आपको डेफिनेटली मोटिवेट करेगी। सच है, विपत्ति जब आती है कायर को ही दहलाती है। सूर्य नहीं विचलित होते, एक क्षण नहीं धीरज खोते, विघ्नों को गले लगाते हैं, कांटों में राह बनाते हैं, मुझसे न कभी कहते हैं, संकट का चरण न कहते हैं, और जो आ पड़ता है सब सहते हैं। रहते हैं सूर्य नींद नसाने को, खुद विपत्ति में छा जाने को। जब विपत्ति आती है तो क्या होता है, ज्यादातर लोग कहते हैं कि विपत्ति आ गई, विपत्ति आ गई। तो है कौन विघ्न ऐसा जो टिक सके वीर हम लेता है जब के जाते पांव दुखड़। मानव जब जोर लगाता है पत्थर पानी बन जाता है। मानव जब जोर लगाता है पत्थर पानी बन जाता है। गुण बड़े एक से एक प्रखर छिपे मानव के भीतर, जैसे लागू है व्यक्ति का रोशनी रह नहीं पाता है, बत्ती जलाता है रोशनी रह नहीं पाता है। जब फूल गिरे जाते हैं हम उनको गले लगाते हैं, जब फूल गिरे जाते हैं हम उनको गले लगाते हैं। वसुधा का नेता कौन हुआ, वसुधा का नेता कौन हुआ, भूखंड विजेता कौन हुआ, भूखंड विजेता कौन हुआ, अतुलित यश का क्रेता कौन हुआ। नवधर्म को मिलता कोई हुआ, जिसने न कभी आराम किया, विघ्नों में रहकर नाम किया, विघ्नों में रहकर नाम किया। और जब विघ्न सामने आते हैं तो जब विघ्न सामने आते हैं, सोते से हमें जगाते हैं, मन को मरोड़ते हैं, तन को झंझोड़ते हैं, और जो नक्शा ग्रह में जलते हैं वही सूरमा निकलते हैं, वही सूरमा निकलते हैं। अब बढ़कर विपत्तियों पे छाजा, मेरे किशोर मेरे ताजा, जो स्वयं तेज भयकारी है, क्या कर सकती चिंगारी है। तो सच है विपत्ति जब आती है कायर को ही दहलाती है, सच है विपत्ति जब आती है, सच है विपत्ति जब आती है। तो भारत माता की, भारत माता की, भारत माता की जय हिंद जय भारत।
So guys, if anybody wants to perform something on the stage right now, stage is open. Anybody guys, please?
Music if anybody is interested in dance.
Okay, so for the end, we are playing some music. If anybody wants to dance and enjoy, so please, they are welcome.